Limitations live only in our minds, but if we use our imaginations, our possibilities become limitless. A very good morning and a warm welcome to our chief guest, Professor Dr. C.P. Joshi, Professor and Chair, Department of Biological Science, Michigan Technological University, USA. Guest of honor, Professor Dr. Jargot, Professor Emeritus, Laboratory of Statistical Genetics, the Rockefeller University, New York, USA. Honorable Chancellor and Chairman AUMP Dr. Asim Chauhan, Honorable Vice Chancellor Lieutenant General V.K. Sharma, Pro Vice Chancellor Professor Dr. M.P. Kaushik, Dean Academics and Director AIB, Professor Dr. R.S. Tomar, Deans, HOIs, faculties of AIB, and other institutes of AUMP, learned delegates from corporate and academia, and my dear students. I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, feel privileged to welcome you all on behalf of Amity Institute of Biotechnology, Amity University, Madhya Pradesh, Gwalior, to the second international conference on recent advances in biotechnology and nanobiotechnology, that is Into Bio Nano 2022. Today, we all gathered here as wisdom comes not from age, but from education and learning. As a genius said, the aim of education is the knowledge, not of the fact, but of the values. It is on the occasion like this, we get opportunity to test our knowledge and understanding. We look forward to get an exposure about what the best of the brains think about emerging techniques. To make this event a blessed one, we invoke Goddess Saraswati by Saraswati Vanna for seeking her choicest blessings.
शुभं कुरुत्वं कल्याणं आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणं आरोग्यं धनसम्पदः.
May I now request Professor Dr. M.P. Kaushik, Pro Vice Chancellor, Amity University, Gwalior, and Chairman of the inaugural session to kindly give opening remarks. I feel immense pleasure to introduce you, Professor Dr. M.P. Kaushik. Dr. Kaushik completed his post-graduation from Jivaji University in 1977. Then he joined Defense Research and Development Establishment, Gwalior, to work for his PhD under the supervision of Dr. R. Vaidyanath Swami. This collaboration continued during the time here as scientist from 1978 to 1986. In 1987, then he moved to the Utah State University, Logan, USA as visiting scientist in Professor Karen W. Moore's group. In 1988, he came back to India and served DRDO in various capacities. In 1993, he became full professor in MIT's Gwalior and served there till 1995 as professor and head of chemistry department. In 1995, he rejoined his parent organization and served as head of process technology development division and as program director, NBC DRDO, and took over as director and outstanding scientist H of Defense R&D Establishment Gwalior and served there till October 2014. In April 2015, he joined Amity University Gwalior as Pro VC and Professor of Pure and Applied Sciences. His contribution has been recognized with several awards such as DRDO Cash Award, Defense Technology Spin-off Award, National Technology Award, Technology Group Award, DRDO, Scientist of the Year Award, and Agni Award for the Self-Reliance. Sir has also published more than 181 national and international publications in various journals of repute. May I now request Professor Dr. M.P. Kaushik for his opening remarks.
Thank you very much, Dr. Pallavi.
Thank you, sir.
Honorable Asim Chauhanji, Chairman and Chancellor, Amity University, Madhya Pradesh. Professor Dr. C.P. Joshi, Professor and Chairman, Department of Biological Sciences, Michigan Technological University, USA. Professor Dr. Jargot, Professor Emeritus and Head of Lab, Rockefeller University, New York, USA. Honorable Lieutenant General V.K. Sharma, AVSM retired, Vice Chancellor of Amity University, Gwalior. Dr. Anil Vasist, Deputy Pro Vice Chancellor, Dr. R.S. Tomar, Head of the institutions, all the panelists, and my dear participants. Very good morning to all of you. It is with great pleasure I warmly welcome Professor Joshi and Professor Ott and all the dignitaries and all the participants to the second international conference on recent advances in biotechnology and nanotechnology. We are happy to announce that this is the second conference in series in biotechnology and nanotechnology, which we are organizing at Amit University, Madhya Pradesh, with all your cooperation and all your support. As you all know, that biotechnology has become an important field in the modern era with various applications. And innovative and cutting-edge research in the field of largely impacting the society in a positive manner in all aspects, including human health and environment. In current pandemic scenario also, biotechnology has played a major role towards managing the COVID-19 and including rapid and accurate diagnosis and vaccine development. And India has played a very important role in vaccine development and to managing the COVID-19, which I think has been appreciated at all that level. And this was possible only because of the biotechnology development which has taken place in the recent past. And because of new and innovative research in biotechnology, the doctors in surgical room can now operate on patients remotely from their computer screens with guiding robots' arms to an accuracy of a few nanometers. This is all because of the biotechnology development. The genetic laboratories equipped with the DNA, splicing enzymes, and more sequence of a polypeptide chains can make wonders happen today. The entire genetic makeup of human beings can be deconstructed into an understandable genetic code. Similarly, nanotechnology and biotechnology at the same time, the medical biotechnology has moved forward towards leaps and bounds in the last few decades. And some of the biotechnological breakthroughs which have taken place recently in medicines are, which has inspired me and which has motivated me to talk today in this international conference, is the stem cell research, human genome project, targeted cancer therapies, 3D visualization and augmented reality for surgery, human papillomavirus vaccine, face transplantation, which is possible right now in case of the persons who have got injured or because of the fire accident or because of any mechanical injuries. The CRISPR, that is CRISPR, is a technology that can be used to edit genes, 3D printing organs. So these are some of the new technologies, or what I should say, the biotechnological innovation which has taken place in the recent past and which has impacted in order to improve the quality of life. For example, stem cell research. Imagine an individual with a degenerative spinal disorder that severely impacts the quality of life. And with the help of stem cell research, it might be possible to grow these stems in vitro in a lab and then implant back into the affected individual body. This would help restore their cognitive acuity, vision, hearing, and other physical features. This may sound far-fetched today and like a plot from a science fiction movie, but the preliminary results has been very promising and a lot of research is going on in this direction. Similarly, target cancer therapies. Cancer therapies, for example, 2-deoxyglucose, which was developed by the DRDO, and I was one of the inventor. No doubt this particular 2DG was developed initially for the cancer, but it has played a very important role in case of managing the COVID-19 patients also, because the oxygen deficiency was there, and this 2DG has helped in a very big way in reducing the burden of the hospitals by having less amount of oxygen which is being required for the patient, those who have been suffering from the COVID-19. Currently established standard chemotherapies are toxic for human healthy cells. Targeted cancer therapies are drugs that works either by interfering with the function of a specific molecule or by only targeting a known cancer cell. In order to minimize the damage to the healthy cells, this is the 2DG which we developed, because it will selectively kill the cancer cells and not the others, the useful cells which are there in the body. And in that case, it improves the quality of life. No doubt, I don't say that 2-deoxyglucose can be used for cancer patients, or it can treat 100% the patient tumor, but certainly it will improve the quality of life. Similarly, the human papillomavirus is one of the causative agents of cervical cancer, and it is the second most cancer in women, second only to breast cancer, killing 275,000 women worldwide every year. Therefore, successful HPV vaccination is considered a major medical accomplishment because of the biotechnological and nanotechnological development which have taken place in the recent past. The face transplantation, the biotechnology has given a new directions. For example, a face transplant is in the process of using skin grafts to replace all and parts of the patient face with the donor face. The first partial face transplant, which was performed in France in 2005, and the next successful transplant was performed 5 years later in Spain. And this was also the first ever full face this transplant which took place, which was they got injured because of human injuries. And the transplant, the patient whose face was severely damaged in an accident received a new nose, lips, teeth, and cheekbones during the 24 hours of long surgery. So you can see that how the biotechnology or medical biotechnology is helping the patients in a very big way. Similarly, another important application which the biotechnology has given in the recent past is 3D printing organisms. The artificial limbs have been in the use for centuries, and these have been steadily improving the mobility and versatility of bionic limbs. Now, new advances in bionic technology and 3D printing have taken it further. It has made it possible to artificially construct internal organs like heart, kidney, liver. Doctors have been able to implement those individuals that need them successfully. And there have been successful examples which has recently come in the news, and it has been the talk of the town that how the artificial heart and kidney are playing an important role. Some of the heart also have been taken from one particular animal, and then it has been implanted to the human beings also in one of the cases. The International BioNano 2022, which we are organizing today, is a unique platform for researchers and academicians to share and discuss their research and innovation toward the societal benefits. Whatever we do today, it should have an application for the public at large. And I'm confident that deliberations by eminent scientists like Professor Joshi, like Professor Oat, and all the dignitaries who are going to deliberate in the next 2 days during this conference will provide a new direction for new research and open new avenues for research in the field of biotechnology and nanotechnology. I wish this conference a great success, and I'm sincerely hope this the technology, all the participants will be getting benefit in a very big way, and they'll become more knowledgeable by attending the conference. And once again, I thank all the eminent speakers, Professor Joshi, Professor Oat, and all participants or dignitaries who are participating in the big way and helping and supporting the MIT University of Madhya Pradesh in successful conduction of this second international conference on biotechnology and nanotechnology. Again, I wish the organizers a great success in managing the international conference. And I want to compliment the organizers for arranging this international conference in such a scenario where there is a need to create an awareness among the public at large. Thank you very much, and Jai Hind.
Thank you very much, sir. Your thought-provoking address set a perfect platform for our speakers to deliver their presentations in their area. I feel immense pleasure to introduce Professor Dr. C.P. Joshi, Professor and Chair, Department and Professor of Plant Molecular Genetics at Michigan Technological University. Professor Dr. C.P. Joshi, he is a leading plant molecular biologist and biotechnologist. He is working toward deciphering the process of biosynthesis of the cell wall in bioenergy trees such as poplars. His current research interests include molecular genetics and genomics of cellulose and lignin synthesis in trees, genetic improvement of lignocellulosic product for bioenergy and paper industries. Dr. Joshi has recently served as a director of a 40-faculty strong biotechnology research center and also was the director of graduate program at the School of Forest Resources and Environmental Science at Michigan Tech. Since 2012, he is serving as the chair of the Biological Science Department at Michigan Technology University. He has authored over 228 journal articles, several presentations, patents, and book chapters. He is a recipient of a highly prestigious NSF Career Award, Michigan Tech's 2011 Research Award, and is an inductee of Teaching Excellence at Michigan Tech. During 2009 to 2013, Professor Joshi visited Chonam National University, South Korea, as a world-class university distinguished visiting professor and participated in establishing a new department of bioenergy science and technology. He gathered over $10 million in funding from various national and international agencies for his research. His current research with the genetic improvement of bioenergy trees has many direct applications in the areas of bioenergy productions, forest and crop productivity, and plant improvement. I request you, sir, to kindly deliver the keynote address.
Thank you very much for the introduction. It's really great pleasure to be here with you all. And I would first of all like to thank Professor Tomar for inviting me for this really world-class occasion and share some of the recent results in my lab. When I looked at the list of different people who are going to give speeches today as well as tomorrow, I noticed that it's really the best research that's happening all over the world. Professor Kaushik just now really very nicely introduced what can happen, what we can do, as well as what we are already doing using biotechnology, as well as biology. I said this particular century is the century of biology. If you look at COVID is virus, which is part of kind of biological system. Our response is biological. The way we are trying to save ourselves is also biological, the vaccines and everything. So every aspect of our response to COVID, you may say, is some or other way connected to biology. So this is really century of biology. And biotechnology is one branch of entire biology, and biology naturally is very huge. So it's really a great time, and I will not be able to stay whole night awake to listen to all talks, but I wish, first of all, my best wishes for the whole conference, and I'm sure it is going to be a very successful event. I will try to share my screen with you. Give me a second, please. Okay. So today I'm going to give a short talk about plant cell wall biotechnology. I will try to show you a few things that we have been doing in our lab recently. And I'm from Michigan Tech. If anybody wants to contact me, that's my email address, cpjoshi@mtu.edu. So I welcome all of you, and I really hope that one day this COVID will be gone and we all can meet together in person, because I do want to meet all of you and share thoughts, more longer interaction than this 30-40 minutes interaction. That's what is going to happen today. So first of all, let's talk a little bit about plant cell walls. And I have made my presentation not extremely complicated. I want to explain everything, and I hope that you all will appreciate how things get complicated from very simple things. So first of all, let's talk about plant cell walls. So why do we care about cell wall? What is so special about cell walls? The cell wall is the main distinguishing feature of plants. Anytime we say how plant differs from animals, we talk about cell wall. So all plant cells, they have a primary wall, and primary wall is thick, it's expanding, and basically it protects the cell. It kind of delimits the cell. Primary cell walls are made up of two types of main carbohydrates, main polysaccharides, cellulose and hemicellulose. There are some cells which are also economically very important, such as wood cells, the xylem, and fibers. So xylem and fibers have an additional wall. And even though I say one wall, in reality, there are up to three layers of secondary wall. So primary wall is present in all plant cell walls, but secondary wall is present in specialized cells, such as wood cells, as well as fiber cells. And there are a number of examples of these secondary wall containing cells. And these are the main things, and they are made up of cellulose, hemicellulose, which are polysaccharides, and polyphenolic lignin. So just to remember right off, primary walls and secondary walls are different, and I'm actually going to talk more about secondary wall today. So if we look at just the cell wall structure as such, there is obviously the plasma membrane, and plasma membrane is a site where where the cell walls are produced. So at that thing in the beginning, the primary wall is made. primary wall gets pushed out, and then inside the secondary wall is made. And these secondary walls are present only in cells like xylem and fibers. That's what we should remember. So how different are these two different cell walls? So if we just look at cellulose, which is present in the primary and secondary wall, the primary wall is thin. It has about 10 to 20% cellulose. The degree of polymerization, the number of glucose units that are present, is about 500 to 2000. Its crystallinity is low, so actually it's very easy to break down if needed. It has the cellulose fibers are in a particular angle we call microfibril angle, which is 50 to 90 degrees. And mainly the function is helping the expanding cell wall as such, or expanding cell. The secondary wall, on the contrary, is thick. It provides the rigidity and strength. It has about 40 to 50% cellulose. The degree of polymerization, the number of glucose units present, are about 14,000. The crystallinity is high, so it's very difficult to break it. The microfibril angle is low, and the function is to provide rigidity and strength. So these two types of celluloses that are present in these two types of walls are completely different in terms of the structure as well as the function. So if we just look at the woody tissues, because that's what I do my main work with. If you look at it, about 50% of the cell wall, or the 50% of the wood, is cellulose. And this cellulose is the main rather material that we want to use for bioenergy production later on. So about 50% is cellulose, 25% is lignin, and another about 20% is hemicellulose. And I'm going to talk about all these three aspects as we go along. So paper and product industries are very interested in woody materials. What they do is they take all these secondary cell walls and they separate cellulose from lignin and hemicelluloses. Then they treat it with caustic chemicals that are expensive, that spoil the environment, and the byproducts are very stinky. That's why if you go around paper product companies, you always get the bad smell, and that comes because of the delignification process. So ideally, if plants or if trees have less cellulose, or less lignin and more cellulose, it's going to be good for paper and pulp industry. There was a calculation done that suppose you reduce lignin in the cell walls by 1%, and normally, as I said, we have about 20-25% of lignin, and everybody uses our trees, $1 billion could be saved every single year. So you can imagine what is the capacity. So this is not just a pipe dream. This is not just a theoretical thing. This is very practical, and we can actually change the world in a small little way by utilizing this technology that is being developed. The other use is, so all trees, all plants, they sequester carbon, and that's really good. And the carbon is actually inside the cell walls. That's really good for protecting us from global warming, because the amount of carbon dioxide remains outside is not good. It's good to put it in cell walls and preserve it. So that's one utilization. The second utilization is you can use the same cell walls to produce bioenergy. That's what I'm going to talk about more today. So what is really bioenergy? Any energy that is produced by biological organisms. So when you push a car, that's also really bioenergy you could define. However, when we say bioenergy, it's used in a very specific term, that we take the biomass, the plant biomass, we convert it into biofuels. So taking biomass and releasing its energy and convert it into biofuels is basically the bioenergy research. What do we want? We want better biomass that will be produced, and we want more biomass. These are the two aspects I'll be talking about in my today's talk. So biofuels can be of two types. One is the bioethanol, which we put into our car along with the gasoline or petroleum, and we drive the car naturally. The other is biodiesel, and biodiesel is produced by a different pathway. It comes from fats and oils, and by transesterification, you can produce biodiesel. And I'll be speaking both a little bit about bioethanol and also about biodiesel production in my speech today. So Question is, why do we need bioenergy? The number of reasons are: number one, we are running out of petroleum or gasoline, as we say. So if you look at this particular graph, we actually peak the amount of this gasoline or crude oil that we can produce. In year 2000, we already peaked. In about 200 years, we almost took most of the crude oil or most of this petroleum that is present underground. And now it is going to be only a downhill. And by year 2200, there'll be hardly any of the underground fossil fuels will be left. The other important aspect of bioenergy, so this is one problem. We got to find alternative to gasoline. The other thing is the import. There are a few countries that have this crude oil and they export it. And most of the other countries import it. So if you look at this particular second part of the picture, so every single day, 7.9 million barrels of crude oil is imported by United States. And then there is obviously India, China, name it, all these countries, and the numbers are written, they are importing this gasoline. So we are dependent on some other countries to provide the gasoline or the petroleum. The third important reason we got to do is we have to reduce our footprint. And if you look at this footprint, how much this carbon footprint we are producing, United States is producing a huge carbon footprint. China is the next. That's where India is, Japan and South Korea. And obviously all countries are there. So we all are producing greenhouse gases, and we all must do something to reduce these greenhouse gases and reduce our footprint. Again, bioenergy could help us in reducing this particular footprint. And last but not least is the population growth. We are about 8 billion. We are going to be 10 billion by 2050, and we are going to grow. As the population grows, the demand grows. We have to do something to find alternative to the gasoline or the petroleum, as we say. So we are really speaking in biofuel revolution. We are running out of gas, so we got to do something. We're addicted to foreign oil, and we are dependent upon it. Our economy and our security depends upon it. And that's why different governments and oil companies have started putting billions of dollars in finding out alternative to the petroleum or gasoline. What kind of alternatives can we have? So the first generation bioethanol was produced in Brazil by using sugarcane. So you just squeeze the sugarcane, get the sugars, ferment them, get ethanol and use it. And this really became extremely successful for a while. Countries like USA and European Union, we don't have sugarcane. So we went with corn. Obviously, corn has utilization as food and feed. So that was not the best utilization. The way Brazil came in trouble because they have to sell their sugar to countries like India and China. And now they have to import bioethanol from another country. So it's a very weird kind of problem was going on. And that's why basically Brazil using sugarcane, yes, they can do it, but it's not really the best solution that we should be using food crop to produce the ethanol. The corn has the same story. Yes, we have it, yes, we have plenty, but still it's a food crop and we have to do something. So people started looking for alternatives, and that's where the second generation cellulosic bioethanol came. And the reason is it could be abundant, it is non-food, and it is sustainable. So politicians obviously help us in bringing money as well as setting up the goals. So in 2007, our US President Bush proposed that by year 2022, 36 billion gallons of renewable fuels will be produced every single year. And in 2007, we thought 2022 is really far away. We can do a lot of research and we can reach. Guess what? We are in 2022 now. So we should be having 36 billion gallons of this renewable fuel. So question is, are we there yet? Here are the numbers where we are today. We do produce 50 billion gallon tons of conventional first generation bioethanol, but only 36 million gallons. Remember, there's a billion and million. So this 36 million gallons of second generation bioethanol has been produced from 2012 till 2020. Why? Because the problems are big and we are trying to solve one of those problems. So I'm not saying that this is hopeless. And in fact, our President Obama has said something very nice. He said, we have to acknowledge the progress that we have made, but understand that we still have a long way to go. So that things are better and still not good enough. And that exactly is the sentiment about this second generation bioethanol. So in order to make this bioethanol, we are going to need all these cell walls, all this biomass. Where are we going to find? There are about 4 billion hectares of forest. There'll be leftover things from forest. I never say that go and bulldoze the forest. Naturally, that's not going to be good. So the leftover pieces, when you cut trees, there'll be sawdust kind of things will be left, we could utilize. So we got to look for renewable resources to do this particular work. If you look at the plant biomass, 50% of it is cell wall. So if we can use cell wall, if you can modify cell wall in such a way that we can produce ethanol out of it, or biodiesel out of it, is going to be really very useful. So considering that, in Michigan Tech, I have been at Michigan Tech for the last 25 years. And these are the things we have decided we would do. First of all, we'll focus on secondary cell wall production, formation. We'll focus on trees, although we will use the model plants we needed, but we want to do work with poplars, eucalyptus, and pines. We'll focus on lignin, cellulose, and hemicellulose, which are the 3 components of secondary wall. We'll clone a lot of genes, characterize them, and modify them in such a way that we will produce the real trees that have been genetically engineered. So basically what we do is we genetically engineer trees to make them better for bioenergy production. So the question is, can we really do that? That was the first question came in our mind. So the way we did it, these were our goals. In the beginning, we focused on lignin, the one which is shown here in blue color. And our goal was to reduce lignin from 25% to maybe 15%, maybe 10%, whatever we can do. We want to alter the type of lignin that's produced. There are 2 types of lignin. One is called G lignin, S lignin. S lignin is easy to take out. G lignin is difficult to take out. So try to improve trees in such a way that there'll be more S lignin and improve the cellulose production. Increase the cellulose, amount of cellulose that's produced, and reduce the hemicellulose. That's what we wanted to do. Just to say why we care about the cellulose, because cellulose is really chain of glucose. So if you take this chain of glucose, and if you break them, the glucose will be released. We can ferment the glucose, produce bioethanol. That's basically the logic. If we look at the plant cell wall, they're made up of cellulose, covered by hemicellulose and lignin. If you look at the cellulose, it's a wonderfully organized structure. And it is so wonderful that it is very difficult to break the bonds in between them. So ultimate goal is to separate these glucose molecules and use them for bioethanol production as much as we can. Once we do that, so this is the stage 1, take a tree, take a plant, take biomass and harvest it, chop it, cut it in smaller pieces, pre-treat it, then obviously release the sugars. That's called saccharification. Ferment this using microbes. Microbes will produce alcohol and use this alcohol, go wherever you want and add it with the gasoline. And in the United States, we have about 10% ethanol is added. Whenever we go to our petroleum pump, as we say in India, here we call it gas pumps, there is already 10% ethanol there. So this is the basic process. And we focus on step 1. Why not to modify the trees, the plant biomass itself, so that we can produce better product at the end. So in general, just this is the theme. I always say we need to know how plants make it so that we can break it when we need. So it's a complex cell wall, basically, but cell wall has cellulose. We treat it, release the glucose, ferment it, and produce ethanol. That's basically the scheme we have. And so in order to go to cellulose, get the cellulolytic enzymes to cellulose, you have to go through hemicellulose, which surrounds cellulose. That is surrounded by lignin. So you got to go in. And the way you can do this by reducing amount of lignin, or reducing the type, or changing the type of lignin, reducing hemicellulose, but make cellulose accessible to cellulolytic enzymes so the sugars can be released. And that is basically, those are the 4 targets we have. We want to modify lignin, then modify hemicellulose, modify cellulose, and finally improve the cell plant growth. I'm going to give you some examples of this work that we have been doing. The tree we work with is poplar, and this is very economically important species. There are about 30 species, and in terms of trees, they're fast, 4 to 5 years they take to regenerate, and it's a small genome, and it's a bioenergy tree. So what we have done at Michigan Tech, we have produced all this series, and we can produce the trees from a single cell. So this is what I will show, the transgenic trees that we have produced. I'll just tell you a little bit story about lignin. We cloned all the enzymes that are present in lignin pathway, and we modified them. I'll give you one example for SIA. This is one enzyme we modified. We produced trees that are reduced in lignin 45%, fast growing, and more cellulose produced. If we can take these trees and look at the bioethanol production, they always have more sugars released. And this came in 2007. And from then, basically, people are saying we can modify trees in such a way that they can produce more sugars at the end. So this is, again, I'm showing the same scheme, change the biomass so we can make more bioethanol. So here is one paper we had published in 2013 to show that in virtually each step was changed. In each step, the gene was modified, it was suppressed, the lignin changed, and more sugars were released. As you can see, in each stage, more and more sugars have been released. So we are currently working on a paper. There are about 119 papers published in this field, and number of species used, number of ways is done, the promoters change. The lignin can be reduced up to 52%. The sugars released up to 350%. And basically, you can produce more ethanol if you have more sugar. So this is very exciting field of research that's happening. And currently, we also want to see, it's great that if we can produce 3 times more sugars, but then what happens to the plant? Sometimes the plant doesn't like it. It gets sick, it breaks. So we want to make sure that we don't reduce lignin so much that it will be detrimental to the plant, but it will still produce more sugars. And so that's the project. We want to alter the lignin that's present. And there's some examples I want to give you of the work that we have done. We worked with 2 different genes. One is lactase and other is peroxidase. These are involved in polymerization of lignin. We chose about 4 genes at the end. It's a multi-gene family. And I'm just going to show you some results, how we have done it. So we put this gene under a promoter that drives the expression only in xylem. So it's specifically done in body tissue. Here I have shown very quickly how we do transformation. We take the agrobacteria, that's where the gene is cloned. We take the pieces of leaves, we cut them, we co-cultivate them. After they grow for 2 to 3 days, the gene that we are trying to put goes into the plant. We wash them after a few days, put on selection medium. That's how we grow with antibiotics. The callus is developed. We're going to shoot elongation medium. Then the shoots are produced. You can see these tiny shoots. They go to root, make sure the DNA is present. The plants grow them into the greenhouse, and finally, that's what I call transgenic forest that we produce. And this we can do with virtually any gene that we want to work on. So from a single cell, we can produce the entire tree And then we can actually, by the way, we have tested this not just in greenhouse, but also outside. So few examples. So this is an example of peroxidase 25 that we modified. Here is what happens when you try to do something, plant does different. We're trying to overexpress it, plant did the post-separation and actually reduce the levels of peroxidase. So you can see, instead of going up, it went down. We checked the enzyme activity, it was reduced tremendously. And then we checked whether lignin content was affected. We didn't as much change. Rather, it was not very conserved there. The S2G lignin ratio I told you earlier, that was reduced. But the most important result is we could release more sugars, 39 to 65% more sugars were released. So this was very positive data. Although the gene was reduced in expression, the sugars released were more. We looked at the other gene, the LAC27, the opposite thing happened. So everywhere the expression went up, the enzyme activity went up. We actually looked at the xylem produced, the wood produced. There was more wood produced in these lines. And you can see here, all the transgenic lines had more wood was present. The lignin content was reduced, which is always very good. When that happens, we almost are sure that sugar is going to be released. The SGA ratio was not changed as much, but there was 28 to 45% more sugar release. The most important question was, okay, you have more sugar in the plant, or more sugar is released, but are the plants normal? There was no change in the phenotype and growth. All plants looked perfectly, actually they're indistinguishable from the wild type or from the control. So the conclusion of this part of work is lactases and peroxidases we can manipulate, and they can increase saccharification efficiency without affecting tree growth. So more sugar has potential to make more biofloc. What about hemicelluloses? So this work has been done in another lab as well as in our lab. If you reduce the hemicelluloses, here you can see this 30 to 40% increase in saccharification. So this was done in a gene called 47C. In our lab, we have done, when I was in Korea, Dr. Raju and many others in my lab, basically they did this particular work. We used to mix our virus-induced gene silencing at that time, and we released 40 to 75% more sugars from these plants. And obviously we did this stable transformation, and this was the same thing we observed afterwards. From there, and perhaps I should say a word about it. So this was DUF 579 involved in xylem production, but NAT7 was a transcription factor. And when we saw this, we wanted to see what happens in really populars. So very recently, this paper just got published, I guess last month or 2 months back in Frontiers of Plant Sciences. So here we modified NAT gene expression. So NAT7 is a transcription factor. We overexpressed it in plants. We underexpressed it, we suppressed it, and also we put Arabidopsis gene into popular. So those were the 3 experiments we did. And in all these cases, the lignin was, when we suppressed it, lignin was reduced, not much change was there in lignin, but in all these cases, the S to G ratio was increased, which is an indication that there is possibility that more sugars will be released. And that's exactly what happened. About 24 to 58% increase in saccharification happened as compared to all the controls that we used in this case. So genetic manipulation NAT7, again, could be one of the strategies to improve biofuel production in these plant circles. But the question is about cellulose synthesis. Ultimately, cellulose is the good guy. Cellulose is what we really want and modify. So here are some questions. Not all questions we answered. Something interesting does happen when you do experiments. Can we increase the amount of cellulose? Can we change the property? Can we reduce the crystallinity? Can we change the degree of polymerization? Can we change microfibril angle? And can we really keep more sugars in the plant? Those are some of the questions you can target. So we want to understand how trees make cellulose. So again, my theory is if you know how they make it, you will be able to break it when you need it. And this will be important for paper and biofuel industry. If you look at the cellular synthesis, 3 important genes are involved. The sucrosynthase provides UDP glucose. The cellular synthesis makes cellulose, and Corrigan cellulase actually edits and makes sure that the good cellulose is produced at the end. So we took multi-pronged approach. We cloned all the genes from popular Arabidopsis, name it. We took the genes, modified them, studied them, and see what impact it makes on the plant. So here's an example of sucrosynthase. When we overexpress it in Arabidopsis, more biomass produced, okay, more activity, more biomass, and more dry biomass. Was there more cellulose? And obviously we want to see this in trees. Same thing that happens in Arabidopsis happened in trees. We got basically more leaves, taller plants, thicker plant, and also 3 to 8% increase in cellulose content. So a single gene transfer, again, that's what I'm trying to say again and again. You can transfer a single gene sucrosynthase that will change the structure of the plant. It will look nice, but at the same time, it will release more, it will produce more cellulose, and it will be making more biomass. So in one shot, you can get 2 different types of results. Then we thought, why not to modify cellulose synthase? And we thought cellulose is simple, right? So why not to try? However, cellulose is complicated. I always say these are the 3 different tiger cubs you have to tame in order to control cellulose biosynthesis. So there is one cellulose synthase of one type, there are 2 of the second type, and 3 of the third type. Is 6 things together have to come in a particular format that we call rosette, and then only cellulose will be synthesized. So if we look at this, it's a pretty complicated story to really modify. However, we found out the 3 genes that are involved in secondary wall formation. We call them 478. They are present at the right place, at the right time. They are co-expressed. So we knew that we had to modify most probably these 3 genes to get the final result. By in situ hybridization as well as immunolocalization, we proved that these 3 genes are present at the same time, at the same place, and they affect secondary cell wall production. So we said we have to modify all 3 at once. That was my NSF career project. And our idea was we will clone these genes, which we had cloned actually, we'll express them together. So if we put these 3 genes together in the plant, and we can easily separate out 1 gene, second type, third type, 2 genes at a time, 3 genes at a time, then we'll be able to see the impact it makes. However, before we did this, something else started happening. And basically we produce trees that make no cellulose in their secondary walls. Basically they're devoid of cellulose. This is not what we wanted, but this is what we got when we did this work. So just very quickly, I'll try to tell you the story. So normally when you put trees in, we produce trees, they grow straight. We saw something weird. All these trees had wavy or weak stems. You can see normal tree grows up. This one is like a creeper, and it actually goes on the ground. So when we saw this, this was a really very weird observation. This, you cannot perhaps imagine, this is a 5-year-old tree, but it's not even a foot tall. So basically, when there is no cellulose made, the tree doesn't grow up. It actually becomes crawling, and it actually spreads on the ground. We said, what happened? We were trying to overexpress a gene. Instead of that, something else happened. Exactly same thing happened. There's a co-separation. So the gene we were trying to make more became less, and that basically changed the structure of the cell wall. So we looked at the xylem. This is how the normal vessel looks like. This is how fiber, the thick fiber looks like. The transgenic, on the contrary, you can see here all irregular vessels. And this irregular vessel is a sign that the wall is not properly made. Look at the thickness of fibers. This is thick, because all these fibers are very thin. We did the SEMs, scanning electron microscope study. That's how a vessel look like. This is irregular xylem, broken vessel looks like. The thick fibers became thin. So something drastically changed in these plants. What about cell wall? how much cellulose was there. These plants did not have enough cellulose. Actually, there was a 75% reduction in total cellulose. And the reason I say there is almost no cellulose made in the wall. They are without cellulose, but we have about 50% increase in xylem, 56% increase in lignin. So we're still excited, something interesting happening, but that was not real either. If you look at the ratios of cellulose to lignin, cellulose to xylem, xylem to lignin, the ratio of xylem to lignin does not change. It only changed with the cellulose to lignin, cellulose to xylem, meaning thereby that the pathway that makes cellulose was shut off, but the pathways that make xylem and lignin continued without taking any break. So in general, we looked at the cellulose fertility, it had only primary wall cells. So we made the trees, not that we wanted to make, but that's what happens sometimes. We made the trees that did not make secondary cell wall at all. The secondary cell was cellulose at all, but it had only primary cell wall. And this would be perfect material to make bioethanol if you want. So basically you can change trees by manipulating the genes that are present in cellulose biosynthesis. I'm going to shift gears very quickly in 5 minutes. I'll tell you another story. Can we make trees that can be used for bioethanol production as well as biodiesel production? So that was the experiment we did, and we published this back in 2016, I believe. And here what we did, we took 2 genes, one called LEC2 and other DG18. And we said LEC2 is a transcription factor that controls oil synthesis. We took it from Arabidopsis, put it in popular and under a xylem specific promoter. Look at the tree, look at the plants that we produced. They were absolutely normal looking plants. So this is one very important thing we want. We do not want abnormal plants. We want plants that look perfectly normal. We looked at the gene expression. They all had, as expected, overexpression, as we call it. LEC2 is a transcription factor. So the genes downstream from them all are turned on. So everything is working out well. We looked at the oil production. These plants, basically, they're making more oil in the xylem itself or in the wood itself. But that is hard to see there. Here is the picture of fatty acids, and we could get as high as 132% increase in the fatty acid production. So we got some plants that were making more, but most of the plants, as you can see, were making more oil because we manipulated the gene that were involved in this, and this is DG80 as well as LEC2. So basically, you can transfer single genes, alter biomass, and change the cellulose production, change the saccharification. You can certainly change cellulose synthesis, and that produces weird phenotypes if needed. You can manipulate genes in oil. They can get more fatty acids released. And finally, plant biotechnology is really a very powerful tool for improving biofuel and paper production. And you can change what you want by doing the experiments and basically fine-tuning these experiments so that you can design a plant that you want to fulfill the things. Now, these transgenics, as you have heard perhaps, are GMOs, genetically modified organisms. They come with the issues that we have. And the issues like they're unnatural, so we have unknown impact. They may need more water, more nutrients, we don't know, but we have to study that always. They might have more diseases. They may become invasive. They may affect economy. So these are the kinds of questions that are still there. But we have to, first of all, see what is possible. Is it really good for the society? Is it going to help the society? And then only we should do it. But biotechnology certainly has capacity to change the things at your will for producing the products that could be used for not just mankind, but for all living organisms in the world. So that's basically the message I wanted to give you. Certainly be happy to answer any questions if anybody has. But do want to thank all graduate students, postdocs who worked with me over the last 30 years or so at Michigan Tech. A lot of people worked with me in South Korea. As I said, I was to go there for 4 years. Number of colleagues and collaborators all over the world and the funding agencies. And I want to thank you all for your attention and if you have any question, I'll be more than happy to answer. Thank you very much.
Thank you very much, sir. The presentation was really very informative and may provide a new insight in understanding the importance of biocomponent like cellulose. Now I request our guest of honor, Professor Dr. Jargot, to enlighten the young minds. Professor Dr. Jargot earned his PhD in zoology from the University of Zurich in 1967 and his master's degree in the biomathematics from the University of Washington in 1972. Presently, he is serving as professor in Laboratory of Statistical Genetics, the Rockefeller University, New York, USA. In 1986, he accepted position as professor of genetics and development at Columbia University and research scientist and director of the Department of Statistics at the New York State Psychiatric Institute. Ott came to Rockefeller in 1996 as professor and head of the Laboratory of Statistical Genetics. He was a merit awardee of the National Institute of Mental Health from 1991 to 2003. He received a Medal of Honor from the German Society for Human Genetics in 2007, the Ming Suang Lifetime Achievement Award from the International Society of Psychiatric Genetics in 2008, and the Allen Award from the American Society of Human Genetics in 2010. He was a professor at the Beijing Institute of Genomics, Chinese Academy of Science from 2006 to 2010, a visiting professor at the Institute of Psychology in Beijing, Chinese Academy of Science, and has been the recipient of 2 4-year project grant from the Natural Science Foundation of China. I now request our guest of honor, Professor Dr. Jargot, to enlighten the young minds and please give their presentation.
Thank you for this nice introduction. I would like to thank Professor Tomar for inviting me to give this presentation. And I very much enjoyed listening to Professor Joshi and his highly successful research. So now I try to share my screen. Okay. So what I would like to discuss today is association analysis with more than just one genetic variant.
Please share the screen. The screen is not visible.
It's not visible.
Yes, sir.
Can you see it now?
Not yet. Not yet, sir.
Yeah, No?
Yeah.
Okay.
So you can
So-
make it full screen, sir.
Pardon me?
Sir, you can make it full screen.
Yeah. Well, is this okay?
Okay, sir.
So science develops independently in different fields. In human gene mapping, we have a long history of localizing disease genes along the human genome. But in an area of computer science, people have developed what's called frequent pattern mining. And what I would like to discuss today is how these two areas are getting together. And we use in human gene mapping techniques from pattern mining. I will give you details very shortly. Basically, when we have two variants, two DNA variants, and let's say they have minor alleles A and T, even when they are on the same chromosome, the frequency of AT chromosomes is generally the product of allele frequencies. That means this haplotype has a frequency which is equal to the product of these two frequencies, which is referred to as linkage equilibrium. But when variants are very close together, this no longer holds. And this refers to linkage disequilibrium. We can assume that one variant is a disease variant. And if a marker variant is very close to the disease variant, then we can make use of this fact that there is linkage disequilibrium. Basically, what we do is we look at genotype frequencies in markers, among cases, people with the disease, and among controls, people without the disease. And if these frequencies are very different in cases than controls, then we have a good reason to believe that this particular marker is very close to a disease variant. And this has been known for recessive traits for quite a long time, was popularized in 1987. Oops. But more recently, it was also shown to be the case in dominant variants. So if we look at a single variant and try to look at the effect of genotypes on disease, again, we distinguish case individuals and control individuals. We'll assume X is equal to 0, 1, or 2 for genotypes, for the three genotypes at this variant. And we define a probability pi of X. This quantity is the logit of pi of X, and it can be expressed in a linear fashion, alpha plus beta X, where X again are the three genotypes. Why is this important? With this logistic regression formulation, we can handle multiple variants, multiple genotypes and multiple variants. So we can measure the combined effect of different genotypes on disease. We can also look at interaction effects by simply looking at the product of two of these expressions. So this would be a possibility using logistic regression for working with multiple variants. However, this would refer to two specific variants. And the question is, what if we don't have two specific variants to look at, or three specific variants? How do we find pairs of variants that are jointly responsible for disease? First, a few examples. This was published 20 years ago. All these 5 individuals, they have a disease, hyperinsulinemia, and they are heterozygous at each of 2 variants, and nobody else in the family is heterozygous at these 2 variants. And I have to emphasize that the 2 variants are on different chromosomes. So here we have a situation where variants on different chromosomes interact to produce disease. This has been more recently described by Schäfer, but this table goes back to Ming and Menke, 2002. A good example is retinitis pigmentosa, RP. There are 2 genes responsible for this disease when they are both present, both mutant variants are present. But if only one is present, the phenotype is normal. And if only the other is present, the phenotype is also normal. So this is a clear case of a digenic inheritance, where two genotypes from two different variants are required for disease. How do we analyze interaction effects? Again, we assume we have two specific variants, variant 1 and variant 2, and there are 3 genotypes at this variant and 3 genotypes at this variant. So we can compute chi-square when we have numbers of individuals here in this 3x3 table. And we can compute chi-square for the controls. And we can also assume there is no difference between cases and controls, so we combine the two and then get another 3x3 table this way. If there is big difference between cases and controls, then we assume that this plus this will be very large. So the difference between these two chi-squares and this chi-square is then referred to as the heterogeneity chi-square. In this particular case, it was not very strong with 40 degrees of freedom. Another way of displaying the data is looking at the 3 genotypes at variant 1, and then within each genotype, look at the 3 genotypes at variant 2. So in this 2x9 table, we can also compute chi-square, which is the chi-square for the total table. But then we can compute chi-square for variant 1 by collapsing these three table columns and collapsing these 3 columns and so on. And that gives us a 2x3 table with 2 degrees of freedom. And we can look at the chi-square for variant 2. So this would be the main effect for variant 1, the main effect for variant 2, and the total table. If we add these up, they will generally not be equal to the chi-square for the total table. What's missing is a potential interaction effect. So this would be another way of looking at the interactions. Now, it turns out it's the same as when we compute this heterogeneity this way. So heterogeneity and the interaction are two sides of the same coin. And you can do this or this, whichever seems more intuitive. Now my main presentation, how do we find disease-associated pairs of genotypes? You can assume we have at least, say, 100,000 variants throughout the genome. Generally, you have many more. But let's assume, for the sake of argument, we have 100,000. So how do we fish out pairs of variants that are underlying disease? This is probably the very earliest approach that was done in human genetics. This is not the earliest paper. It was the first described a little before 2000. I will discuss each of these briefly. You can also try to evaluate all possible pairs of genotypes, an exhaustive evaluation, but this is generally not possible because there are just too many pairs of genotypes. We can apply pattern search algorithms, and I will describe this in detail. The such pattern search algorithms were developed in computer science, and only recently people have started to use them in human genetics. Using a Bayesian network is also a possibility. I will give you a description and an example. And last but not least, I started collaborating with this group in London. They are computer scientists and have come up with very sophisticated novel approaches. So here is MDR, multifactor dimensionality reduction. I don't want to go into very great detail, but for two given pairs of variants, for one pair of variants, that means two variants, there are 3 by 3, meaning 9 such cells. And in each cell, there is a number of cases and a number of controls. This method distinguishes between high-risk cells and low-risk cells. A high-risk cell is one with more cases than controls, and the low-risk cells has more controls than cases. This is done for all possible pairs of variants, and for each pair, there is such as a pattern of high-risk and low-risk cells. Based on all pairs of variants, a model is made, which eventually predicts for each individual whether the individual is high risk or low risk. This is done in 90% among all the data, and the prediction is applied to the remaining 10% of the data. Depending on the outcome, whether the predictions were good or bad, the model is changed. So I cannot discuss in great detail how this is done, but this is the gist of what's being done. The user comes up with a model that maximizes validation, cross-validation consistency, and minimizes the prediction error. It's still being used nowadays, MDR, although not as often as in the past. The main disadvantage is that you look at all possible pairs of variants. This is also more recently that people have tried to look for all possible pairs of variants. In this example, they mentioned there is an enormous search space, of course, for all the, they probably have had many more than 100,000 variants. So what these authors did, they focused on the biological plausible interactions among patterns. So they made a selection of variants that could possibly make sense biologically. So that's usually how people look at pairs, or all possible pairs of variants, by looking at it as a suitable subset. Now, frequent pattern mining. In the area of consumer habits, when people buy items in a supermarket, when you pay at the cash register, what you have bought goes into a database. So these supermarkets, they collect all this information in huge databases. And this happened even 30 years ago. So somebody then developed a computer program to make sense of all this information. This first program was the a priori algorithm, was developed by Agrawal in 1993. This is clearly an Indian name. Now, this a priori algorithm can do two things. For a given item, for example, when a consumer buys bread and milk, the question is, how likely is that person to also buy wine, as an example? So this algorithm developed conditional probabilities that a person will buy a specific item, given we know that the person has also purchased some other items. This is called an association rule. And we now then we implemented this algorithm into a new computer program that should search for genotype patterns. And for a given genotype pattern, predict whether a person is a case or a control. This was published some 8 years ago by Jingwen Zhang. She was my student, PhD student in Beijing, and now works in Canada. Newer implementations, oops, now I went into, or I should go back. I'm not sure how to go back.
So you can close and reshare.
Yeah.
Yes, sir. Now back.
Yeah.
That's okay.
So new algorithms can also be used now. However, it turns out these off-the-shelf algorithms, they have huge memory requirements. So this has not been very fruitful. Also, this approach here by Jingwen required an incredible amount of computer power. 2,000 or 1,000 computers were hooked up together to do the calculations. Bayesian networks is another approach that people use. Basically, one makes a model with certain variants of importance, variables that are genetic and other variables like cases and controls and so on. And then one estimates these variables with a Bayesian approach. This has been used quite frequently, and it has been relatively successful. But again, the data that have been published, they have been usually relatively small. For example, the person developing this algorithm used as an example only 1,000 SNPs, 1,000 variants. Now, more sophisticated computational approaches have been developed. This is a husband and wife team in London. I started collaborating with them about a year ago, and it's amazing. They can develop computer programs that are extremely fast for detecting patterns. That means for detecting pairs of genotypes, even among 80,000 or 100,000 variants. The first step is usually to look at one variant at a time. Single variant analysis, the trend test, for example, this is what has been done in the past, looking at one variant at a time. Now, I'm looking at this data here that was published by Andy Dewan. He was my PhD student at Rockefeller. He published an AMD age-related macular degeneration dataset in Science in 2006. It turns out this is the p-value based on permutation analysis. Highly significant. The first two variants are highly significant. And then there are two variants without an assigned chromosomal position. So I decided to disregard these variants because I want to see whether it's possible to find pairs of genotypes of variants that by themselves don't have much of an effect. So here we see the largest p-value is only 0.6 when we focus on variants other than the highly significant ones. And then I applied the newest search algorithm, and I find there are two patterns, two pairs of genotypes. The genotypes in a pair, one is a variant from chromosome 3 and one is a variant from chromosome 4. They are actually close together, but the two patterns are from variants on different chromosomes. And the p-value is 0.015 for each of these two patterns. So clearly this is a situation where pairs of genotypes have much more effect on the phenotype. We have a significant effect, while when you look at one variant at a time, the best p-value is 0.6. And I think this is going to be the rule more than the exception. Currently, researchers consider it as an exception when they find pairs of genotypes responsible for the disease. But when you think biologically, this is what should really happen, because most phenotypes are the result of a cascade of effects, of genotypic effects. And any pair of genotypes or variants in that cascade, if it breaks, then it would lead to a pair of variants that are disease-causing. Yeah, this is what I wanted to present. And so I hope I have not been too theoretical. If you are interested in learning more about this digenic inheritance, feel free to send me email. I'll be glad to respond. And if you have data that you would like analyzed, I will be happy to apply these new techniques. And there is no cost involved, except I would like to participate in the publication. Thank you very much.
Thank you, sir. In fact, you have given a very good insight into this particular field of statistical biology. And if there are any questions from the participants, I can allow them to have a few questions. So, Dr. Pallavi, can you see some questions from participants?
No, sir, no such question is there. So one question is there. Just a minute, sir. So there is a question from Dr. Monica Prakash Rai. Is the gene transfer equally applicable to algal system for improved fatty acid synthesis?
This question is for whom? Dr. Oat or Dr. Joshi?
No, not for me.
Okay.
They have not
Sir,
mentioned.
for Dr. Joshi, I think.
Dr. Joshi, yes, yes.
Yeah, I answered that question on the chat that transforming algae is still difficult. It's not as easy as higher plants because not much research has been done, but it certainly is possible to change or genetically engineer algae to make more fatty acid synthesis. The way we do it has to change. The vectors we use, the promoters we use needs to be different, but it certainly is possible to do that as long as we do some more research. Thanks.
Sir, I have one query regarding the transgenic plants. Just a query that whenever we do any kind of manipulations with the plant kingdom, then it certainly affects the ecosystem. So whether we are considering that particular aspect when we are manipulating the plants, because later on you have seen one photograph that was quite cause of concern for me, that whenever you are converting the genotype of any plant, then its whole morphology changes. A tree became like a creeper. So certain such kind of things may take place in the ecosystem. So how they will be impacting the ecosystem as such? Are we considering that aspect, sir?
Yes, obviously, because first of all these are experimental organisms. So when we do experiments, they are most of the time in greenhouse, and actually by law, we have to destroy them. We do not release these plants. We are using this technology to learn how system works, but we are not releasing them in nature. Actually, the way we and imagine how this will be doing. So if we get a plant which is good, which is useful, we'll be growing them in plantations. So we are not going to release them in forests. We're not going to release them in natural ecosystem. They will be in very controlled system. And by the way, in the United States, we already do some of these experiments, that we do it in a very controlled experiment. It's monitored, but at the end of the experiment, we destroy all these plants. So they do not contaminate. They do not get released outside. Now, some of the plants, I know that particular creeper you saw, I was just showing what could, sometimes you want to do something, something else happens. Because ultimately it's a life that's responding to these changes that we do. So, but not, those plants are just curiosity. They're not going to be released in nature, naturally not. Nobody needs that, those plants. So you have to make sure that the plants are in good shape and they're always grown in controlled environment. They're not in normal ecosystem. So hopefully we will not disturb the natural ecosystem.
Right, sir. You are not going to.
This was a good question.
With the ecosystem, right?
Absolutely. Good
Right.
question.
Thank you, sir.
Thank you.
Sir, I have one little query.
Sure.
This is a question to Dr. Joshi, sir.
Oh, okay.
Sir, in case of transgenics, there is the
Uh-huh.
the cellulose content that is going to be diminished as compared to hemicellulose, means in case of transgenics. What may be the probable reason for that?
You're saying why they change? When we change the cellulose, why hemicellulose changes?
Means in the graph that you have shown,
Uh-huh.
in case of transgenics, the
Right.
cellulose content that we have seen, that is going to be diminished there in case of that.
Uh-huh. Uh-huh.
So what may be the probable reason for that?
Right. So basically, we did that experiment on purpose. We were trying to see what happens when you affect the cellulose production. So you have three different compounds, three different polymers are working together, cellulose, hemicellulose, and lignin. When one of the three goes down, the other two look like are more. Okay, but there is no real synthesis. That's what I was showing. So there is no real synthesis of more lignin or more hemicellulose. It's only one component went down. So if suppose you take three colored marbles and remove some, you may feel like there are more marbles of other color if you take 100 every time. But that's not real, because we didn't really do anything for other two. In fact, we are saying these are three independent mechanisms. However, the net result is, if you take 100 grams of this material, it may appear that it has more lignin and more hemicellulose. There is no biological mechanism to change that.
Yes.
So actually what I was trying to get the data, trying to get the message is, these three are three different pathways and they do not talk to each other, which is very interesting, but that's how we want to learn about a system as such. But that, what you asked is a very good question too. Yeah,
Thank you. Thank you very much.
Thanks.
Good morning, sir. Thank you for such an informative talk. I have a query related to how the gene was underexpressed. We know how it is overexpressed under the control of a constitutive promoter.
Right. So just to answer, there are multiple ways of how you can reduce the expression. So the two ways we have done, one is called RNAi. So we take a small piece of cDNA, but that cDNA is put in a construct in such a way that it will destroy the original gene product. Okay. So that's called RNAi. The other one which we used recently is antisense. So instead of gene going in one direction, we put it in opposite direction. So what happens is the normal gene and its opposite direction copy, they bind to each other and they destroy each other. So that's how we suppress a gene expression. So it has nothing to do much with promoter. It is much to do with the gene itself, is what we do.
Okay. RNAi mutants were generated.
RNAi or You can do other mutants also, like CRISPR-Cas9.
Yes, yes,
As Professor
CRISPR.
Kaushik was saying some time back.
Yes,
So you can certainly use CRISPR-Cas9 to
yes,
target and create a mutation also.
yes.
Thank you.
Thank you. Thank you. sir.
Sir, this is Dr. Raghavan Saxena. Sir, I have a question that can we also introduce abiotic stress tolerance through the modulating the expression of genes?
I think I missed the first part. Could you repeat, please?
Can we develop a abiotic stress tolerance
Biotic stress.
by modulating the gene expression?
Absolutely, yes. If you know which gene is involved in that process, basically the biotechnology, plant biotechnology is much, much advanced, as you know, perhaps already. So you can do this at will if you know which particular gene is involved in which pathway. You can certainly do that. And actually it is being done. Although currently I'm not working on it, but abiotic stress resistance, you can absolutely work on. It's no problem at all. In fact, it's very easy to work on those things.
Thank you.
Yeah, thanks.
Sir, there is one question in chat box for Professor O from Ananya Singh. She's asking about how do we calculate the nucleotide diversity stats?
This is a question for me.
Yeah,
Yes,
it's
sir.
for you,
Yes.
sir.
How do we calculate what?
Calculate the nucleotide diversity stats.
Nucleotide diversity test. Not familiar with this test. Yeah,
Okay.
sorry, I'm not
Okay.
familiar with this test.
Yeah. Sir, you are muted, sir. Dean, sir, you are muted.
Ananya, can you elaborate the question? I think your question is incomplete. Dr. Oat is unable to understand what you're asking actually. Okay, no problem.
Excuse me, sir. I have a question from Professor Jargot. Sir, would you please enlighten us regarding the frequent pattern mining method in genetics?
Yeah, I use these frequent pattern mining methods. You know, the main advantage of these frequent pattern mining methods is that they don't need to evaluate all possible pairs of genotypes. They have a way of knowing which ones they don't need to evaluate. But I'm not that much of a computer scientist. Actually, I'm not a computer scientist. So I use these methods like the a priori algorithm or the new algorithms and implement them into my current way of doing association analysis. Instead of looking at single variants and their association with cases and controls, I look at pairs of variants. But I'm just making use of these pattern search algorithms. This is a collaboration between computer scientists and me as a kind of a statistician.
Okay, thank you, sir.
Dr. Oat, I have one query regarding your Bayesian networks.
Yeah, again, I'm not a Bayesian.
It is related to actually AMD, that is age-related macular degeneration, in which you have set the p-value 0.01. Can we brought it further down to 0.001?
I mean, people have analyzed the AMD data, not the ones I presented today, but an earlier version of AMD data. with the a priori algorithm and with Bayesian networks, it turns out there is actually a relatively good agreement between the two. Not 100% at all, but I was kind of surprised that they furnish similar results.
Okay. Right, sir. Thank you so much.
You're welcome.
Thank you so much.
Sir, I have a question. So there are a lot of softwares available for genetic linkage map analysis. So, but very few softwares are there which are free and they are having a lot of advantages. So can you suggest name of any 2 or 3 softwares which are readily available and they are free?
Yeah, the linkage programs, they are a little bit older, but they are free. They're called linkage. And then there is the Merlin program. Many people use Merlin. My favorite is pseudomarker. It has very good properties. It uses association analysis and treats marker genotype, the marker allele frequencies as it estimates the marker allele frequencies at the same time. So you don't have to worry about the allele frequencies of markers. I think that is probably the best program, but not too many people use it. But if you are specifically interested, I can send you web pages, web links for these programs.
Thank you, sir.
You're welcome.
We have many invited talk presenters online as a panelists. So if there's any question from their side, we can take 1 or 2 questions from them also to Dr. Jargot or Dr. C.P. Joshi. If no question, then we can proceed further. Dr. Pallavi?
Okay, sir. Thank you very much, sir. The presentation was really very informative and may provide a new insight and understanding the importance of genetic linkage and computer program on human linkage analysis. It will be great pleasure for us to listen to you again in our future events.
I will be my pleasure.
Now, I would like to request all the dignitaries to kindly release e-abstract book.
Movinji, can you make it a large picture so we can click a picture also?
Yes, sir. It's already in gallery view.
Great.
Yes, Mubin, we can see it. It's quite visible. Okay. Right. Thank you so much. Thank you all. Thank you very much.
Welcome.
Thank you, everyone. It is a great pleasure of listening to the eminent persons who made this event unforgettable and added glory to it. Please permit me to start the technical session by thanking you all for making time to be here at this event. Thank you all.
Dr. Pallavi, before we switch to technical session, I would like to thank our foreign delegates. They are the experts in their fields. Dr. Joshi, Dr. Oates. It's really a very great pleasure for all of us that you spare a valuable time, and that too in late evening. I think it's supposed to be around 11:30 or 12 o'clock. So it's so nice of you, sir.
Yeah, it's 1 a.m.
1 a.m.
Yeah.
Right, it's a late night. Thank you very much, sir. Taking so much pain for us. And thank you, Dr. Joshi. Thank you so much, sir. Thank you very much.
Thank you, sir.
Thank you, Chairman,
Thank you,
Professor
sir.
Dr. Kaushik, sir, for sparing valuable time, sir. And I'm thankful to all the panelists who are online. And we shall be switching to the technical sessions very soon. Thank you very much. Thank you all.
Bye-bye.
Thank you, sir.
Thank you, sir.
Thank you,
Thank you.
Professor.
Thank you all.
Thank you.
Further, all the invited talks will be continued here. Rest the participants who are having presentations in this time slot can move to the respective session by joining through the Google Meeting link provided. Remaining all the participants will continue to remain here with a Zoom link. Thank you, everyone.
Prasad sir is online. Prasad sir, can you hear me, sir?
Yeah, I'm here. Good morning.
Very good morning, sir. Nice to see you, sir, on this particular platform online.
Yes, yes.
And this particular session handed over to you, sir. There are three ITs in this. Dr. Manoj Prasad, who is from NIPGR Delhi. Dr. Sailendra Goyal, he is from Department of Archery, University of Delhi. And Dr. Vikas Jain, he is from IACR Bhopal. So, sir, after your presentation, these three will be presenting their work. So I hand over this to you, sir. I shall be there to assist you. Thank you, sir.
So how will it work now? I mean, I thought I want to leave after Manoj's talk because something has come.
No problem, sir. I will take over after that. You give a presentation. After that, Dr. Manoj Prasad can give. The rest I will take care, sir.
So nice. Okay.
Nice.
Okay.
Dr. Pallavi, you can take over.
Yes, sir.
Pallavi, you will introduce the speakers?
Yes, sir.
Okay.
The plants have been the basis for the medical treatment through most of the human history, and such traditional medicines is still widely practiced today. Modern medicines makes use of many plant-derived compounds as the basis for evidence-based pharmaceutical drugs. The session will be enlightening your wisdom with the recent advances in molecular biology and plant biotechnology. On behalf of Amity University, Madhya Pradesh, I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, heartily welcome Chairperson of the session, Professor Dr. Rajendra Prasad, Dean Faculty of Science, Engineering and Technology, Director AIB, AIISH, Amity Institute of Biotechnology, Gurgaon, Manesar. Co-chairperson, Professor Dr. Rajesh Singh Tomar, Director, Amity Institute of Biotechnology, Dean Life Sciences, Amity University, Gwalior, and to all the invitees, presenters, and attendees. I request Professor Dr. Rajendra Prasad to kindly deliver his talk.
Okay. So please allow me to share the screen.
Movin, allow sir to share the screen.
Sir, everyone have right to share this in this panel, sir. Sir, you have right. You can share.
Yeah, it's visible, sir. You can make it full screen.
Sir, sorry for interruption. Sir, Ms. Arya, I just want to introduce Dr. Rajendra Prasad.
Just a minute, sir. I think one thing is missed out for you. That's very important. All the ITs would know about it. Dr. Arya, just go ahead, please. Just take 2 minutes. Sir.
I belong to MIT. I don't need introduction.
No, sir, your introduction is required for the people from outside MIT, sir. Please.
Yes, sir.
Sir.
Good morning, everyone. Myself, Arya Sahai, PhD scholar of Amity University, and I'm going to request Dr. Rajendra Prasad to give their presentation. So, Dr. Rajendra Prasad is a Dean Faculty of Science, Engineering and Technology, Director AIB, AISS, Amity Institute of Biotechnology, Gurgaon, Manesar. He has done his PhD Biochemistry from Agra University in 1970 and MSc in Biochemistry from Lucknow University in 1967. Dr. Prasad was former Vice President, Indian National Science Academy, former Pro-Vice Chancellor, JNU, former Dean, School of Life Science, founder Chairperson, Special Center of Molecular Medicine, Vice President, Translation Society of India, Research Ambassador, DAAD, and Germany Coordinator, Focal Point of Horizon 2020 European Commissions. He has received several fellowships and awards like a Royal Society Commonwealth, Bursary, Alexander von Humboldt and Stephen Fellowship, Wellcome Foundation, Biotechnology Overseas Fellowship, Mercander Professor at the University in Bonn, Center of Excellence GBT DSR UAGC Award. He has more than 50 years research and teaching experience. His research interests is focused on clinical resistance to antifungal mechanisms and regulation of resistance in a new antifungal molecular mycology. He has published more than 270 research papers in various national and international journal of repute, and that has completed several research projects. Some are still undergoing. Sir, I have request to kindly deliver your talk on one gene, one protein, and one life. Please, sir.
Okay. So, you can see my screen?
Yes, sir.
Okay. So thanks for the nice introduction, which are not necessary. But she a PhD scholar, right? Who
Arya is a PhD scholar, sir.
Okay. So nice to listen from a student's point of view. Like she said, I have a great lot experience in research, and which is almost 50 years. And so I don't want others to say all this, because that also start reflecting my age. So let us keep it that way. I'm still working. I have students and continuing to do my work. So, and I particularly selected this topic for students' sake, just to show that how little you can do even in 50 years. So I'll focus my talk on one gene, and that of course is protein, and my own time spent on this. And you will realize soon that I'm talking of a particular gene, which is a multi-drug transporter. And that fluxes out drugs from inside to outside. And that belongs to a fungi that we work on. So I'll go slowly one by one and try to avoid giving you a lot of data, but nonetheless, give you some background and then show you briefly some of the highlights of the recent work. And I will be happy to be interrupted if you at all want to do that. So for the benefit of the others who are not of the field, that multi-drug resistance implies it is defined as a resistance of an organism against a spectrum of drugs that neither share a common target nor a common structure. So that is something very, in that sense, very unspecific, because any drug can develop, the organism can develop resistance against that drug. It is not important what is the target of that drug and so on and so forth. So very, very unspecific phenomena, but occurs throughout the evolutionary domain of life. I just remind you this famous, everybody knows, this is the discovery of penicillin, which was rewarded to 3 great scientists of that time, of early part of last century. That was discovery of penicillin. So why I'm showing you this, because the penicillin was one of the first drug which was introduced in clinic, and it was said at that point that it is a miracle drug. So after that, you will not be able to kind of require any other drug to treat infections. And really, it was a wonder drug at that point. But I want to show you a next slide, which shows that as and when any drug has been introduced over the period of time, this is a slightly old slide of goes up to 215, but see that all these antibiotics of different class generations, they were introduced, and soon after the resistance has developed, if you see. And if you see slowly, the later part of the drugs which were developed, like say for example, diptomycin, soon it is introduced, resistance developed. And this time started taking much lesser and lesser in developing resistance than what it took in the initial days. So bacteria started learning faster how to resist these kind of drugs. And that becomes a big problem. So it was a kind of a rise of resistance to all the drugs which are thrown to bacteria. And it was not restricted to bacteria. That is my interest also, that it is common in fungi also, and in cancer also. Cancer, you are very well aware that half of the cancer types are to begin with are resistant to treatment. And the interesting point which I like to bring out is that the strategy or the philosophy to become resistant is quite one of the common strategy adopted by all three domains of life, whether it's bacteria or fungi or cancer. I work with a fungal infection, as I mentioned, and fungal infection has been rising over the years, as you will see from this particular picture which came in Science some years ago, that whether it's a plant infecting fungi or animal infecting fungi, or of course human, that includes humans as well, are all is in the rise, and you can see around the world it is in the rise. And the most disturbing part is that the fungi infections are more severe in that sense that it also causes species extinction, to say that. Like we have seen that many frog, birds, and from crops have been kind of becoming extinct because of the fungal infections, and that is still remains to be, it's not able to be cured. As far as our human health is concerned, we have fungal infection, of course, very common. Over 1 billion population have these fungal infections. And unfortunately, fungi can infect almost any part of your body. And the only basic thing is that mostly whenever you have a weakened immunity or immunocompromised, these fungal infections takes over, are very common. So some human pathogens which are very common, like Candida species, Cryptococcus, Aspergillus, and so on and so forth. I work on Candida and its species, but if you've seen already that new and new fungi keep emerging. It's not a new fungi, but you have seen that COVID time, the mucorals, which we wrongly call black fungus, also became very common infection because of this COVID infection and many other due to related factors. Just to say that these fungi are infections is on the rise, and we do not have very many options to treat these fungal cells to combat infection. What we have, for example, much better arsenal in bacteria. Whether it is bacterial resistance or in fungal resistance or in cancer cell resistance, you will find that there are several strategies which are adopted by these cells which become resistant. For example, decreased drug uptake, or decreased drug activation, inactivation, overexpression of target, mutation in the target, and so on and so forth. But you know, the box item which I said, increased drug efflux, is one of the mechanisms that is very common to all the organisms where we find resistance. Okay, and this is what interests me, that what causes this increased drug reflux, and that too in fungal cell. So there, this is true for everywhere, that we have two huge family of transporters. One we call ATP binding cassette proteins, the first one, and the other is major facilitator. Both are drug efflux pump, but if you see the difference, if I can show you again. So here, because ATP binding protein, it fluxes drugs at the cost of ATP hydrolysis. Okay, in the second case, it is also effluxes drugs, but it's like antiporter. The proton gets in and then the drug goes out, and so on and so forth. And these are the two transporter families, very huge families, which are present all over. And I work mostly on this first transporter, this ABC protein, which I'll talk about. And here, if you see, the ABC protein, whether it is from bacteria or from yeast or from humans, are very conserved. And I show you this because the blue color represents the conserved domains of this particular protein. So very conserved throughout the evolutionary scale, and are very common in terms of functionality, to say. So this became apparent quite early in 70s and 80s, that if you see anti-cancer drug transporter were discovered, they found that the anti-cancer drug is retained much less in cancer cells, which are become resistant to ectomycin T, for example. So in other words, there was always a increased outward transport happening in resistant cancer cells. Same thing was noticed at another occasion in terms of bacterial cells, where they found that antibiotic efflux is increased. In this case, it was tetracycline resistance in E. coli, and that again was due to a particular efflux activity attributed to a efflux pump protein. Here comes our contribution. We were looking also in early 90s that whether such mechanism would exist in fungal cells, and we found that it Candida albicans, which is very most common fungal infection among humans, to say that all of us, especially women folks, will always get at least once in their lifetime infection of Candida because it's opportunistic pathogen and can invade any part of the body tissue and so on and so forth. Anyway, so here resistance was there, but not so common, but we started looking for this possibility of such a mechanism that was found in cancer cells or in bacterial cells. And we were, to cut the story short, we were able to identify the first fungal efflux pump protein in a clinical, or you can say in pathogenic fungi, where we could show that the drug accumulation is decreased in resistant cells. So what we found was that in a normal cell, if you have the target and the drug goes and hit the target and the cell is kind of doesn't grow. But if you have the flux pump protein also, so they will kind of push the drug towards the protein to be fluxed out. And so it will never reach to the target. As a result, the cell will survive. So that happens because these are the flux pump proteins in yeast or anywhere else, and they are overproduced because of a lot of transcription changes. So the resistant cell, not only it fluxes, but it fluxes with much more rapidity. So the concentration of that lethal, otherwise lethal drug is not maintained intracellularly. So the cells are resistant. So that is what we discovered, and it became one of the very classical papers, and almost 500, 800 citations now. Over the year, then we kind of worked on this transporter and associated activities related to this particular gene and protein. And this is the one of the review we made a history of the ABC protein development in yeast, and you can see the box events which are boxed are the contribution made by our group in this course of evolution of ABC proteins. To suffice to say that there is so much to be done about this particular protein and associated other pumps in yeast, which have very important role in antifungal resistance. So what is important is now to understand the efflux protein catalyze extrusion of noxious compounds, xenobiotics or drugs from the cell is one of the most frequently used strategies for resistant to cytotoxic drugs everywhere in prokaryotes and eukaryotes. So that is a common philosophy adopted by most of the resistant organisms, among other strategies, of course. The problem comes now because these transporters proteins, I'm talking now only ABC, but it is true for other class of superfamily I just mentioned, that they have multiple members in whole genome. For example, in Candida, if you say we might have probably more than 24 ABC proteins, and so in Saccharomyces. And very few are only drug transporters. So the one question comes that why so many proteins of the same family, what they're doing. Of course, we will not be talking about this at this point, but I'm just trying to say that out of those 25, 26 proteins, ABC proteins in Canada's genome, only 2 proteins have a role, defined role or proven role in clinical drug resistance or antifungal resistance. So in other words, all ABC transporters are not drug transporters. So they are of course doing something else. But we will worry about this transporter, how it is transporting drugs and how it becomes changed to how it functions in resistant organism. So we have basically 2 strategies. One is that at the cell surface level, we know the drugs are thrown out. So we want to develop some kind of inhibitors which can block this efflux. So the same drug can be reused or repurposed. The second approach is that because these drugs, the pumps, the drug pumps or the flux pumps are overproduced, we want to block these expressions by transcriptional manipulations that they will never overproduce, and as a result, the drugs will not be effluxed and again become effective in killing the cells. So in the first approach, where we want to design some inhibitors, we spend a lot of time in understanding the basis of the efflux, how these drugs are transported. So I'll only talking about that. I'll not speak anything else about the signal transaction efforts, but just some few highlights how we have approached this problem. So if you see these ABC protein, it is more deeper. It has domains. So these are the helices which are part of the membrane, and they are preceded by 2 domains where these ATP I was telling you binds and hydrolyze, and that drive the efflux of the drug. Why I'm showing you this? That because this is domain based, but there's a basis of it, that these green domains are highly conserved. It could be in humans, in bacteria, or wherever. However, the blue ones, what we call transmembrane domains, they are really speaking very variable structures. And that is where the kind of the trick of the protein comes from. So because they can, I have to also say that these proteins are very, very unspecific. I would say polyspecific. They can bind any kind of structures and throw them out. So it's a big problem. So we wanted to know how it is happening. So what we have done is these are 12 helices of the whole protein. And what we have done is we have 12 helices have, say, for example, if you calculate, you know, the hardly about 21 amino acids are there in each span. So all 21 times 12 amino acids, which comprised of these transmembrane domains, we had replaced everything by another amino acid residue and identified of the whole protein how many residues are important for drug transport, for example. So don't worry about it, it's a very complicated slide, but the numbers only show that these are the amino acids, if you replace, they affect the drug transport. Okay, and these distribution of important residues, which you call critical residues, is not uniform. Some helices, if you see like 7, 12, 6, or even 10, they don't have so many residues which are important. Some have quite a few important ones. Anyway, because of this whole strategy, we made about 250 to 50 to 55 mutants of this whole protein and analyze each one of them and could show that how drugs and where drugs bind. For example, if you see this deduced structure, we know that there are some domains in the structure where certain amino acids are very critical for the binding of the drug and for the efflux. We could even figure out by way of different drugs efflux that some amino acids in the protein are common to all, some are very specific to each other, and so on and so forth. So one could show that there is a whole lot of specificity as well as polyspecificity inherent in this particular protein. And that is how they become very versatile efflux proteins. We could also compare this kind of strategy adopted by such proteins, like this is another family of transporter, antiporter, This is the one I'm talking right now, and this is from a human transporter of the same family. And all of them have a common philosophy that they have got certain amount of residues which are common in the sense they provide this central binding pocket, and then there exist small mini pockets specific by certain amino acids which are very critical, and that brings the polyspecificity or promiscuity to this kind of proteins. So it's very interesting, and still we don't know enough actually, so that we can really pinpoint and make a structure or a molecule which can totally block this. But nonetheless, the efforts are underway. In the process, if you realize in the same protein, if you see there are domains, and their domains cannot be remain disconnected, so they cross talk to each other. And we could also kind of using this suppression genetics, where we isolate another mutant which can recover the parent mutant and so on and so forth. And we could come out and show in the whole molecule how these domains cross talk from each other, and how they have the compensatory mutants from here to here, how they achieve these long-term interactions to communicate between these very apart in terms of distance, these domains. And that is how they decide substrate selection and the flux and so on and so forth. So it has been a lot of work, and I'm just trying to show you a gist of 5-10 papers of each of coming to show you this kind of slide. So we know a lot about this protein, but like I said, I think we have spent almost close to 30 years on this protein, and we know quite a bit, but frankly, I can also say very happily that we know nothing about it. And there's so much to do. And because I told you that there are so many of this protein in superfamily, this is a story of just one protein. And we have many proteins, like 25 to 24 proteins in different yeast. And we have worked out great detail how, in what other physiological relevance they have in physiology of the yeast, whether it's pathogenic or in Saccharomyces and so on and so forth. And just to give you an idea that since we identified this gene, there are almost 300 to 400 labs are working on these flux pump proteins. And a number of papers must be close to 1,000 now. And we must have published and contributed almost half of it in this literature. But we still don't know anything about it. We even celebrated 20 years of this particular gene identification, and we're also hoping to make a 30-year event in 24. Personally, I'm quite established in my field, and so on and so forth, but that is not the satisfaction. The satisfaction and the setback is that if you say this, that the only true wisdom is in knowing you know nothing. And believe me, in spite of, this is the point I want to make to all the students, that in spite of doing very focused research, you rarely come out with a solution that, okay, this is it. But that doesn't mean one would stop working. Of course, we are much wiser, and we hope to get wiser each day by continuously working and in a focused way. So this is, of course, helped by many of my collaborators in our national context, international context, and also has led to, I must have done almost now 70, 75 PhDs, and only I could show some in this. This is not an update slide, but I can say that I have spoiled their lives. They trusted me, and then of course, they are all very well placed. And thanks to them that I'm here. And I must have spoken about half of their work in these 10, 15 minutes. And this is my present team in Amity. which is again working on the same proteins in different aspects. And thank you very much. This is also a bird from our campus, taken by one of my colleagues. Thank you very much.
Thank you very much, sir. That was really a remarkable talk. Thank you so much. Now, I would like to invite Dr. Manoj Prasad, National Institute of Plant Genome Research, New Delhi, and Department of Plant Sciences, University of Hyderabad, to deliver his talk on the Trojan horse against tall CNDV infection in tomato. Dr. Manoj Prasad is working as a professor and J.C. Bose National Fellow at National Institute of Plant Genome Research, that is NIPGR, New Delhi. After completing his school in Kalyani, West Bengal, he is PhD in botany from the prestigious University College of Science, Bellingham campus of University of Calcutta in 1994 and 1999 respectively. Prior to work at NIPGR, Dr. Prasad served as Alexander von Humboldt Fellow at the Leibniz Institute of Plant Genetics and Crop Plant Research. His research interests include plant molecular genetics, genomics, and stress biology. Dr. Prasad has published more than 150 research papers, reviews, and book chapters in highly reputed international journals, like Proceedings of the National Academy of Sciences USA, Plant Journal, Journal of Expert Botany, DNA Research, Theoretical and Applied Genetics, Trends in Plant Sciences, etc. He is also serving as associate editor in renowned international journal of high repute. Sir, I request you to kindly deliver your talk on the Trojan horse against tall CNDV infection in tomato.
Dr. Prasad, welcome to this platform.
Welcome, welcome. Good morning to everyone.
Good morning.
Hi, Manoj.
Hi, sir.
Good to see you.
Yeah, nice to see you also, sir. So I have made my slide in full screen. Is it visible in full screen?
Not yet. Yeah, now it's visible.
Perfect.
So, where is the pointer? Pointer.
So usually on the left side.
So this is the spotlight one, is the pointer?
No, there's a red cursor. It comes if you click the right thing. But you can use your mouse. It's okay.
Yeah,
cursor. Cursor is good. Cursor is good enough.
Pointer option. Yes, yes. I got it, sir.
Yeah, this one.
Yeah.
This is normal.
So, good morning, everyone. And first of all, I would like to thank Professor Tomar for inviting me for this international conference on advances in biotechnology and nanobiotechnology.
So, Manoj, 1 second. I in fact forgot to thank Dr. Tomar, and he forced me to come, and I had to say, of course, yes. I never refuse him. So, thank you very much, Professor Tomar.
Sir, it's a great pleasure for me and all the young generations are following you.
Yes, sir.
You are also young. You are also young.
Manojji, chaliye, please go ahead.
Yes, sir. So today I'm going to talk about one story recently published in PNS. That one PhD student of mine, and she was awarded. As you know, NIPG was affiliated with JNU, a School of Life Sciences, and so she got the degree from JNU. And the talk which I'm going to tell today is SW5A, one resistant gene from plant, and how this resistant gene helps against providing tolerance against this notorious virus, plant virus, that is tomato leaf culminated virus infection in tomato. So coming to the my lab work, I worked in 2 areas and 2 crops. One is foxtail millet. This is all you know, that kangni, that nowadays you got all this ragi biscuit, ragi dosa. And this crop is basically stress resilience crop, and it grows in a harsh environment and doesn't require much more water and care. This is foxtail millet, and as I said, this crop is a climate resilient crop, and to know what are the genes and proteins which makes this crop, which is a C4 crop, and rice is a C3. And photosynthetically, it's more efficient than other C3 crops. So you also try to identify the C4 photosynthetic attributes of these crops so that we can transfer them to C3 crop in rice. And at the nutritional point of view, all the millets are nutritionally 5 to 8 times superior in terms of micronutrient and others, vitamin and other attributes as compared to rice and wheat. So today I'm not going to talk about this millet story. Today I'm going to talk about how we decipher the resistance mechanism through small RNA mediated resistance against this tomato leaf curl virus. So as you know that in tomato is belong to family Solanaceae, and it is rich source of vitamin A, C and minerals and natural antioxidants of carotene and lycopene. India is the second largest producer of tomato after China, and this productivity of this tomato has severely declined due to different stresses like avidic stress, like water, temperature, light, and chemicals. And so far as the biotic stress is concerned, the stress, biotic stress by virus, and it comes under either insect or pest or parasitic plant. So it causes 70 to 100% yield loss once this infection is occurred in the field. So far as the Gemini virus structure, genome, and infectivity is concerned, it's a twin isohedral structure and single-stranded DNA is a genome, and caused by this Vagom virus and transmitted by white fly, that is Babesia tabaci, and causes severe leaf curling. You can see here, this is the normal plant, and this is the leaf curling stunting also is there. You can also see it in your garden, in the kitchen garden, the chili leaf curl virus, the chili plant, all the Solanaceae crop, papaya leaf curl virus. So this is one example that how this virus once infected the plant, then the leaves become crunkled actually, and the plant becomes stunted. And yield loss, as I said, that it ranges from 70 to 100%. So far as the genome organization is concerned, this virus is a bipartite virus, and it has DNA A as a main component, is responsible for the virus replication and transcription, and DNA B for the movement of the virus from one cell to the another cell. So far as the composition of the DNA is concerned, it is 2.7 kb in length, and it has 6 viral protein, and these are the viral proteins. It was written here that we have intergenic region. This is the promoter region, and then the replication initiator protein, then RAIN protein, replication enhancer protein, transcriptor activator protein, capsid protein, and the silencer suppressor. This is AC4. So these are the various important component of the DNA, which helps the virus for his replication. And so far as the DNA B is concerned, it is 2.6 kb in size, similar to DNA A, and it has 2 important protein. It is movement protein and the nuclear shuttle protein. So it helps the virus from movement after replication is one from the one cell to the another cell. So for our study, we have received 2 tomato varieties from International Indian Institute of Vegetable Research. This is ICER Institute from Varanasi. They are the collaborator for us. We did a lot of field trial there. And this H8878-1, which is a natural tolerant, but not resistant. There are no resistant variety, true resistant variety available till date against this virus. So that is another problem working with this virus. Whereas the fruit size is small, but very tasty. This is in the market, you get it, the very round shaped small one, this desi tomato, and they're very sweet. But the farmer prefers this Punjab Chuhara because this is the big size, and the profit-wise farmers found that their yield is very good, and as the size is very big, similar to brinjal actually. Sometimes we get a tomato like smaller size of brinjal. So these are the 2 natural variety which we have obtained from Banaras. And to decipher the molecular mechanism against this virus, we studied the infectivity analysis in these 2 cultivar, H88 and Punjab Chuhara, and we did this experiment through agroinoculation in the control condition after 7 day, 14 day, and 21 days of post-infection. And when we check the viral infectivity, viral titer, we found that that in the tolerant cultivar at 21 dpi, the viral titer is very less as compared to the susceptible cultivar. But when we check the synthesis of siRNA after this virus infection at 21 dpi, we found that the viral siRNA is more in the tolerant cultivar, where the viral titer is less. But in the tolerant cultivar, the generation of siRNA is less, while the viral titer is more. What does it mean? Means that this lower level of viral DNA accumulation in the tolerant cultivar attributes to an enhanced siRNA generation upon viral infection. And it basically, this tolerant cultivar was able to induce the gene silencing with the production of virus-specific siRNA. Till date, there are 6 TY genes has been identified, which are located in different 12 chromosome of tomato, and they are responsible for the providing tolerance against this virus. So far as the TY1 and TY3, they are allelic to each other, and this is RNA-dependent RNA polymerase. And TY2, it is a NBR class of resistant R gene. TY4 is a locus with minor effect in the tolerant against this virus. TY5 is a recessive gene. This is known as pelota gene. And TY6 is yet to be identified and characterized. So these are the TY6 TY genes. They have been identified till date. But today I'm going to talk about a host R gene, that is SW5A, which is located on chromosome 9 of tomato, that has not been reported by any group. But this, to check whether this our SW5 is a TY or not, we have checked this that our both the cultivars H88 and Punjab Chuhara against this TY6 TY genes, and we found that our 2 tomato cultivar H88, which is tolerant, and Punjab Chuhara, which is susceptible, doesn't contain any of the TY genes in these Indian varieties. Then we ask the question, do microRNA play any role in providing defense against this tomato leaf curl virus? To know this, we have constructed small RNA libraries under the mock treated, this is a control, and the tomato leaf curl virus infected condition in both the tolerant and susceptible cultivar. And we have done the expression study of the 13 potential microRNA, which are found to be differentially expressed in the small RNA library. And this data was further validated by the Northern blot. So you can see here that microRNA 159, this U6 is the control, at this microRNA 159 is differentially expressed, that it is down-regulated in the tolerant cultivar, where it is up-regulated in the susceptible cultivar. This you can also see here. Here, that it was down-regulated in tolerant, but up-regulated in susceptible cultivar. Then we try to find out that the target of this microRNA 159, and to know this, we have done in silico study, and we identified that it has 7 potential targets of this microRNA 159. And it was further validated by the RL-Embrace, and we found that out of 8 sequencing experiment, 7 has been clipped in the MIP33, which is a transcription factor. So through RL-Embrace, we found that microRNA 159 as the target, and that target is a transcription factor, that is MIP33. And then we check the expression of this MIP33 upon virus infection. As I said, that microRNA 159 is down-regulated in the tolerant cultivar. The target will be automatically, it will be inverse, up-regulated in the tolerant cultivar. And the susceptible is the vice versa, because microRNA is 159 is up-regulated in susceptible cultivar, but the target is down-regulated. So this data was further confirmed by the short tandem target mimic, that is known as, popularly known as STTM-mediated silencing of microRNA 159 in both the cultivar showed a dramatic increase in expression. You can see here level of MIP33 in STTM lice as compared to the control plant. So we postulated that microRNA 159 possesses the ability of specifically silence MIP33. to know the real function of this MIP33, whether it is providing tolerance or not, so we silence this MIP33 through virus-induced gene silencing in the tolerant cultivar tomato, and we found that when we silence this MIP33 in the tolerant cultivar showed susceptibility. And you can see here that after this infection that of the MIP33 TRV, that silence plant, they are downregulated. So silencing of MIP33 transform the tolerant phenotype into susceptibility. And when we check the viral titer in the MIP33 silence tolerant plant, we found that more than 4-fold increase in viral titer as compared to the control plant when we silence this MIP33 in the tolerant cultivar. So loss of MIP33 function may assist in viral replication, thereby enhancing the viral infection and multiplication. So these are all the transient assay. Shortly, we can characterize the gene instead of going for overexpression. Then we ask the question, what is the function of MIP33 in tomato leaf curl nidiri tolerance? So to know this, we have done the, because MIP33 is a transcription factor, so it should have DNA binding and transcriptional activation assay. For to know this thing, we have done East1 hybrid assay, and we through audiobacillin screening, and we found that through East1 hybrid assay that MIP33 bind to a MIP binding domain, which is TAAC-AAA. And when we mutate, there is no interaction. So through East1 hybrid assay, we proved that MIP33 has DNA binding and transcriptional activation property. To identify the downstream target of this MIP33, we did chip sequencing. And we identified that 60% of the binding site is in the promoter region, 12 gene, that is the 34% in the inside the gene, and the 5% in the terminator region. To validate this chip data, we studied the expression of these 21 genes in MIP silence as well as non-silence tolerant and susceptible plant during control and virus infected condition. A significant reduction of the transcript abundance of all the genes was observed in the silent plant. But interestingly, we found that one gene, that is SOL98130, which encodes for a leucine-rich repeat containing protein, which is we termed as the SW5, has 6-fold upregulation in the tolerant and 7-fold downregulation in the susceptible cultivar. So we identified through chip sequencing of the downstream target of MIP33, that is R gene, that SW5, leucine-rich repeat containing R gene. This description of this R gene is a coil-coiled NB-LRR class of plant-resistant genes. SW5 consists of 5 paralogs, among which SW5B confers resistance against the orthotospovirus. This is a RNA virus. In our case, it is a DNA virus. And mechanism of resistance through SW5, that is for the RNA virus, involves interaction of non-structural movement protein, that is NSM, through which that virus protein and elicited of a hypersensitive response. In our study, we identified that MIP33 was found to modulate the expression of SW5A, which is the paralog of the SW5B, and which is responsible for the susceptibility factor for the RNA virus, tomato spotted wilt virus. So basically, take home message is this: SW5 has 5 paralogs. SW5A, which we identified in our study, it is showing tolerance against the DNA virus, whereas in 2016, D. Oliver et al., they have identified SW5B, which is another paralog of SW5A, and it is showing tolerance against the RNA virus, that is the tomato spotted wilt virus. But these 2 viruses, SW5A is not working against the RNA virus, and SW5B is not working against our DNA virus. So this vice versa. So as I said that SW5, check the SW5 promoter binding activity of MIP33, and because you know that to validate this MIP33 mediated regulation of this R gene SW5, we have performed the electromobility shift assay, EMSA, And we found that a high affinity protein DNA complex was observed when SW5 and MIP33 was there. In the mutant is the mutant in other, when the mutant is there, there is no binding. And so we said that binding of SW33, MIP33 on SW5 promoter may regulate the transcription of this SW5R gene. And this data was further confirmed by the CHIP assay. What we did that MIP33 was cloned in the PGW17 expression vector, transiently overexpressed in the leaf of this torrent cultivar, and the CHIP assay was performed with anti-MIC antibody. So in this case, we validated the SW5A along with another 2R gene, that is TM2 and S1N gene. And we found that, yes, through this CHIP assay, that MIP33 has the binding capacity and regulate the expression of the SW5 in planta, but not by another R gene, that is TM2 or S1N gene. Then we ask the question is whether this SW5 will play a role in providing defense, and if so, what is the underlying mechanism for so? So then similarly, like MIP33, we silenced this SW5A in the tolerant cultivar, and we found that the silencing of this R gene SW5A in tolerant cultivar becomes susceptible. This data was further confirmed by the viral titer assay, and you can see here when silencing of SW5A in the tolerant cultivar, more than threefold induction of the viral titer in the tolerant cultivar observed as compared to the control plant. So when we silence this R gene SW5A, which leads to the enhanced accumulation of the viral genome. To find out the mechanism of SW5A, initiate the HR, hypersensitive response upon the infection of tomato leaf curl virus, the leaves of tolerant cultivar H88 as well as MIP silenced and SW5 silenced, and SW5 silenced and MIP overexpressed plant were infected with this tomato leaf curl virus. So we found that there is no HR, only HR is observed only in the control condition, but no HR was observed in the MIP silence, SW5 silence, and SW5 silence and MIP overexpressed plant. So what does it mean? It means that SW5 regulate hypersensitive response, that is HR, during tomato leaf curl virus infection. To find out the interacting partner of this R gene SW5, we basically try to identify the SW5 mediated inhibition of virus spread, and through identification of downstream target of this SW5A, which is a AVR factor, ovulance factor. As I said, that virus has 10 proteins, 6 protein in DNA A and 4 protein in DNA B. When we did the East2 habit assay of this viral 10 proteins, virus 10 protein along with the SW5R gene, we found that through East2 habit assay that this host SW5R gene was interacting with the viral pathogenesis protein, that AC4, through Y2H assay. And this data was further validated by the GST pull down assay and the co-immunoprecipitation assay. So whatever data we are showing here, it was validated additionally by 2 or 3 alternative approach. So we showed that through this approach that AC4, the viral pathogenesis protein, is the downstream arborescence factor of the host R gene SW5. And through in silico analysis, we identified that that this 4 motif RTSK is important for this interaction of viral pathogenesis protein HC4 with the host R gene SW5. And this RTSK motif was present in the C terminal region of this viral AC4 protein. We performed the East2 habit assay, and you can see here when this SW5 C terminal region, you will get more colonies, but in the mutant there is no, and in the N terminal region is less colony. So we showed through this East2 habit assay that RTSK motif in the viral AC4 gene is important for this interaction between SW5A and AC4. This data was further confirmed by the biopsy assay, where we have showed that when we mutate this AC4, there is no interaction But when this SC4 and SW5R gene is there, you will see clear fluorescence. So 4 amino acid of the RTSK motif of AC4 is necessary for this interaction with the host R gene, which is SW5A. So we did a side directed heterogenesis to identify the which of the 4 amino acid sequence is required for this interaction. So we muted this RTSK simultaneously, and we've identified a serine residue which is present at the 48th position in the AC4 protein. And when we muted this serine residue of the RTSK motif, there is no interaction. But in R, T, and K, the interaction is there. So in conclusion, what I can show you that I have shown you that the microRNA 159 is downregulated in tolerant cultivar, while the target MIP33 is upregulated, and in the downstream, this MIP33 binds to the host R gene SW5 and initiate the transcription and translation. And SW5, that host R gene, interact with the viral pathogenesis protein AC4 and initiate the hypersensitive response and initiate this cell death to restrict the virus for spreading, and the plant become tolerant. In contrary, this in susceptible cultivar, this microRNA 159 is upregulated. That's the region the target MIP33 is downregulated. And so the target is downregulated, therefore it cannot bind to the promoter of the host R gene SW5. There is no transcription, no translation. And so there is the downstream mechanism is not happening. So there is no HR response and the plant became susceptible. So in conclusion, I can say that presence of functional R gene does not necessarily imply downstream resistant response. In our case, I have shown that involvement of both positive regulator, that is in our case, MIP33 is the positive regulator, and microRNA 159 is a negative regulator of the R gene SW5A expression, adds to the complexity in activation of defense response against this virus infection. So considering the role of SW5A in case of tomato leaf curl virus, which is the DNA virus, and SW5B in resistance against the broad spectrum orthotosporovirus, this is the RNA virus. These two homologs could be used for gene pyramiding through breeding on for achieving the broad spectrum and durable disease resistance against diverse virus causing yield loss in tomato. So we have to pyramid both SW5A, which is responsible for the DNA virus, SW5B responsible for the providing tolerance to the RNA virus. If we pyramid these two genes, SW5A and 5B, in our local tomato variety, we can generate a broad spectrum resistant against both DNA and RNA virus. So we have filed the patent in 2017, Indian patent. And this paper was now published in PNS. And you can see here, all the authors are from India. There is no, and with the collaboration of the JNU, Supriya Chakrabarti, who helped us in providing the infectivity clone of this tomato leaf curl virus. And Namisha, she got the PhD degree from JNU, is an NIPJ PhD student. And she got all the awards like INSEE Young Scientist Award for this work. She is currently working as a Sir Women Excellence Awardee from DST Sir. In my lab, she's continuing. And I would like to thank the funding agency like Department of Biotechnology and Department of Science and Technology for funding our work, and all the other lab members which are very dedicated and very sincere and hardworking. Thank you very much.
Thank you very much, sir. The presentation may provide a new insight in understanding the field like virology, stress biology, and plant genetics. Now, I would like to invite Dr. Shailendra Goyal, Department of Botany, University of Delhi, for his talk. Dr. Shailendra Goyal has done PhD from University of Delhi in 1999 on rDNA sequence evolution and molecular phylogeny of Vigna and Fasciolus. His area of interest include molecular genetics, molecular cytogenetics, molecular marker applications, genomics, genome organization, apomixis, and its developmental aspects. I'm also trying to develop, He's also trying to develop major research program on safflower improvement. Dr. Goyal has published several research papers, reviews, and book chapters in highly reputed international journals. He has coordinated several research projects, major grants, research collaborations. He is actively involved in interdisciplinary work and collaboration with different people working on different aspects of plant research. Sir, I request you to kindly deliver your talk on an integrated approach using genomics and breeding for a traditional Indian oil seed crop, that is safflower.
Well, welcome.
Good morning. So again, I should start by thanking to Professor Tomar for providing me this opportunity. I work on various aspects, have been to Amity, Gwalior many times, and I always like being there and the atmosphere there. It's unfortunate that now I have to do it online because of the COVID situation. I hope we can do it in future by being there only. Right now, I am working on apomixis mainly, the sexual reproduction, and the aspects of male and female differentiation in plants. But besides that, I also work on a crop, which is called as safflower. So I will share my screen. How do I share the screen?
So go to the bottom line, there will be some commands. Share
I'm
screen,
not seeing that.
Can you see?
Yeah, I see. I saw it now. Okay, I hope my screen is visible.
Yeah, now it's visible. You can make it full screen, please.
Yeah. So this crop is an Indian traditional crop, and we are trying to develop a very comprehensive program on it. Been working on it for about 8 years. Various people have worked on this crop. And the important aspect of this crop is that it's a seed oil crop and a traditional crop. And India do require such crops because we have a huge gap between our demand and supply of the edible oil. So the historical account of safflower goes about 4,000 years back. The remains have been found in Tutankhamun Egyptian pharaoh's pyramid. And if you know that the pyramids always were given the products of luxury, and this was a product of luxury at that point. So safflower had a long-standing value. It went to India and China somewhere 2 to 3 BC, reached to Europe in 15th, 16th century, and immediately gained importance because it is also used as a dye, source of a dye called carthamin. Carthamin was a major source of dye for clothes for a very long while. Although after say about 19th century, it was replaced by the synthetic dyes, but it is still being used as an organic dye and also as a food color. So in 15th, 16th century, people used to use it for thread coloring, the thread red color. And that thread was used for covering the papers of importance in the government. And that gave the if you know, today we also say that red tapeism. So the red tapeism is actually came from that. The color carthamin was responsible for it. In the present day, it is still being used as an industrial oil and edible oil, also an alternate to saffron. It is actually an adulterant. It is used as a color, as a bird seed, and various medicinal like Ayurveda, Greek, and Chinese. So the crop is very important. And if you look at the overall seed oil scenario in world, the safflower is almost non-existence. The major oil crops are rapeseed, soybean, and sunflower. And if you look at the oil composition, the one which is most celebrated is actually olive oil, which has around 71% of oleic acid in it. And if we compare safflower with it, we have two types of oil available. One, which is high on linoleic acid and is mostly used as an industrial oil. It is in industry, it is like a different kind of oils and also the cosmetics. That's where this is used. And then the edible oil, which has high oleic acid and which is as good as olive oil and even better. So the crop is, actually can be grown in semi-arid zones. It is drought tolerant, salinity tolerant, and that's why it is being used in area of Deccan Plateau. Even after all this, the crop is still lagging. And this is the scenario of the oil. You can see most of the oil crops are palm oil, rapeseed oil, soybean oil, and sunflower. This is the scenario of Indian food, edible oil. This is the demand, this is the supply, and this is the import we are doing every year. These are the recent year statistics. So you can see in 1990, the orange one, which is the import, came down, but so also came down the total demand. And you know that the oil prices have shoot up very hard. So this crop is more important in those terms. If we look at the international status, the Kazakhstan is the top producer, although India was a top producer till 2014, but it has lost its place. It's not because we are going down in the production per hectare, it's mainly because we are losing the land under cultivation for safflower. So in the Indian farmer is not very happy with safflower. This is the world scenario. This is the Kazakhstan scenario. It's increasing. The production of safflower is increasing. In Indian scenario, it's going down. And the main reason for this is actually there are some problems with safflower. The low oil content, low yield, spiny nature, susceptibility to biotic factor, lack of seed dormancy in inefficient agriculture practices. So all this is making it less useful for the farmer. But there are problems and there are solutions also. So when we looked at the germplasm, we found that there are high oil content varieties also available. We have plants which are not spiny by nature. We have plants which are source of the resistance to different biotic factors. We also have plants which can tackle the problem of lack of seed dormancy, the wild species even showing dormancy. And agriculture practices are being taken care by different institutes like IOR, which are also a partner with us on this program. So we started with a simple premise that we will procure the germplasm, we will characterize the germplasm, and we will utilize the germplasm. And we also started generating various genomic resources because this is a traditional crop which is orphan. So there are no resources available. So we started working on all this. Although last year a Chinese group did sequence the crop and the sequence is out now, but there is still a lot to be done in this crop. So we have procured 2,300 genotypes from USDA, which were belonging to 50 countries, and almost all the countries where safflower is existing was represented in this germplasm. We use this germplasm, so these are the countries which are being represented in this germplasm. We use this germplasm to extract 531 accessions from it based on the various agronomic traits which were available in this. And we use this information by using these traits like pre-harvest traits and the post-harvest traits. Post-harvest traits like oleic acid oil, oleic acid content, seed oil content, and 100 seed weight is the important set of traits because it can help in increasing the production of this trait of this crop. As you see in our germplasm, we have range 16% oil to 50% oil. Oleic acid is 9% to 82%. Linoleic acid 13 to 87%. Seed weight 1 to 8 gram. So we have a huge range in this germplasm of 531, and it helped us in starting the program quickly. This is the oil content accessions, ranging from 41 to 50%. The linoleic acid 80 to 87%. So these are high acid content and high oil content accessions. And when we looked at the regional pool by countries, we found that all these regional pools are being represented in our data. And so we had good diversity, good agronomic traits, good representation from different countries. So we were all set to start with this program. These are some of the pictures. You can see the normal spiny safflower and non-spiny safflower. Head is very small and head is comparatively much bigger. This is the one of the progenitor, Obsicantha, and this is the normal Tinctorius. This is the oil content percentage, which we have figured out in all the germplasm. And we figured out that there are some lines which are really high on oil content, which is about up to 50%. Here, you can see up to 54%. The fatty acid content, this is 2 years data. So the correspondence between data is also good. And you have 80% of oleic acid, 80% of linoleic acid also. We used all this morphological data first and created different groups by using programs like NJ, structure analysis, and we figured out that the germplasm is showing us enough variability in terms of morphology. Once we were done that, we started with a very simple marker, which is the AFLP. It was somewhere in 2009 when we started with it. And we figured out profiles. Based on this, we have done a grouping of all the germplasm, and we figured out in our germplasm, there were at least 19 groups based on the structure analysis. The NG analysis showed us 6 groups, and the one major thing we noticed is that the Indian germplasm was available in all the groups. Each and every group had Indian germplasm in it. Although the literature said that the Indian germplasm is not good and doesn't have enough genetic diversity, the US germplasm was told to be the best and having the most genetic diversity. Our analysis showed it totally reversed. Indian germplasm was good, had a lot of genetic diversity, and the American germplasm was very narrow in their genetic diversity. So this was quite promoting for us. We were happy to see these results. And the two takeaway home messages were our germplasm was good and can be used for improving the crop. And the American, although they do have some important agronomic traits, but it's poor in the genetic variability. So using this, we created our core collection. The core collection was done by 2 different programs, by different methods. And when we looked at these methods, individual germplasm, individual core collection didn't have representation from all the countries. Can you see that? There are zeros in these countries. So what we did, we created a composite germplasm, a composite core collection. This composite core collection had representation from all the countries. So finally, we were able to come up with a core collection of around 120 accessions, which now became the basis of further improvement program. And we started generating the genomic resources. The first resource we generated was SSRs. This is the status before we started. There was almost nothing. And when we started, what we did is Illumina-based sequencing, figured out microsatellites, used primers depending on these microsatellites. The microsatellite were having normal expectation. Most of them were mononucleotide, but we had a good representation from trinucleotides. So we used around 325 SSR loci to analyze our core collection. We were now focusing only on the core collection of 120 accessions. And when we did that, we found that maximum transferability was into Palestinus and Oxycanthus. These are the 2 species from Carthamus genus, which are actually the progenitors of the Tinctorius, the safflower. So it was expected. We used all this data. Along with it, we also generated the transcriptome. And from transcriptome also, we found the genic SSRs and included in our repository. This data was also published. All of this data is actually published. And we also tried to generate different kind of markers. One of them was the transposon-based marker. So we analyzed transposons in our Illumina data. We figured out different kind of transposons which were available, T1 copia and gypsy. We even these are the different transposons which we figured out in the genome analysis based on the Illumina data. We then tested the variability of these retrotransposons within the genome by cloning and sequencing them. And we figured out there is enough variability available in the genome. Overall, we found around 65% of repeat element in the genome. And using that, we tried to devise markers from the transposons. So this is just transposon data. We figured out the complete elements also for copia and gypsy both. And this data is now ready for creating the markers based on transposons. Now, the traditional way of using transposons is doing transposon display, which is something similar to AFLP. But we think it is now an outdated marker. So what we are trying to do, bioinformatic analysis and trying to figure out the distribution of transposon throughout the genome. More sequencing data is coming, so we'll be doing it with different genomes and try to figure out what kind of transposon can be converted into a marker for being used throughout the genome. So because a good marker has to have a good distribution throughout the genome. So beside this information, we started with the association mapping because we had a panel of 120 core accession in our core collection. So we used it for association mapping. For association mapping, all these plants were taken and then we characterized them for the phenotypically and we characterized them based on the SSR markers. Use these two data sets to do the association mapping. Before you start with the association mapping, one very important aspect is to learn the relationship between the plants within the association panel. If there are any IBDs, which is identical by descent, or any kind of keen relationship between the accessions, your association fails most of the time because the existence of the marker is mainly then because of the relationship rather than the relationship with the trait. So our first part was to do that. We did a structure analysis and we found there was almost no clear grouping in this whole set of 120. We did kinship analysis on this and we figured out that the kinship analysis have shown very little relationship between these plants. It's almost non-existence. So our core collection is an ideal set of markers which has very important agronomic traits in it and has no relationship with each other, so can be used for association mapping. So considering that, we did association mapping for 8 traits. And we found markers with very good R-square value for each of them. So our association mapping was very successful. But we want to now reach to the loci, close to the loci. So we have to increase the density of markers on our maps. So what we started doing, so this is also published in Frontiers. High genetic diversity in core collection, weak population structure, low or known kinship relatedness, significant marker trait association, high number of marker trait association was found in both the cases. So what we need now is just the increased density of the markers. So now the current work plan has moved towards GBS-based mapping, genome sequencing and resequencing, and genomic selection. And of course, we are making more and more crosses for different contrasting traits. GBS, this is just showing what GBS is. What it does is uses a restriction enzyme digest. So the restriction enzyme digest reduces the complexity of the genome. You are now dealing only with a part of the genome from which you sequence only a smart part of genome. So you just have a representation of the genome in your sequencing, which gives you enough SNPs to distribute them throughout the genome. So we are using MSC1 and we might use PST1 also in future. We have 2 accession, A1 and A2, which are now the beginning of the linkage mapping program. These 2 accessions have very good traits, contrast between them, like plant height, head diameter, head number of heads, seed yield per plant, 100 seed weight, oil content, oleic acid content, and linoleic acid content. So using this, we have created a map in the F9 population. We have already sent out the tissues for GBS and we have received the first set of data just this week. We have around 760 MB of data for each accession. We are right now processing them through TESSEL pipeline to extract the SNPs and create a map, which I think should be done within a month or so. We did face problems during the COVID because two of our crops were lost, lab was closed multiple times. So, but overall, we are now in a good situation. So another thing, because since we are generating the A1 and A2 based maps, what we are trying to do is we are also trying to do the genomic selection. So genomic selection has a simple, it's a complex genetic algorithm, but it's a simple concept. What we are trying to do is we are looking at the phenotype and trying to find a correlation between phenotype and genotype. We normally correct it with the residual error. But in this program, we are trying to include the environment also. So we are looking at the micro environmental variables. And for that, we have done the planting in different places, which is Fulton, Nagpur, and Delhi. Fulton is the Nari Institute, which is known for safflower. They are collaborating with us. Nagpur is actually a seed company. The daily is we are doing it ourselves. So what you are seeing here is the contour plots. The contour plots show you the distribution of that particular trait in XY coordinates, and it just is mapping the third coordinate onto it, which is pH in this case, the salinity in the second, and electrical conductivity in the third. So using these traits, we will be now doing the genomic selection in this crop. So overall, if you look at our program, the program started with the accession, which we procured from the USDA. We have selected 531 accessions from it, did a characterization using AFLP. This is the genetic characterization, and we found genetic clusters based on it. We did the phenotypic characterization, and we figured out the accession which were having the unique combination of characters. We have created a composite core collection using these two data sets. This core collection is now used for identification of parental lines which are being crossed. We are creating biparental mapping populations. We have generated genomic resources of SSRs. The AFLP are being used for these mapping populations also, but actually we have abandoned AFLP. We have now moved on to the SNP only. And the linkage mapping is being done. At the same time, we have initiated the association mapping with the help of our composite core collection. We are doing a resequencing of the core collection also, and we are also generating better, because the one which is published has about 80% of genome. So we are trying to come up with a better genome also. The data for that is also coming. I think within this month we will receive it. It was targeted for last year, but again, the COVID delayed our plans. These are various papers we published from this data, +1 frontiers, again frontiers, and more papers are in the pipeline. And the program has been running with us for almost 8-9 years now. So there are 4 people who did their PhDs from this program. They all are well placed now. 2 of them have gone to US for their postdocs. One of them is now working with a company, a bioinformatic company. Bharti is also gone to Canada. Krishna has just submitted his thesis. Pooja has left our lab after 4 years of hard work, and now she is working in Canada. She is doing some kind of PhD only there. It's a good lab, although. Sapna, Megha, Varun, and Palomi are comparatively recent and working with us. They have, I think they are in 2nd or 3rd year of their PhD. Annie is a Ramalingam Fellow. She is responsible for the initiation of the genomic selection program. She is a statistical genetist. Then there are 3 professors in our department who are collaborating on this program. Amar has retired. Manu and Arun are still working with me. Overall, we have 3 different collaborators. IOR is the Indian Institute of Oilseed Research, and NARI, which is the Nimbakar Agriculture Research Institute in Fulton, Denmark Technical University, which are helping us with the different kind of bioinformatic analysis of the data, which is coming up. A large amount of data is coming up, so we needed some bioinformatic help. So we have collaborated with the Denmark Technical University. Funding has been provided to us with the University of Delhi, because it's a traditional orphan crop. So getting a funding was not easy, but University of Delhi helped us in the beginning. And then DBT has given us a bigger program, and DST has also given a bigger program. Government is also aware of the situation of oilseed in India, and safflower is now a part of the national mission of oilseed. So we hope that we will be getting more work done in future and maybe make some impact on this crop. So thank you. I can take questions. Hello.
We can have a panel discussion afterward. Another lecture we can take up.
Yes, sir.
Thank you, sir.
Thank you very much, sir. The presentation may provide a new insight in understanding the field like molecular genetics, molecular cytogenetics, molecular marker applications. Thank you. Now, I would like to invite Dr. Vikas Jain, Microbiology and Molecular Biology Laboratory, Department of Biological Sciences, Indian Institute of Science, Education and Research, Bhopal, for his talk. Dr. Vikas Jain is a professor at Indian Institute of Science, Education and Research, Bhopal. He has done his PhD in Biological Science 2006 from Molecular Biophysics Unit Indian Institute of Science, Bangalore. His research interests include microbiology, molecular biology, biochemistry, molecular biophysics. With his expertise in molecular biology technique, he helped troubleshoot PCR reactions. His unconditional and immense support helped speed up the molecular lab work with the precision. Dr. Jain has published several research papers, reviews, and book chapters in highly reputed international journals. He has coordinated several research activities, major grants, research collaborations. Sir, I request you to kindly deliver your talk on deciphering the physiological relevance and molecular properties of mycobacteriophage D29 Holland.
Thank you, Dr.
Then
Pallavi.
welcome, Dr. Jain, welcome to this platform.
Thank you, Professor Tomar. How are you?
Fine, thank you so much.
So thank you so much for the introduction, Dr. Pallavi, and I really appreciate the opportunity given to me again to discuss my research work with you. And let me begin by thanking the organizer for arranging the platform in such a beautiful manner. So there were very interesting talks before me, and so let me see if I can, match up to their presentations and their potentials. I really enjoyed the talks that were there. So my camera was not on, so please don't think that I was not available. So, okay, so let me begin my talk by introducing you to the to general work that is going on in my lab. So I'll just quickly go through this. So we focus on the mycobacterial work and the mycobacteriophage research work, and we study various aspects of these two organisms to bring to the world some kind of phage therapy against the mycobacteria, and we try to do phage engineering to understand if we can develop them as therapeutics against the mycobacterial infection. And then we also study phage resistance, just like the antimicrobial resistance, because that also may become a problem in the future once the phage therapeutics begin in the light. And my lab also works in the area of applied molecular biology, and where we try to develop various tools and methods to ease recombinant DNA technology work. This is a very latest research work from our lab, positive selection expression vector, which was very recently published. So today I'm going to talk, and before I begin my talk, let me just quickly give the acknowledgement for my PhD students and students who have graduated, project student and BSMS students, and the collaboration, the financial support from the DBT, SCR, BCSR, and ICMR grants. The work that I'm going to talk about today is done primarily by Soumya, who has graduated long time back, and very recent graduate from my lab, Varun Bauda, who has contributed significantly in this particular protein that I'm going to talk about, and some contribution by Dr. R. Mahalakshmi. She is a colleague in my lab, in my department, and she is a very good protein biochemist, but we took her help of peptide biochemistry in order to address how this particular protein that I'm going to talk about actually works. So we could actually solve some very important aspect of this protein. Although that data I'm not going to talk about, we have published that data already. So I'll be focusing more on how this protein really functions and what really happens with this protein is they are present in the phage and what it does. So this is a very standard phage lytic life cycle. So we focus only on the lytic phages, primarily because those are the phages that lyse the cells and do not undergo lysogeny, which is a process where the genomic DNA that enters into the bacterium integrates into the genome and just stays with the bacterium. So we are interested in lytic phages, and the bacteriophage, mycobacteriophage that we study in the laboratory is actually a lytic phage. And this is a general life cycle. Let me not go into the detail of this, but this is the last step of this life cycle where the phages eventually lyse the cell and they are released into the environment so that they can infect the next available cell. It is at this step that we are most interested in, and there are certain proteins that are produced by the phage which allow for the lysis of the cell. And now the cell is being lysed inside out. It's not from the outside in. So these proteins are produced by the phage, which help phage lyse the bacterial cell. And two families of these proteins are endolysins, because bacteria have a cell wall made of peptidoglycans. So these endolysins, they work on the cell wall, peptidoglycan, and lyse it. And then you have a cell membrane, which actually surrounds everything inside the cell. And so holins that are produced by the cell, by the bacteriophage, they break down the cell membrane. And it is these holins that we will be talking about today. So, excuse me, how do you hide it? This does not go. Okay, so So mycobacteriophage is what we are interested in. And why is that? Because we know all about the Mycobacterium tuberculosis and the kind of infection that it causes. And it's one of the most known deadly diseases. And the Mycobacterium tuberculosis actually is considered as one of the most, one of the smartest pathogen known to mankind, because it adapts to everything that is given to it, all kinds of antibiotics it is resistant to, etc. Now, this is a typical cell wall structure of the Mycobacterium tuberculosis. What we have is a standard plasma membrane followed by a peptidoglycan layer. But in the case of Mycobacterium, we have additional mycolic acid layer and arabinogalactan layer, which attaches mycolic acid and the peptidoglycan together. So in order for mycobacteriophage to lyse this kind of cell, it requires proteins which will break down all of these layers. A second. Which will break down all of these layers. So in order to break down these layers, mycobacteriophage produces 3 proteins. So one is the lysine A, and these proteins are produced in the cytoplasm. Lysine A and lysine B are the 2 endolysins, which are required for the peptidoglycan as well as the arabinogalactan layer. And then the choline that we had talked about earlier, it is produced and it goes into the plasma membrane, makes holes, which allow for these lysins to diffuse out so that they can work on the respective layer. So in this way, the entire layer of the mycobacterium is broken down and the phages can come out from the cytoplasm to the outside. So we study all of these proteins in our laboratory, and today we are going to focus only on the choline. How does a choline look like? Now, most of this information is available from the E. coli phages and from other bacteriophages, but for mycobacteriophage choline, the information is very less known. Usually, choline is having transmembrane domains. You can have 1 or 3 up to 3 transmembrane domains, which actually are required to make holes in the membrane. And then choline also has a charge C-terminal region. It's a long charge C-terminal region. And the types of choline, actually, there are of 2 types of choline. One is the pin choline, which only makes small holes, and so that it allows for the proton gradient to disrupt. This requirement is very different from the canonical choline, which actually forms bigger holes, which allows for the diffusion of endolysins outside so that they can break down the peptidoglycan layer. In the case of mycobacteriophage D29 that we work, we do not know which kind of choline it is. It is either a pin choline or a canonical choline. And we have data for actually in support of both. So it's very difficult to judge right now at this stage what kind of choline we have. How does a choline function when it is produced? So once it is produced, it goes into the membrane and stays there. And once they are produced in large quantity, they start aggregating in the membrane. Now, and once they start aggregating, they form what is we call as the death raft, because there is a highly clustered form of structure over there. And once the rearrangement happens of this death raft, they start forming holes, which will allow the endolysin to come out of the hole. So, and this endolysin can then degrade the peptidoglycan layer. So this is a general mechanism known for E. coli cholines, but for mycobacteriophage, whether the same mechanism happens or not, we still do not know. And the work in this direction is still going on. So in our lab, we started dissecting the mycobacteriophage D29 choline and to understand its toxicity, cytotoxicity. So we started by dissecting the protein with shortening its C-terminal region to find out how exactly and what region of this choline actually causes toxicity. So what we found out that by dissecting this protein, and we actually started deleting roughly 30 amino acids from the C-terminal region. So we did that, and we found out that after deleting this from 116 onwards, the protein becomes non-toxic. Before that, the protein was toxic. So we thought that there is something in this region itself which is required, and this region is definitely important. So we first expressed only the C-terminal region to see if that can cause toxicity, but C-terminal region alone is non-toxic. So we actually took this small region over here and fused it with this smallest protein that we had generated, which was non-toxic. So this small region was added, and what we find is that this particular construct actually shows toxicity in the cell. When I say toxicity, I mean the cell is getting lysed, actually it's getting broken down, and all these experiments have been done in E. coli. So this choline does not really differentiate between mycobacterium and E. coli. So if you express it artificially, it can break down E. coli cells as well. What we then found out is that within this region, we found out a small coiled coil domain, which is present in this region, and probably it is this region that is required for choline to function. So if you delete this region, then the choline should not work. And we did that experiment by knocking out this particular domain from the choline full length protein. And we found out that once you delete this particular coiled coil domain, the protein stops working. it does not show that kind of toxicity that it is supposed to have. So the cells remain alive longer than the wild type choline. Now coming, so with this experiment, we showed that the coil domain in the protein is really significant in order to show toxicity in the, in order to show the toxic effect of protein on the cell. Now coming to the C-terminal part, which I had told you that the C-terminal region is highly charged region. So this is D29 mycobacteriophage choline protein, and these underlying regions are the two transmembrane domains that insert in the membrane, and this is the long C-terminal region which has a lot of charge residues. And we wanted to find out what is the significance of these charge residues in the protein, and you see a lot of positive and negative charge residues present here. So what this student did, Varun did, is that he mutated all of these charge residues in the following fashion. He either retained only the positive charges in the C-terminal region, only the negative charges, or he completely removed all the charges and made them serine. So it's all neutral over here. And then in the coil region also, he'd made some mutations to retain only the positive charges here. Only the charges here. And what we found out after all of these experiments, what we see is that only a positive charge protein, that is that a protein that was carrying only the positive charges in the C-terminal region, was showing the toxic effect on the cell, and all other proteins actually became significantly less toxic compared to the wild type or this protein. So what we conclude from here is that it's the positive charge present in the C-terminal region. It is that is required for choline to show its toxic effect. What we think is that the positive charge residues can interact with the negatively charged membrane, and that is what is required for the protein to aggregate in the membrane and then can cause the toxicity. Now, after we gathered these kind of information, we thought that can we engineer choline molecules so that it can become even more toxic than what we are seeing right now. So to try this experiment first, we just extended the C-terminal region of the wild type choline. That is that we just took this portion and added it extra over here. So this protein became bigger. And what we see is that the protein is even more toxic compared to the wild type choline. And this experiment of fluorescence recording was done at 20 degrees. That is a very low temperature for E. coli. And it is at this temperature that we could actually properly capture the amount of toxicity this protein was having. If you increase this temperature, you will find that both the proteins are extremely toxic. But we wanted to actually compare with higher temperature, are both the proteins equally toxic or differently? What we found is that the choline, our HC construct, that is the extra C-terminal region construct, actually is more toxic. So this assay is a beta-galactosidase release assay from E. coli. So once the E. coli cells lyse, the become porous, the beta-galactosidase enzyme from inside will come out, and you can assay that. That tells you whether the cell membrane is porous or not. And within 15 minutes of induction, what we see is that this protein, when it is expressed, it shows higher beta-galactosidase compared to the wild-type protein. All right, so this is one portion that we wanted to find out whether how choline really kills the cell, E. coli cell. And what we found, because this was a question that people asked that whether it's only disrupting the plasma membrane, why would it lyse the cell eventually? So we expressed the protein and we did a SEM analysis of the E. coli cell as a function of time. And what we found out that the moment you start expressing choline protein, the cell structure starts to deteriorate. And when you have longer time duration, you start having pits, and eventually the cell becomes completely, a bad shape, and plasma membrane rises, and then cell actually loses its integrity. So finally, we wanted to actually find out what is the significance of choline protein in mycobacteriophage physiology. So in order to understand a gene importance in an organism, you have to knock it out to find out now how does it differ from the wild type. So we knocked out this choline gene from the D29 phage, and that's a long process that you do it. Once we knocked out, we started studying it, and what we find is that this gene is not essential for the phage survival, because we could actually isolate phages with the gene knocked out. But what we find is that it delays the cell lysis ability by the knocked out phage. And it also delays the, so basically cell survives longer and it delays the cell lysis process. However, this difference is not because of the morphology of the bacteriophage, because even after deletion, the morphology of the bacteriophage stays the same. So it has something to do with what the protein really contributes to when it comes to cell lysis. In order to understand that, we did two experiments. One is the plaque size diameter, and we find that the D29 phage actually forms the Holin knockout phage forms lower plaque size compared to the wild type phage. And this is a one-step growth curve, which tells you whether the phage is really better or worse than the wild type. And what we find is that it has a longer burst time, and it has a smaller burst size compared to the wild type phage. So what we actually conclude here is that Holin deleted phage is viable, and it shows lower burst size and plaque diameter, and it is less efficient in killing host cell, and coil domain is essential for the activity. And we could engineer the using the C-terminal region, we could engineer the Holin with higher toxicity. And why we did that is now we can introduce this in mycobacteriophage back to have an even more virulent bacteriophage that can break down the mycobacterial cells rather quickly. So that's it from my end, and I thank you for your time and attention. I hope I finished it within time so that we can go for the panel discussions and lunch. Thank you so much. Thank you.
I think we can open the forum for discussion. If there are any question from anyone from all the three our respected invited speakers, Dr. Manoj Prasad, Dr. Sailen Goyal, and Dr. Vikas Jain. All the participants, if they have any question, they can put in the chat box or they can ask.
Can I ask Dr. Prasad a small question?
Yeah. Please go ahead, Surrender.
Okay. So I wanted to know the nature of the effector from the virus.
This is a white fly. It is naturally causing in the natural condition infection. But we are not relying on this natural condition for our screening and doing all sorts of experiment, because we never know that what other infection is going on in the field. So we have done this agro inoculation, that we have make an infectious clone with the help of Professor Supriya Chakravarti, who has made this clone, and he has also taken the patent for this making this agro infection clone. And that is in the greenhouse growth chamber condition we used, control condition we used to do the infection and do all the viral titer analysis. This is a white fly, Bemisia tawasi, and this is a common fly, and it occurs everywhere in India.
So you must be doing the mechanical inoculations of the agro.
Yes, through agro inoculation.
Okay, thank you. Excellent. Also, I really like it.
So any question from anyone in the chat box or question answer session?
I have not seen any of the question. Only two questions are there. That's the old one.
Yes, sir. Okay, sir. Now we can close the session, and I'm really thankful to all the three IT and on behalf of chairman who had some important task. So he left a little early. So I'm going to conclude this session by giving thanks to all the three invited speakers, Dr. Manoj Prasad, Dr. Sailen Goyal, and Dr. Vikas Jain. The talk by Dr. Manoj Prasad that was focused on the Trojan horse against tall CNDB. So this CNDB is New Delhi virus?
Yes. This is New
Why was given New Delhi virus?
Yes. Because there are several strains of this virus. We also has Gujarat virus. We have also a Bangalore virus, Karnataka virus. But the North India strain is a bipartite. DNA and DNA B is there. But whether it is in southern part, they have only DNA A. But in place of DNA B, they have beta satellite.
Okay.
But in the southern part, it's monopartite. Here in the northern part, it is bipartite.
It was really a very good talk by Dr. Manoj Prasad on tomato leaf curl. I think it's a basic problem with the tomato plant. It is causing a lot of effect on the production of the plant. That means tomatoes. So it's really good work. Also, he filed one patent that was really a good work done by him. And I think nothing can be said about the genuinity and everything done by Dr. Manoj Prasad. I know him from last, I think, 1 decade. He's doing really good work on these sort of species. Thank you, sir. Thank you very much.
But I was missing visiting Gwalior actually. That is as I told you that online lecture nowadays I avoid to give because that feeling is not there actually.
The feeling
the physical
right,
thing was.
We are eagerly waiting for opening of the these restrictions. Once they are lifted, then we will certainly be meeting 1-to-1. Thank you, sir.
Yeah, thank you.
Another talk by Dr. Sailendra Goyal. He is also working on the plants, and safflower is, I think, the future of oilseed. So a lot of work has been done by him, and a lot need to be done. Dr. Sailendra Goyal, I think you are doing a very good work on this particular oilseed, and in future, the demand of oil, basically the edible oil, is very much increasing with the increase in population. So I think you will be one of the contributors in overcoming this issue. Thank you, sir. Thank you very much. Dr. Vikas Jain, he is actually working on that particular disease that is prevailing in India from, I think, 4, 5, 6 decades. And still we could not overcome it. And his work on this mycobacteriophage D29, colon particularly, is really a good approach. And very soon we shall be seeing new outcome in this particular field and overcoming the problem of MDR and all some aspects which are related to the to tuberculosis. So thank you very much, Dr. Vikas Jain.
Thank you so much.
For valuable time for coming over here and sharing your valuable thoughts with us. It's really a good work, and I think our participants will certainly learn something from all these 3 presentations. Yeah. Thank you all. Thank you very much.
Thank you.
Good afternoon.
Good afternoon.
Thank you, sir.
Thank you very much. On behalf of AIB AUMP, I would like to thank all the eminent persons who added glory to this session. Now, as per the schedule, there will be a lunch break, and the next technical session will start after the lunch break. Thank you so much.
At 1:30. 1:30, we shall assemble again on this platform, where Dr. Rajiv Prakash is the chairperson of the session, Nanotechnology and Biosensor, and one scientist, Dr. Vasilis Fotopoulos from Cyprus. He is the co-chairperson and the invited speaker also. So there shall be 2 talks from these 2 chairperson and co-chairperson, followed by the talks by Dr. VP Sharma, Dr. Aparna Khanna, Dr. Seema Nara, and Dr. Semendu Ranjan. So we shall all come again on this platform at exactly at 1:30. We are abiding by the timetable, and I think we are not lagging behind, and we are as per our program. We had to close this by 12:50. We are closing at 12:46. Right. So we shall meet again at 1:30 exactly. Thank you very much.
So we will start just in a few minutes because some of the students are joining after lunch.
No problem, no problem, Srivastavaji. Take your own time. You are very much in time, as Professor Sharma said, that this is really this is difficult to control time in such big conferences. So please take your time. I am very much online here.
Okay, sir. Thank you very much, sir. So hardly we will start in next 2 to 3 minutes.
Thank you.
Sir.
Good afternoon, Dr. Prakash and Dr. Vasilis from Cyprus, Dr. Prakash from Varanasi.
Very good afternoon.
Sir, welcome to this platform. And this particular session is under you, Nanobiotechnology and Biosensor. So I hand over the session to you, sir. I welcome you, Dr. Vasilis, who is also from Cyprus. And he is very much coordinating with all of us that how I shall manage the time, and my time is just 3.5 hours behind you. So, sir, I hope we have managed it.
Everything is perfect. Thank you.
Thank you, sir. Thank you so much. So, sir, I hand over this to you with the talks of yours and Dr. Vasilis Fotopoulos, followed by 4 talks by Dr. VP Sharma, Dr. Aparna Khanna, Dr. Seema Nara, and Dr. Semendu Ranjan. Sir, Dr. Prakash.
Oh, thank you very much. And I request Dr. Westless to please proceed because I'm not having details of his speakers. And I'm supposed to give my first talk, so please, you proceed. Thank you.
I have no, yeah, so you would like me to do the talk first? Okay, sure.
No, I'm saying that you proceed this session, and because this is my talk is a first talk, so you please proceed the session.
Ah, so to introduce you, you mean? Okay, yes, okay, sure. Let me just, sure, just a second.
So, sir, before we proceed, Dr. Vasilis, I would like to request our faculty, Dr. Pallavi, to please introduce you, both of you, with the audience, with the participants, and then we can go accordingly.
So Dr. Prakash can do that first, and then I can follow them.
Yeah. Yes,
Good afternoon, everyone. We are starting with the next session, that is session 2, Nanotechnology and Biosensors. The application of nanobiotechnology include therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, waste treatment, and energy production. The session will be enlightening your wisdom with the recent advances in nanotechnology and biosensors. On behalf of MIT Institute of Biotechnology, MIT University, Madhya Pradesh, Gwalior, I, Dr. Pallavi Singh Chauhan, Assistant Professor, AIB, heartily welcome Chairperson of the session, Dr. Rajiv Prakash, School of Material Science and Technology, Indian Institute of Technology, Banaras Hindu University, Varanasi. Chairperson of this session, Dr. Vasilis Fotopoulos, Department of Agricultural Sciences, Biotechnology and Food Sciences, Cyprus University of Technology, Lemesos. And to all the invitees, presenters, and attendees. I request Dr. Rajiv Prakash to kindly deliver his talk. Dr. Rajiv Prakash is Professor and Dean, R&D, School of Material Science and Technology, Varanasi. Sir has received his MTech degree in Material Technology from Institute of Technology, Banaras Hindu University, and completed his PhD work from the Tata Institute of Fundamental Research, Bombay in 1999. Currently, he is Professor of Material Science and Technology and Dean at the Indian Institute of Technology, BHU. Before joining IIT Varanasi, he served CSIR as a scientist for 7 years. His area of research include polymers and nanocomposites, organic electronics and sensor devices, and energy storage devices. He is having more than 225 publications in various national and international journals of repute and 22 patents in his credit. He has been the recipient of Young Scientist Award, Award of the Indian Material Science Society, Metal Award of India. He is in the editorial board of several national and international journals. He is member of various national committees, including DST TIFAC for Indian Vision 2035 and MHRD Imprint the Program. Member of the Accelerator User Committee of Inter-University Accelerator Center, New Delhi. Member Defense Corridor Project of Government of India and Member Program Advisory Committee for Technology Development Program of DST, New Delhi, India. I request Dr. Rajiv Prakash to kindly deliver his talk on new generation biosensors based on nanomaterial, functionalized transducer. Please, sir.
Sir, you are muted. Please unmute. Yes.
Thank you. Also, thank you very much for kind introduction. And I'm thankful to Amity University and organizer of this conference for giving me this opportunity. I think I have 20 minutes. Yeah. So let me share. Is it visible? My slide is visible?
Yes, sir, very much.
Oh, and I will change now. Now it is changing there also?
Yes, sir.
Yeah, great. So thanks. Let me introduce this topic on advanced material-based new generation sensor. Since I have 20 minutes, so I don't think that I will be able to cover much, but I will try that how the sensors we can make
more advanced and more functional and selective and sensitive. But before using, of course, nanomaterials and functional polymers, that I will come later. But let me, in order to complete this talk, introduce a very basic about the biosensors are sensors. Here I have tried to show through this schematics that a sensing device is having at least the two components, important components, and one is the sensing layer, which is interacting with the target molecules, and recognition is converted through a readable signal through a transducer to a readable signal. Like if this interaction and it is coming in a form of electrical signal, we call it the electrical or the electrochemical sensor. If there is a light, called the optical sensor. If the change of heat, we are calling it as a thermal sensor. And similarly, mass change is a mass sensor and a pressure sensor. So if you see the picture, left-hand side, there is, I have shown a green probe. This is a transducer which is modified with some material. This material is further having some sensing material which is selective towards the target species, and target species I have tried to show through a cartoon, a star line. So this target species means anything of our interest which we want to sense. Example, if it is a virus or bacteria, or it is a molecule, gaseous molecule, or something dissolved in water, or my blood sample, anything. The target molecules are there, and that specific molecule will bind to the material which is present over probe. And this probe, as I said, is having a transducer modified with some material. When I am saying some material modified, what type of material it is? Let me further explain this with this slide. So again, a probe, a gray color transducer, which is having matrix. Now, this matrix is a polymeric matrix or some nanomaterial containing matrix, which is having capability to functionalize, to hold selective materials. And selective materials for biological application, very particular I can say, is some chelating agents, some agents having the specific cavity to hold the target molecule, or DNA, aptamer, enzymes, antibody. So these materials can be put over this probe, over this sensor, in order to make the sensor selective. So in first figure, I have tried to show that if the selective material is a chelating material, is having a specific cavity to hold the target molecule. So when the cavity is opened, means no target molecule is present, then this sensor will give it certain response based on the type of transducer, and that response we will take as a baseline. After that, when the target molecule will come and enter in the cavity, then the response will change. I will show again that how the response will change, and this change in response will be sensed by the transducer as a difference in signal than the baseline. So the difference from the baseline in signal is the sensing response because of the binding of target molecule to this cavity or to this specific chelating material. Suppose this cavity or this chelating material is a DNA, is a single strand DNA. So if a single strand DNA is present there, it will bind to its counterpart present in blood, present in any fluid, and then this combination can be sensed. I will come again with some example. In a second picture, I have tried to show if the sensing material is enzyme, then here the enzyme, as you know, that the enzyme and substrate reaction, changing enzyme from oxidase to reductase or reductase to oxidase, and this change of the oxidase to reductase means redox reaction can be sensed over the probe, and this change in redox, which will give you a change in current, will be sensed as a as a sensing signal. So with this background, let me also tell then what type of transducer devices we can use. Say when we are calling the new generation biosensors or sensors, we are trying to improve our devices to get the better and better signal. Sensitivity and selectivity depends on the material and of course also the devices, but more on the material side. But when we are talking about the sensitivity and the very low noise detection, then we have to improve our transducers, our devices. Some of the devices I have tried to show here, which are very common in such type of sensors, what we are going to talk today, like a chemiregister, where just the resistance change on interaction of a target molecule is treated as the signal, and this signal is calibration one can do with the target molecules for the sensing purposes. Or a better configuration of the electrodes in the interdigitated electrodes, as I have said, as shown in the center, are in a device like a diode configuration or the field effect transistor configurations. These devices also used to get the sensitivity, high sensitivity, introduce high sensitivity in your sensors. I will try to take one example if time permits. What type of materials I'm talking? So if you see the earlier slide, here I have said a transducer. Say the gray color is the transducer. This transducer is modified with some matrix, some materials, which I have shown in a blue color. So this matrix should have some property like holding your chelating agents and the biomolecules and able to transmit the interaction of your chelating agent and target molecules to transducer. Means there should be some conductivity, or in some way you can get the signal to transducer. It should not stop or behave as an inert layer. In order to get better sensitivity, people are using some typical type of polymers, and the conjugated polymers are also called the conducting polymers, are one of the potential candidates. And some of the example I have shown here in this slide, if you see the polyphenylene, polythiophene, polypyrrole are very famous polymer, polyaniline. But friends, let me permit to introduce that why this is the better polymer or conducting polymer. So if you see this picture of the two polymers, where I have shown the first polymer is the polyethylene and second is the polyacetylene. In both the polymers, you will find the backbone is same, the carbon backbone. In the first one, polyethylene, you have the carbon backbone, and the bottom one, the polyacetylene, you have again this polymer as a carbon backbone. But the difference, the chemist can tell immediately, the difference is the carbon is sp3 hybridized in the first one and sp2 hybridized in the second one. And this hybridization is making some changes, and this is that you can dope these polymers to bring conductivity. So the first one is the insulating or the conventional polymer, but the bottom one, the polyacetylene, is the conducting polymer, not the conventional, where you can dope this polyacetylene to get some usable signals or the conductivity. So how you can dope it, again, time is not permitting me to go detail of the doping, but at this point of time, you consider that you can make these polymers conducting, and conducting means whatever the interaction is taking place at the interface of this polymer and the analyte can be taken to the interface of the polymer and transducer. Means the signal can proceed through this type of polymers further. So now how you can prepare, how you can make these polymers, because issue is this, if I go back and see these are the devices, and what you need, you want to coat your polymeric material over these devices, and then you will functionalize biomolecules over these polymers to get the sensing property. So how you can coat these polymers? There are several methods, several ways of the coating. And you know that spin coating, drop casting, etc. are known, but those are having some limitations. And when you need a very ordered film, ordered films means where the polymer, or if the polymer is having some nanomaterial, that should be uniform. I will show again this. Then you need a very interesting you need some good technique, and this is very interesting technique, not very new. Is a well-known technique called the Langmuir-Blodgett technique, where you are seeing the top picture. It is a turf, and turf means just a bucket of a water bucket, and this water bucket, you are filling the water in this bucket, and you have two arms. You can see I have shown in the white color with the two bold arrows, and these two arms can move. So what you are doing, this bucket size, I can say is a few centimeter, means 10 to 15 centimeter long, and nearly 10 centimeter in width, and depth is hardly 1 or 2 centimeter. So in this bucket, you fill the water and put the arms. Arms are controlled arms, so not manually, but electronically with motor controlled arms. With software, you can put to give the movement of this arm slowly to come closer and closer. When you are putting your solution of the polymer over this water in between the two arms, as I have shown in the cartoon, your polymers, means spherical balls, are you are putting over the water surface. So polymer is not soluble in water, so it will form a type of oil over water like situation. So oil over water, you can imagine if you have a beaker of water and put the few drops of oil, how it looks like, it will be the same like this. So polymer molecules will float over the water surface. We call it at the liquid air interface. At the liquid air interface, molecules are present. So you bring the two arms closer and closer. Then what happened? You can think that the molecules are here and there, and when you are bringing this closer and closer, molecules will also come closer and closer. In this case, the molecules are nothing, they are the big polymer chains, and they will start to come closer and closer. So this you can determine through a very simple way by plotting pressure versus surface area that we call the isotherm. The pressure isotherm in the bottom left-hand side I have shown. Through this isotherm, you can have a feeling then when the molecules come to closest, and then after when you will push, it will collapse. This I will show you through this picture, and I hope this picture is visible on your screen. So if you see a right-hand side picture, in right-hand side picture, just I will concentrate on two pictures, A and C. If you see A is the polymer, closed, dense polymer film. And if you see the C, wrinkles, means when we compress polymer molecules over the water surface, there was a point when it formed very good dense film as A, but after more compressing, there is collapse point, means a wrinkling start, which is shown in figure C. These figures we have captured through the SEM, scanning electron microscope, you know, and this we can see at the nano level or the submicron level size. And then by seeing these images, we have established this technique, the Langmuir-Blodgett film formation technique for conjugated or conducting polymers. I think nearly 10-15 years back, it was not known for conducting polymers. And then for different type of polymers, we established the protocol, and I can say that one can form these films, uniform films, over the air-water interface. Now, then we tried, and this is the first time, why not we can introduce nanomaterial at the air-water interface. Means not alone formation of polymer film, but polymer film along with some nanomaterials. And the beauty of this is not only that you will enhance the property of the polymer film, but if your nanomaterial is encapsulated or are immobilized with biomolecules, then you will introduce the biomolecules in polymer film very easily. Say, if you have the gold nanoparticles functionalize with some biomolecules, antibody, antigen, or aptamers, then these gold nanoparticles you can introduce in polymer film. So automatically your biomolecules immobilized over the gold nanoparticles will come and decorate the polymer film. So again, this I have tried to show through the cartoon that how the MOS2, you know, this the two-dimensional sheets, just like a graphene, and this MOS2 sheets either functionalize with biomolecules or not, they can be introduced along with the polymer. And without going much detail in coming the short, because you can get the details in Langmuir paper in 2017. If you see the left-hand side, or see the next pictures here, that A and B is showing the film, large area film, which we formed over the air-water interface. And C and D, you see that functionalize with the MOS2. So MOS2 is present here and there, more or less quite in a uniform pattern over the polymer films. And now if this film just you lift and put over the device, your device is ready as a sensing device. So far what I tried to explain that how one can develop a sensor device, a new generation, a functionalized sensor, polymer film decorated materials over the devices. So these films, which are already if decorated with biomolecules, can come over the devices, or after formation, you can decorate your device with biomolecules. Both the possibilities are there. When I'm talking about the decorating your sensor with biomolecules, means making your sensor selective for target species. And one of the best way is this, what we tried. Either you use antibody against your target molecules, if you are talking about some biological sensors, or you use the aptamers or the DNA. Both are there. So if I'm talking about antibody, so it's a very complicated process how you can raise the monoclonal antibodies, but we are using monoclonal antibodies available in market. And if we are talking about the DNA, then just you have to open the double strain DNA and take the single strain. And this one single strain you immobilize over your device. But need not to immobilize whole full single strain DNA. You can select a small piece of the DNA. Here the biologist will help you to find out the more representative small strain of that DNA, and of say 10 base pairs, 15 base pairs, 20 base pairs. And that is small strain of the DNA you can functionalize directly over your device. Again, with this cartoon, I am trying to explain that what is the meaning of the immobilization of the strong DNA over the device. So again, recall what is the device of sensor device. Yellow color is the transducer, green color is the polymer film which we have formed over device, and then this is coupling with this a small strain of DNA. Here I have tried to show that the DNAs are present in a very ordered form, which is not the case actually. And then when the complementary part is coming, and attaching with this single strand DNA, you will get the double strand DNA. So single strand DNA and double strand DNA differences you have to sense through the device. How you can immobilize a small strain over your polymer. This I have given the chemistry of this. This is nothing, the carbamide reaction. So functional group present over the polymer is the carboxyl group. The functional group over the DNA are the biological molecules, the suppose amino group. Then the carbamide reaction through the carboxyl and amino, you can perform a simple chemical reactions and you can get the polymer tagged with single strand DNA. Once the polymer tagged with the single strand DNA, then there are the two ways you can go. One, as I discussed with the device, those devices as the chemi-resistant devices are the FET-based devices. There are, you can also try some impediometry or the voltammetry, means where you are using just simple integrated electrodes, having the two electrodes and both are covered, electrodes are covered with your polymer and functionalized with the biomolecules, and you are trying to find out the total impedance in some suitable electrolyte. So when there is a single strand DNA, the impedance will be something. After having the double strand DNA, the impedance will change. And the change of RCT, those who are aware with the Z prime are the this tau plot or the Z prime Z double prime plot, Nyquist plot. If you are aware, then the RCT value that will tell you very easily that DNA is single strand DNA or it form the double strand. Let's take one example, and this we have tried to settle some issues that how a aptamer-based sensor or the antibody-based sensor, we call the apta sensor and the immunosensor, and how the performance-wise they will differ to each other for a particular target species, and that we have taken a label-free PSA, prostate-specific antigen. But the prostate cancer, prostate-specific antigen is a very good marker. I'm not saying this, our biologists are helping us. And then if we are interested to determine the PSA, then what? There are the two ways, as I said earlier also, that you have the antibody against that PSA, is an antigen, and then you have an antibody against that, or you have aptamer. So we took both and we developed two different types of sensors. One was immobilized with aptamer, another was the enzyme. If you recall, I have said in the beginning that how you can get the functionalized polymers with these molecules, and we tried to sense the aptamer using the voltammetry, means the IV characteristics, as well as the impedance. So in this way, the two different sensing species, aptamer and the antibody, and two different techniques, the voltammetry and impedometry. And we try to see that how we can determine a target species PSA, which one is better and which one is stable. We observed that you can see also here that aptamer-based sensors are better in comparison to antibody-based sensors in terms of stability of the sensor. And some typical plot which we have developed for the PSA sensing, you can see here the log PSA versus delta I. And both are showing very sensitive detection of aptamers and antibody for the PSA, and even for the impedometry also, the more or less the same picture we are getting, that the aptamer-based sensors are better, stable, T is high, you need not too much worry about the handling of antibody. And the beauty of these sensors devices are, they are the quick, fast, low cost, and single use. So friends, I tried myself best to give you a general picture with the information of the formation of development of biosensors and application of the biosensors. And this is the aptamer sensing, aptamer-based sensing of PSA in a real sample. And you can see the sensing, we can make it to quite low to the feasible region in nanogram per liter concentration. So with these words, I think I should stop here. But before I come for discussion, let me acknowledge my students, those who are working in this area, my PhD students, Rabi, Subhajit, Anurudh, Nikhil, Priya, Sweta, Nupur, Radheshyam. They all are working in a various aspects of the polymers, sensors, devices, and PDFA students. Monica is completely devoted for the biosensors. Ashish is more working on polymer area. Neeraj, he is working on optical devices, and Nirala is working again on the biosensors, and especially for the bacterial and viral sensors. Collaborations we have with our Japanese group, Tokyo Institute of Technology, Professor Maneka and Professor Kanato and Professor Pandey from the Kyushu Institute of Technology. And we have for the gas sensing, we are collaborating with Professor Kumar from Cambridge University. I'm thankful to funding agencies, DBT, DST, and recently the DST, JSPS, and SERB, Imprint, and Spark projects we have. And under these projects, we are working on these areas. So thank you very much. And I will be happy to answer your questions.
So questions we can take up together in panel discussion. I think we can proceed with the session. And you are the chairperson, you can proceed, sir, as per your choice.
I think because something happened, the slides are open. So if there are some quick question, one or two, I can take if related to this work. Otherwise, you are right that at the end, we will go.
Right, sir.
So
any questions? Any questions from anyone? Please ask.
Yeah. Okay. If no more, then we can go, and at the end of the session, we will have the discussion. So thank you once again. And Dr. Vesalius, I think your next talk, but before you start your talk, I request to Dr. Tomar to kindly introduce our next speaker.
Sure, sir.
Thank you, sir. Thank you very much for the informative talk. Now I would like to invite Dr. Vasilis Foutopoulos, Department of Agricultural Science and Biotechnology, Food Science, Cyprus University of Technology, LEMSUS, Cyprus, for delivering his talk on a potential use of nanomaterials towards the sustainable agriculture. Dr. Vasilis Foutopoulos is an associated professor in structural and functional plant biology and head of the CO2 plant stress physiology group established in 2008. He has done his PhD in 2004 from Southern University School of Biological Science and MSc in 1999 from Imperial College, London Department of Biology. His main scientific research focused on the study of nitrooxidative signaling cascades involved in the plant response to the stress factor, while emphasis is being given in the development of the chemical and biological primary technology towards the amelioration of antibiotic stress factors and promotion of the plant growth. Relevant research has resulted in the publication of the patent and the provisionally filing of a second one. In addition, he is involved in a research studying plant secondary metabolites and antioxidant response in a fruit crops during the ripening. To date, Dr. Foutopoulos is the author of more than 80 scientific papers and book chapters published in a peer-reviewed journal and with a reputed publisher. He currently serves as an editor board member in a Journal of Experimental Botany, Plant and Physiology and Biochemistry, BMC Plant Biology, Gene Frontier in Plant Science, and 5 other journals. I request Dr. Vasilis Foutopoulos kindly deliver his talk on recent advances and strategy for sustainability and regulatory aspects. Please, sir.
Thank you. Well, it's a pleasure to be here, even remotely. I would like to thank Professor Tomar for the very kind invitation to deliver this talk. As mentioned before, I will be talking today briefly about some of the research that we do here in Cyprus. Hopefully, everybody can see the slides. Yes?
Yes, the slides are visible. You can make it full screen, sir.
Yes, it is visible.
Yes,
Thank you. Thank you. I will try not to keep too long because of the, to keep on time. So effectively, I will focus on one of the parts of research in my group, which is examining the potential use of nanomaterials towards a sustainable agriculture. Just as a brief overview of what was mentioned before, we initially started off being a stress physiology lab studying nitroxidative responses of plants to a number of factors. So we have a number of published papers examining responses to drought, salt, heat, cold, and generally climate change related stressors. And recently we've moved to combinations of stressors like drought and insect attack, or also CECs, contaminants of emerging concern, because they are quite novel stressors. We also examine as a side project, as part of usually PhD thesis through a collaboration, secondary metabolism and antioxidant defense in fruit crops. So we've had so far PhDs running in topics like vitamin E biosynthesis in olive, lycopene in tomato, carotenoids in loquat, many different scenarios. And this is usually during ripening phases, but also after post-harvest storage. But the biggest part of our research is focusing on priming, and we employ a number of agents, mainly chemical priming so far, both with natural and synthetic compounds. We also do biological priming with our vascular mycorrhizal fungi and plant growth promoting bacteria. But recently we entered the field of nanomaterials. And this is exciting because priming technologies work both for stress amelioration, but also for improved growth and development under controlled conditions. So very generic slides that I'm sure everybody's aware of. Of course, as you know, plants are constantly submitted to a number of stressors, both abiotic and biotic, because they are sessile organisms, they are stuck to the ground. So by receiving multiple stimuli from the environment, as a result, several metabolic pathways are switched on in response to the accumulation of signal molecules. And the plant is trying to defend itself the best way possible. So it is quite clear that abiotic stresses are major problems in worldwide productivity. I mean, this is attested by thousands and thousands of papers published every year in the subject. And of course, the global rural land area that is affected by some kind of stresses is reaching levels of about 95%. So nobody escapes. So it's important. So when it happens, of course, the plant has things happening in the cell. So we have a major pathway involved in the production of reactive oxygen species. I will not go in detail through these diagrams, but this is just to show that they are complex mechanisms that involve multiple organelles. They involve multiple enzymes, but also multiple antioxidants that are non-enzymatic, like vitamins, glutathione, and so on. And in addition to the reactive oxygen species, we of course have the involvement of reactive nitrogen species, like nitric oxide, for example, which is again quite a complex pathway in the cell. In plants, of course, the famous nitric oxide synthase that is found in mammals is still elusive, the major source being nitrate reductase in any case, but NO is involved in numerous processes involved in growth and development, stress, and so on. So we know for a fact that reactive species are involved in a molecular crosstalk, and this is important because they are known to be toxic to the cell in high concentrations, but to actually regulate transcriptional, translational, and post-translational modifications when happening in lower concentrations. So all of this is well known, and it's a fact. What about solutions? What can we do about it? So one example, of course, is genetic modification. The problem with that is that in certain nations, and this is definitely the case where I work in Cyprus, GM technologies are not acceptable ethically or to the public. Europe in general, as you probably are aware, is quite anti-GM. I think India might be a bit more receptive to GM technologies, but in Europe, this is not the case. Now with CRISPR, things are trying to change, but it's still quite difficult to get there. Of course, we have the solution of conventional breeding, which is a safe choice, but it is extremely time consuming. And plant priming is now a different option, which is very appealing because it offers great protection in a much speedier process compared with conventional breeding. So what is priming? The process of priming effectively involves exposing plants to the stressor itself before the stress in milder conditions, thus making it more tolerant to the future exposure. So there is an analogy, of course it's not the same, an analogy to human vaccination. I say analogy because the plant doesn't produce antibodies, of course. Priming, however, can also be achieved by applying exogenously different things. So, for example, chemical priming can be done through application of chemical compounds, which then act as signal transducers that activate the plant's defense system. So this is taken from a review we did in the past, showing that effectively what happens is that when you prime the plant, especially with chemical agents, but also with nanomaterials, these agents are actually taken up by the plant. So there is signal perception involved, as well as systemic transduction of the molecules through the vascular tissues. And then when the plant in the future is exposed to a stressor, this is then leading to the induction of stress-inducible transcription factors, a number of PTMs, a number of stress-responsive genes, such as HSPs, ion channels, antioxidants, and so on, are induced. And the final result is that we reach reactive species detoxification, we reach osmotic protection, protein homeostasis, ionic homeostasis, and physiological improvement in function. So this is when it comes to the chemical priming that we have a broad publication record. But as I said, recently we entered the field of nanomaterials, which I think is extremely exciting. So we believe that advanced nanomaterials are effectively a good approach to proceed. This diagram is taken from a recent review of ours on the subject. And of course, there's many different types of nanoparticles in nanomaterials. We also work with polymers when it comes to coatings of seeds, but I will not talk about this today because of time limitation. Of course, there is a huge library of materials that can be prepared, as you are well aware, that span from organic to inorganic nanomaterials. As I said, the choice is broad. So I will just briefly mention some recent examples of publications of ours to show just the potential there is for applications in agriculture. These span from either direct application of nanoparticles, such as in this case where we examined titanium dioxide nanoparticles in plants that were submitted to, aromatic plants that were submitted to salt stress, for example. And it is great because, of course, in high concentrations, like everything, effectively, you do reach toxicity, but in lower concentrations, we did observe significant protection of the plants against the stressor. And also, it was interesting that through metabolomics, through volatilomics, GCMS analysis, basically, we did the essential oil profile of plants, and then we saw that a broad range of essential oils are actually regulated in response to the nanomaterials, direct application of the nanomaterials. So another example that could be employed is through the use of MWCNTs, basically modified multi-wall carbon nanotubes. This was employed in basil plants. All of these are published examples, so people are welcome to examine the actual papers if they would like. So in this case, for example, again, in a dose-dependent manner, we did observe protection again of the plants against salinity stress. This was supported by a broad array of analysis. Again, we did do essential oil profiling for that, and we saw a similar regulation being achieved with these different nanomaterials with MWCNTs. And this was supported by improvements, as you can see, in, for example, enzymatic antioxidants like activity of catalase, ascorbate peroxidase, glutathione peroxidase, and so on, as well as many other different parameters that I will not go in detail for the sake of time limitations. So the exciting new avenues that we're following now are effectively not through direct application solely of nanomaterials and nanoparticles, but now we're trying to functionalize nanomaterials with chemical compounds. So we have, again, through collaborations with material science experts in Cyprus and Iran for the time being, we have a number of compounds that are basically conjugates, and we try to see if this can achieve even further improved protection. So an example of a recent paper of ours that was published is this, where we actually prepared carbon nanotubes that were functionalized, in this case, with putrescine, which, as you most likely know, it is a polyamine, which has been shown to actually act in a protective manner when applied in the past directly to plants. And we actually also have publications where by applying polyamines in the plant, we reach protection. But here, by functionalizing carbon nanotubes, in this case, with putrescine, in this case, in a grapevine, we actually did observe significant protection. Extending the range of materials. Another example is polyamine-based, again, nanomaterials and silica-based nanoparticles like POSS. This is work that, again, was evaluated in a number of crop plants, and this manuscript is currently under review. This is quite exciting because we also managed to conjugate nanoparticles with fluorescent dyes in order to monitor the delivery of the nanomaterial in plant in real time, which of course is a very useful tool in our hands. And as I said, now we actually currently run in my group 2 PhD projects, where we actually, a big part of this approach is to screen all of the big libraries that are being prepared to see which ones have potential to be used for agricultural applications. So, for example, we have the production of halloysite nanotubes, which is effectively like nanoclay. And in this case, we have conjugates both with proline, which is a known essential amino acid that is involved in osmoprotection, but also we have conjugates of halloysite with putrescine, for example. This extends, the beauty of this technology is that you can actually extend it to any combination of any nanomaterial with any chemical compound. So the library we have is really extensive. We have currently more than 50 preparations that are being examined. Of course, this is very time consuming, as you can understand, but it is quite exciting because a lot of those materials do show promising results. So as a working model for this whole story, of course, we do see that nanomaterials are a very promising solution, which can be employed both in the plant through foliar application, through spraying, or through soil watering, but also there is great potential as polymer coatings. And we currently have a number of protocols, again, where we coat seeds with both hydrogels, chitosan-based polymers, and other compounds that are conjugated, again, with chemicals. And actually, a recent patent application is based on such a technology. So what we see is that we improve, of course, photosynthetic parameters, which are essential for the well-being of the plant. This involves increases in pigment content. We improve stomatal opening, especially under, as I said, stress conditions, which improves CO2 uptake for sugar production. There is improved water use efficiency. Of course, the antioxidant apparatus is upregulated. We also do see induction in secondary metabolite biosynthesis in a number of essential oils, alkaloids, phenolics, monoterpenes, and so on. And when it comes to stress markers like nitroxidative stress indicators, as well as universal markers like MDA, electrolyte glycates, of course, we always achieve significant suppression of those markers, showing that the plant is indeed protected. So closing, trying, as I said, to stay on time. I would like to thank, of course, members of my group. This is the website for people who are interested to check our publications. We collaborate extensively with many centers of excellence. So we have great collaborators in VIB in Ghent with Dirk and Zay, Ali Ferni in Golm, who actually does our metabolomics analysis, Khosrow Kasfi in the States, who is involved in the chemical priming story, Raffaella Balestrini in CNR in Italy, helping us with biological priming. and Reza Gohari in Maraghe and Theodora Krasya in UCY helping us with the nanomaterial story. Of course, I would like to thank also the funding sources that help us make this happen. And closing, I would like to take the opportunity to actually mention briefly a new journal in the field of plant science for people who may be interested, Plant Stress. Plant Stress is a gold open access journal that started operations in 2021, so we are new, but we pride ourselves in actually having a very strong editorial board and actually trying to promote high quality research in the field of plant stress that covers both stressors of abiotic and biotic components. We are already indexed in a number of databases such as ESCI, the Early Citation Index, as well as in Scopus. So we are well on our way to actually hopefully become a solid established solution in the field. And for people who might be interested, I would like to mention that, of course, being gold open access, there is a publication charge, but in 2022, we offer a 50% waiver for accepted publications, and any submissions to special issues will actually be completely waived, so they will be free. One of which is a special issue on nanomaterials in agriculture. So please keep that in mind, and I will be happy to answer questions if people present have some. Thank you.
Thank you, Dr. Fotopoulos, for your insight into this most important topic that
I cannot hear you, sorry.
All of us here. So now I would like to invite.
The voice is not coming. Say again.
Unmute. Thank you, Dr. Fotopoulos, for your insight into this most important topic that I know affects all of us here.
Thank you.
Thank you, sir, again. Now I would like to invite Professor Dr. VP Sharma, CSIR, Indian Institute of Toxicology, Research, Lucknow, for delivering his talk on recent advances and strategies for sustainability and regulatory aspect. Dr. Vinod Praveen Sharma is Chief Scientist, Regulatory Toxicology, CSIR, Indian Institute of
इनके नेटवर्क में प्रॉब्लम आ रही है कुछ?
Toxicology Research.
Ma'am, let me continue. There is some voice problem at your end. Okay, sir. So sorry about the interruption. Okay, so now I would like to invite Professor Dr. VP Sharma, CSIR India, Institute of Toxicology Research, Lucknow, for delivering his talk on recent advances and strategies for sustainability and regulatory aspects. Dr. Vinod Praveen Sharma is Chief Scientist, Regulatory Toxicology, CSIR Indian Institute of Toxicology Research, Lucknow, Uttar Pradesh, India. Dr. Sharma has done PhD in chemistry from Kanpur University in 1994 and MSc in chemistry from Lucknow University. He has received Healthcare Excellence Award in 2015 and many more to his credit. He is having several research publications in various national and international journals of repute. His area of expertise include toxicology, plastics and polymer suitability evaluation, water quality management system, NABL, GLP, ISO, human resource development leadership, motivation, team building, project management, environmental issues with relevance to toxic implications. I request Dr. VP Sharma, sir, to kindly deliver his talk on recent advances and strategies for sustainability and regulatory aspects. Please, sir.
Dr. Sharma, can you hear us, sir?
I think so. He got disconnected due to some technical issue.
Just a minute, wait. I'm just calling. Dr. Pallavi, we can proceed with the next one because I think there's a power failure. I just had a conversation with him. So
Okay,
take the
sir.
next one.
Okay, sir.
Dr. Aparna Khanna. Whether Dr. Aparna Khanna is online?
Yes, sir.
Okay, Dr. Aparna ji, good afternoon and welcome.
Good afternoon. Good afternoon. Yeah, good afternoon. And at the outset, I'd like to thank the organizers.
Please wait for 2 minutes for introduction.
Sure.
Please, I'd like to introduce you with the participants, please.
Okay, so now I would like to invite Professor Dr. Aparna Khanna, Director, MIT Institute of Biotechnology, MIT University, Mumbai, for delivering her talk on design of novel nanomaterials for efficient stem cell tracking. Dr. Aparna Khanna is PhD, MSc, MD, BA, International Marketing. She has more than 30 years of research and teaching experience. Her research interests include cell biology, stem cell biology, biomaterials and tissue engineering, virology. She is having several research publications in various national and international journals of repute. She has several achievements to her credit, like DBT Overseas Associateship in 1999, DST Young Scientist Award in 1998, Wellcome Trust Postdoctoral Fellowship in 1995, and many more. I request Dr. Aparna Khanna to kindly deliver her talk on design of novel nanomaterials for efficient stem cell tracking. Please, ma'am.
Yes, thank you, Dr. Pallavi, for those kind words of introduction. And at the outset, I would like to thank the organizers, especially Dr. Thomar, for inviting me to talk about my group's work in the area of nanosciences and stem cells. I think it's been a great learning experience, right from morning. We've had very excellent talks, and I'm sure the students and the participants are benefiting from this the conference. So I'll quickly come to the area that I'm going to talk about in the next 20 minutes, where there is a blend of both nanosciences or nanomaterials, as well as cell biology, and in particular stem cells. So let me just, since they are two little different areas, so let me introduce them a little separately, and then we will see how it all fits in together and how we can design novel materials for stem cell tracking. Now, what are stem cells, and why is there so much of talk on stem cells? So the reason for it is that they are a very unique kind of cells, and it is only stem cells that have two properties which make them apart from all the other cells. That is this one property of having prolonged self-renewal, which means that you can actually replicate stem cells. It's virtually immortal. So there is no other cell in the human body which can do that. So and the other reason why stem cells are so important is that when you give them certain cues, they actually know how to differentiate into any of the cell types of the human body. So which means that if I ask a stem cell to make a cardiac cell, it can do so. If I ask a stem cell to make a nerve cell or a liver cell, it can do so. So such is the property of stem cells, and which is why they are so unique. Now, where do you actually find the stem cells? So if you look down at the human developmental continuum, when just after fertilization, when you have the early stage of embryonic development, which is at this stage, when you have about 50 cells, this is the cells which are called as being totally pluripotent. Okay? When this is implanted, they give rise to the fetus. Then as you go down, you have the umbilical cord once the baby is born. So the umbilical cord blood, as well as the cord, houses a lot of stem cells, which are called as the umbilical cord blood stem cells. And you've heard about a lot of cord blood banking sectors where they are actually storing these cord blood because if there is the hematological or is a blood related disorder in the family, it can be used for the other sibling. So that's why they do that. And in the adult also, there are a few number of stem cells, and actually all our organs has about 0.1 to 1% of stem cells in each specific organ. Besides that, the bone marrow in cells has also, in the bone marrow houses two kind of cells, stem cells, which is why you hear of a lot of stem cell transplantations for blood related disorders. Now, one of the most recent discovery, this is what we had always thought, and this is like I said, like the central dogma in molecular biology, when you have the DNA making RNA and then you make proteins. Similarly, when you talk about stem cells, it is stem cells differentiating into a particular different cell. Okay, it's stem cell making a cardiac cell, but the reverse was never happening. It's not that you can have a differentiated cell giving rise to a stem cell. But with this Nobel Prize, which was won by Dr. Shinya Yamanaka for physiology and medicine in 2012 for a different kind of cells, which are again stem cells, which are called as the induced pluripotent stem cells. Okay, so what this means simply is that you can actually have 4 transcription factors. I won't go into the details for the lack of time, but there are just simply 4. They are called as the magic cocktail. So if you actually transduce these into a differentiated cell, you can actually make a stem cell. So, that's the path-breaking discovery. And why we are talking so much about stem cells? Because it has got multi-dimensional applications, which means, as you can see from the slide, which was taken from a company which is based out of Menlo Park in California, Geron, who were one of the pioneers who started stem cell research. So what is said here is that from stem cells, you can make actually neural cells which can be treated for Parkinson's disease, or you can make cardiac cells for heart failure, or the more recent one, which I'll be a little bit talking about later, is for like the bone and cartilage disorders. Osteochondral defects can actually be replaced by using stem cells if there is a defect. Okay? So this is the reason why stem cells are talked about. And again, the potential of stem cells is you can actually see it in the lab when you get embryonic stem cells and you try to differentiate them into neural cells or to cardiomyocytes, to pancreatic cells. You can actually see that it happens. Now let's introduce the problem. Now, so we've talked about stem cells. We've defined the importance of stem cells. Now, what is the problem in stem cell research? So one of the major problems that when we're working on stem cells, what we've realized is that even if the clinical data, you've seen a lot of stem cells that have reached therapies to certain extents, but there's always been a question mark among basic researchers that cells, unlike a drug, they're actually injected into a human body. But if you do not know where these cells are going, okay, they might do the bit of repairing and regenerating tissues, but you need to track them, because if you're not able to track the cells where exactly they're going, then you have a problem. You need to do the safety studies. So that's where we came from, where we tried to bridge the gap between basic research and translation research, where we can see whether we can have sensitive, non-invasive imaging modalities, which can be, which with that you can get an idea that how you can use it for stem cell therapy. So keeping that in mind, we went on to use nanoparticles, and we realized that when we did our literature search, that there were a lot of research going on in magnetic iron oxide nanoparticles, simply for the reason, as you know, magnetic iron, you can use it for MRI imaging because they have the properties which can be used there. But one of the problems that one has to consider when you're looking at labeling stem cells with nanoparticles is that the cells will divide. So once the cells divide, then there is a dilution of the label, and then you may not be able to catch it on. So these were some of the considerations that we had to work on when we started this work some time ago. And in fact, there was only one which was approved by the FDA, US FDA, which could be used for tracking, which was called as Feridax. And that is basically your magnetic iron oxide nanoparticle. So how did we start doing our work? So what we first, now giving this introduction, now what we had at hand is that we had to make nanoparticles, and the nanoparticles are toxic, the bare nanoparticles. So we had to we had to functionalize them and also make them biocompatible. Then we had to isolate stem cells, and we used cord here for isolation. Then we had to study the interaction of these, because if the nanoparticles are put into the stem cells, we had to check out the stability and also the toxicity that nanoparticles might have on the stem cells, because we ultimately, the last bit of our work was to find out that whether all of this is not enough to say that in vitro it's working, but we've also wanted to see in vivo, when we have animal models, do they do what we want to do? So the first part is synthesis of these nanoparticles. We used simple co-precipitation method, and we synthesized the nanoparticles, the magnetic iron oxide, as you can see there, here as dry powder. We also use, because we know that the surface ratio being high, you also have the aggregation problem that happens in nanoparticles. So to maintain its colloidal stability, we had to also use a CTAB, which was used for the coating and to avoid aggregation. Further to the coating, okay, these are some of the results that once we did it, we had to characterize that you have proper nanoparticles or not. And one such is that, as you can see, the XRD shows certain peaks which have to be there. These characteristics peaks were seen, which meant that the crystalline nature of the magnetic iron oxide particles were maintained. And we also did one other important thing is that when you start using all these coating agents, you might lose its magnetic property, because if you don't have the magnetism there, then you cannot use it later on as MRI imaging. So we also did that. So you can see even with the coating, the magnetic properties was well maintained with the iron oxide nanoparticles. We did a lot of characterization, which included FTIR and a whole lot of other ones, but I'm just showing you the ones which are more important. Then we looked out at TEM imaging to see how these nanoparticles look like. And we can see if you see the dispersion, we had most of our nanoparticles coming in the range, the bare ones, between 7 to 9 nm. But as I mentioned earlier, the bare nanoparticles are not good enough for us because they will have toxicity on the cells. So we coated it with human serum albumin for the simple reason that we know that that's a biocompatible one when we later on want to use it for either clinical trials. Of course, much before we have to try it out in vivo, this would be a good option. So when we did the coating of the nanoparticles, the coating size was about 50 to 70 nanometers, and we had this was the size distribution, which is also good enough for taking on for further studies with the cells, with the stem cells. So this now our particles are synthesized, they are characterized, they are coated, and all is well. Now we go on to getting our the stem cells. So we isolated the stem cells from human umbilical cord with the proper ethical permission. We received the cord. As we all know, a cord is a medical waste. In fact, so we are making use of the waste. In fact, we are isolating stem cells from the cord. We cut it down to pieces, as you can see here, and we exposed each of them, and each of these actually give rise to these stem cells. What you see coming out of this this is called as the explant method. You can see a lot of these cells. These are the mesenchymal stem cells, so as to say. And then from there, we went on to do a little bit of characterization of the stem cells. This is as per the standards. And what it's showing here simply is that you have through a flow cytometry analysis, we can show that the major population of the cells house the markers which are very important in the mesenchymal stem cells, which are the CD90, the CD73, and CD105. So having this said, that we have a pure population of mesenchymal stem cells. So now both we have our nanoparticles synthesized, we have them coated, we have them so-called in vitro studies that they are biocompatible. We also have our stem cells, which we have characterized. Now we will look at the interaction between the both. So we did a lot of in vitro studies. These are basically cell lines. This was used as a control cell line, and these are our stem cells, where we did a simple incubation with magnetic iron oxide nanoparticles. And we could see that these are just showing you that this is a Prussian blue staining, which shows that these nanoparticles are housed, they are able to enter with just mere incubation. We can see that these particles go in. We did a whole lot of, just show you this one also. Okay, this is an image which was done by Cryo-TEM, which you can see this is a single cell actually. And you can see this is the cell with the nucleolus and the nucleus, and this is the cytoplasm. And this is what happens actually at different stages of internalization. We could see that the nanoparticles, these are the, the magnetic nanoparticles. You can see that they are only in the cytoplasm, which is what we want. We obviously don't want it to go in the nucleus. So we can see that they are there, very much there, and they go in the endosomes. So we could prove that also. And also another important part, as I said, that these nanoparticles are known for its toxicity, and the stem cells are again very fussy and fastidious. So we needed to see that there was no toxicity seen in the cells. So we did a MTT and an LDH assay to see if the membrane was leaky because of cell damage. Then you would have expression of the lactate dehydrogenase, but that was not the case. And the viability of the cells, it was obviously less when it was bare, which means that bare magnetic iron oxide had toxicity issues, but the HSA coated one showed over 90% of viability. Now in the next just 5 minutes, I'll finish this part of the work where what we did, this was very interesting because the preliminary work which we did for about 3 years, which helped us tell us that yes, we were able to synthesize nanoparticles, we were able to characterize them, we were able to isolate stem cells and check the interaction. But I think the more challenging part was actually to see that whether all of this, how does it fit into an animal model, because that is one step getting closer to clinical trials. So we here we used nanoparticles which were different, not the HSA one. We used lysine functionalized nanoparticles, and we used the bone marrow derived mesenchymal stem cells. We did the routine in vitro studies where we could see that there was no toxicity on the cells. And then we had an animal model wherein we created a defect, and this is basically the drilling of the hole. This is in the knee joint. So it's the defect which is called as the osteochondral defect. Osteo is for the bone, chondro is for the cartilage. So it's a bone cartilage disorder, and the mesenchymal stem cells are known to repair this kind of a disorder. So that's why we tried. This was a the first model that we wanted to look at. So this was done. And we again did the same studies first. We tried to check out whether there was, first of all, the cellular uptake, whether the nanoparticles were taken by the stem cells, which it did. And there was, as you can see, that there was maximum almost 85% in 24 hours. We could see that the cellular uptake was the stem cells were taking these nanoparticles. And also when we did an in vitro MRI imaging, we can use, these are the signals which show the hypotense signals, because if the signals are not generated, because we do not know, is this enough amount of iron in the cells to actually generate a signal? Because if you don't know that, then you cannot go ahead with that. So then we did it, and we of course published that data, which showed that lysine-coated nanoparticles were good enough. And then also we created the model which I talked about, and we saw that there was regeneration of hyaline cartilage in those cells. So it was not enough that it was just the model that was created. We used the stem cells. We saw for almost 24 to 48 hours, we could actually track the stem cells, and we could also see that there was repair of the osteochondral defect in these animals. Okay, so I think this was a paper which was very recently published, where it actually showed that we could bring about the regeneration of hyaline cartilage in the osteochondral defect of the cells. I think that's my last slide. I'd like to thank a lot of people involved in this work. My PhD students, Purva, Ruchita, Swati, and Akshata. Most of the work that I presented are of two of my students, Purva and Ruchita. My funding agencies, DST Nanomission, who gave us the first part of the grant for the first part of the work that I presented, where we actually showed the interaction of the stem cells, and the later part where we had the animal model generated, and animal work was funded by the Department of Biotechnology. Professor Dr. Bahadur from IIT Bombay, who's been my PI in all the projects that I spoke about, and Dr. Sudeshna Chandra, my clinical collaborator, without whom I wouldn't be getting my cord samples, Dr. Preeti Galvankar, and at ACTREC, a few of the facilities that we utilized, and support from Dr. Preeta and Dr. Sanjeev Bagmare. So with that, I end my talk, and if there are any questions, I can take them now or later. And I hope I've stuck to the time, since it was 20 minutes. So I'll be happy to take questions if there are any.
Very much. Thank you very much for taking your time and that restricting yourself with the time limit.
Yeah, sure.
Thank you so much, Dr. Aparna.
Thank you. Stop sharing. Yeah.
Now I would like to welcome Professor B.P. Sharma ji, who could not join in the beginning of this particular session because of some power failure. Sir, I welcome you on this platform. Good afternoon, sir. You are muted, sir.
Are you able to see my slides, please?
Yes, sir.
Okay.
Very much. Okay, Dr. Pallavi, introduce sir, then he will proceed.
Yes, sir. Now I would like to invite Professor Dr. V.P. Sharma, CSIR, Indian Institute of Toxicology Research, Lucknow, for delivering his talk on recent advances and strategies for sustainability and regulatory aspects. Dr. Vinod Praveen Sharma is Chief Scientist, Regulatory Toxicology, CSIR, Indian Institute of Toxicology Research, Lucknow, Uttar Pradesh, India. Dr. Sharma has done PhD in Chemistry from Kanpur University in 1994 and MSc in Chemistry from Lucknow University, Lucknow in 1984. He has received Healthcare Excellence Award in 2015 and many more to his credit. He is having several research publications in various national and international journals of repute. His area of expertise include toxicology, plastic and polymer sustainability evaluation, water quality management system, NABL, GLP, ISO, human resource development leadership, motivation, team building, project management, environmental issues with relevance to toxic implications. I request Dr. V.P. Sharma, sir, to kindly deliver his talk on recent advances and strategies for sustainability and regulatory aspects. Please, sir.
Okay. Very good afternoon to each one of you. And at the beginning, I would like to compliment the organizers for a wonderful international conference on recent advances in biotechnology and nanobiotechnology. This crucial concepts and the importance of it is highlighted at the different fronts, but I will try to link with the toxicology and how the things can be taken for sustainability and regulatory aspect, because in the modern scenario, when we have to take up the sustainability issues, these becomes vital. if a basic point of toxicology has to be seen, it has a multidisciplinary aspect. It involves the study of the adverse implication of substances on living organisms and with emphasis on the different translational scientific development. In the modern era, when we are thinking for a local and local aspects and the international excellence point of view, we have to understand that there is a direct understanding is needed for the dose effect relationship, and also to understand how the toxicology becomes very important in the terms of efficacy, potency, how the different type of toxicants are coming into scenario. And we have to remember that Parcellus said in the beginning that it is the dose which is very, very vital. It is the dose which determines the impacts and the different levels. Even the post-COVID situation or during the COVID, we have to take into consideration the scenario which were in front of us. There were different type of materials in front, but we have to think about the toxicity of the different drugs or the environmental issues. So these forensic sciences, legal aspect, general food part, it becomes very vital. When we think about the drug point of view, we have this different organization at the global level which are working in the drug development. The most critical part is characterization, standardization, validation, and also to assess what are the risks and how we can control those risks. The new concept and very effective is about the QSR studies, for which all this organ gathering is knowing about these points. But we have to think, is it only the drug? No, it is not only the drug, but we have to also see about the different aspects of cosmetics, food, fast moving chemicals. And in the present scenario, there is a need of the different R concepts, which is on replacement, refinement, reduction. So even the toxicology is making a great change in the modern scenario, because we have to use the minimum amount of animals in the regulatory part, at the same time to fulfill the requirement of international bodies for the guidelines and specifications. So these wonderful rat or mice, guinea pig look beautiful, but we have to understand that the torture is not done, and it is the rationale is there, which is of the CPCA norms or for the human and ethical committees which are in place in the institutes or the different universities. So now coming to the next point, that how to assess the risk. The first basic point is identify, classify, evaluate, quantify, and measure it. As a quality assurance personnel, I would like to say that it is important to have the risk assessment so that we can minimize the disaster and go to the zero management, because the zero tolerance is the demand of the day. We have to understand about the mitigation, vulnerability to the different aspects. It starts from water and it goes to sanitation, hygiene, health, and environmental issues. So how we can do it? Is it just taking the drugs or the wonderful food? No, it is, the answer is that we need a safe food, compatible one with high nutritious value. And when we are doing this, we have to really assess that the food which we are eating is safe enough. There should be a good ISO or HACCP or ISO 17025, in medical IS 15189. These are the requirements. If I have to go for any testing of COVID or any of the vulnerability of any drugs and other things, or any complications which may happen because of the climatic changes, we have to think that are we going for the SDG goals? And what are these SDG goals? All of you will be strange to know that in the global scenario, still India is lacking behind. It was earlier at 117 position and today it is at 120 position. So this is alarming. And how we can think of the betterment when the education is affected, the recession is there. It does not mean that I am negative in these aspects. I am very positive about it, but we want to say as a toxicologist that drive with caution. And we have to understand the national and international guidelines. There is an organization for economic and cooperation guidelines, the different pharmacopoeias. We are involved in the framing of the guidelines as well as execution of it. So when we talk about the nanotechnology, nanomedicine, how we can have the better strength in composites, the chairperson, Dr. Rajiv, and others, they have highlighted about these issues at the various fronts. But our aspect comes into compliance, that are we really just framing the guidelines or we are going to the compliance? Are we just abiding with the rules to some extent, or it is 100%? So when we have to think about the social, economic, and sustainability or environmental sustainability, we have to think about what is the demand and what is the supply. Are these red books, blue books, green books, they have been framed by the ATSDR and others. But the main critical point at the global level is many of the countries are still not following it. And if they are trying to follow it, it is halfway. So there is a big gap between the implementation and the formulation. The next point comes to our mind is what are the new emerging chemicals? Like microplastic, pesticides, pharmaceuticals, different type of heavy metals, and their direct and indirect effect. So we have to think about the water treatment to real supply of the safe water. And next come in scenario is how the issue of microplastic. We have the wonderful beaches throughout the world, but when we go at the coastal region, we find there are a huge number of microplastic. And by 2050, it is said that there will be more plastics rather than the fishes or the aquatic system. So we have to understand the cry of the flora and fauna. The landfilling is going on extensively. So we need, we have the systems for detection, but are those systems really viable? Are they cost effective and technological feasibly? So we have to understand about the issues which are related to the landfilling, wastewater, air blasting. There was the issue about the PM 2.5 and PM 10. What is these PM 2.5 and 10? Is it just the environment? It is our car and vehicles which are moving on the road? No, it is also affecting our pulmonary system. So we have to understand the implications and at the same time think about the another critical issue when we are talking about the nanoparticles, nanomaterials. These materials are good, but on the other side there is an issue of the waste management. Biosensors are good. And what are biosensors? Which Dr. Rajiv has just said that they are wonderful analytical devices that are able to convert a biological response into an electric signal. But we have to understand how the mechanism is, what is the essential component, and are we really understanding the nucleic acid, the mechanisms, the microbe-based group, and what are the impacts? So we have a wonderful setup. Earlier, we were just doing first, second, and third stage of treatment. Now, today's demand is we have to go for the fourth treatment stage in the wastewater treatment, which includes the aspect of the emerging pollutants, the new type of materials, and the microplastic, which I said in the beginning. Now, coming to the effects and understanding the reality of the biodiversity. Is it just the controlling through the sensors or the monitoring devices or the control for the meters? Meter is to control us, to regulate us. And regulator, when we use the word regulation, the regulation has to be ultimately coming from the heart. It is not just a physical thing. It has to come from the heart, and it has to go for reduction of cost, design new designs, out of box thinking, taking the things to the market. And thus we have to understand that the global annual consumption of plastic, on one side we say it is a bane, but on the other side this is growing like anything. So we have to see that can we go for bio-based? Can we go for sustainable solutions? Yes, answer is through the aspect of green chemistry, new approaches to designing chemicals and to have the chemical transformations that are beneficial for human health and the environment. So all this wonderful group, today and tomorrow you are discussing about the nanotechnology. I would like to say, try to imbibe the concept of green chemistry and the different principles of the green chemical approaches. Here, I would like to say that at IITR, CSIR is a wonderful laboratory setup with 37 laboratories throughout the country for your satisfaction and to giving the best output. We have a good ACSIR facility. At the same time, we have the testing facilities for the GLP compliant and NABL compliant facility at IITR Lucknow. You can take this advantage. It is for the benefit. It is the public money through which we have come up, and we want to have the maximum benefit to the society. So these EU compliances, GLP, whatever we don't know, we will share with the universities like MIT University and the others and the global partners like Michigan University. We had a presentation from the different stalwarts of the field from the different country. So now we have to think as a one partner, one unit as at the global level. That means Vasudev Kutumbakam. And when we say Vasudev Kutumbakam, we have to utilize the facility of toxicological studies or for R&D or for any other purpose and take the benefit to the stakeholders, which is a common man. So the sustainability aspect involves the different type of drugs development, and these drugs should be safe enough. We should understand about the signaling, cellular mechanical aspects, control of tissue growths, how they are going, what is the regeneration, what is the proteomics and genomics behind it, and how we can have the interdisciplinary basic sciences coming to the forefront of the common man. And when we say about these things, we have to understand about the contracts, our quality system at the different levels. There are wonderful books coming up on the different aspects of compliances or COVID, other drugs and other things. We have to see that how the globalization of regulatory standards can be done through the pharmacopoeias and how these demands and supply can really go to the corporate level, because their corporate social responsibility is very good thing, but we have to see in the different facets of our development of bioplastics, the different type of materials which have been proposed by various our speakers. There is a concept of PDCA, that is Plan, Do, Check, Act. Whatever we plan, it should be performed, it should be counter-checked, and ultimately it should go to the beneficiary for not only the human being, but for the future generation and for our biosystems. When we say about the zero waste, there is a need of zero waste to be generated. That means no waste to be generated, in fact. And how we can prevent it? Through the designing, building the good materials, using the concept of reuse and other things, and also seeing that our future generation does not say that the previous generation has created an atmosphere which was highly toxic. And we have to think about the concepts of circular economy when we have to go for the business point of view. So these things also take concept to the non-adulteration. That means when we are taking a food, these should be non-adulterated, whether it is for the wheat, rice, or any other item or any fruit. From outer side, it may look as very fresh, but if we go into the details of the nutraceuticals part or properties or shelf life, we have to think how we can increase the shelf life. Different IITs under the DST, DBT and other projects, we are working hard, and we have to see how we can go for 2030 and 2050 scenario in a good picture. When we say about the sanitizers, the simple aspect, but we in the beginning of this pre-COVID situation, even the sanitizer was at the shortage. So we have to think how we can have a cost-effective, at the same time non-toxic and non-harmful sanitizers, because in the market there was a thrust of all the different sanitizers, the masks, the PPE kits and other things. So these the personal protective equipment has to be available. They are to be in a sufficient quantity, but they should not go into the beaches and the ocean. These oceans are not our only property, they are for the future generations. So now new chemicals are coming into the market or into the scenario, which every one of you know about the phthalate, bisphenol A, pesticides, solvents from the different sources. Even whether we are talking about the water, air, soil and other things, we find these type of new EDCs are coming into the scenario. So coming to the EDCs, just as a glimpse, in order to save time, I am coming to the end of the presentation, just giving a view to you that how we can think for a better and clean environment, and taking into consideration the new type of endocrine disrupting chemicals which are coming through the different sources. When we consider about the different disease and disease pattern, we have also to think about the specification, their chemical constitution, their technical part of it, spatial distribution, and the measures which we can take from one end to the other. So the woods are lovely, dark and deep, but we have promises to keep and mice to go before we sleep and mice to go before we sleep. So when we see about the solid surface, or the concepts or the materials or the particles from the nano to the micro scale structures, they may be created on the layer to stop water. And we can understand from these birds, which are visible at this slide, which says there is a tap, there is a source of water, but still there is no water in these taps. So we have to understand these beaches look good, whether it is of Andaman, Nicobar or of US different countries other than this. We find that we have to have a situation where we can understand from the lotus leaf how the nanotechnological concepts have come up. All of you who are well connected with nanotechnology, they understand the beauty of the lotus leaf. So I would like to say that MIT University and the IITR, the CSIR family and the global scenario, we are together for the development of the world on the different aspects of safe food, safe water, and healthy life. And when we say healthy life, I want the nature to change and also behavior and approaches to change. The psychology and the philosophy and the physiology of the human being should go together for a better world. Thank you a lot for your patience hearing, and I thank all the organizers for giving an opportunity. Even I would like to thank all the previous speakers and the different platforms through which the students are wanting to go ahead in this direction. Thank you a lot.
Thank you very much, sir, for providing a new insight to understand various strategies for environmental sustainability and their regulatory aspects. Thank you so much.
Thank you.
Now, I would like to invite Dr. Seema Nara, Associate Professor, Department of Biotechnology, Motilal Nehru National Institute of Technology, Allahabad, Uttar Pradesh. For delivering her talk, Dr. Seema Nara has done her PhD from Indian Institute of Technology, Delhi in 2009, and MSc from Maharshi Dayanand University in 2004. She has received several honors and awards to his credit, like Dogra Award in 2008 and many more. Her area of interest include immunodiagnostics, nanobiotechnology, development of diagnostic assays for microbes or clinically important molecules, nanoparticle-based diagnostic systems, synthesis and use of nanoparticles and their use in diagnostics. I request Dr. Seema Nara to kindly deliver her talk on aptamer as detection agent in vitro diagnostics. Please, ma'am.
I hope I am audible to you. Am I audible?
Ma'am, your voice is not that clear.
Is it coming now?
Yes,
am I
absolutely
perfectly audible? Okay.
Yes.
I will just share my screen with you. So I suppose the screen is visible to all. Can you confirm, please?
No, ma'am, not yet.
Not yet. Is it?
Yes, ma'am, absolutely.
Visible?
Yes.
Okay. Okay. Thank you so much for the generous introduction. And I would like to thank Professor Tomar for inviting me here to share the lab work that we are doing at our end. The topic of my talk today is aptamers as detection agents in vitro diagnostics, as we are doing a bit in this direction. So I thought I should talk on this. To start with the brief introduction on in vitro diagnostics, though the session is already on sensors and Professor Rajiv has already given a talk on this. But still, in vitro diagnostics, as we all know, includes the tests that can be detected, that can detect disease conditions and infections outside the test tubes, particularly on the blood samples and other bodily fluids. And these tests can be done in the laboratories, healthcare facilities, or even at home. And we can perform these tests using small to large equipments outside the laboratory or maybe inside the laboratory. And the market of in vitro diagnostic tests that basically could be divided into 4 segments, which includes hematology tests, microbiology-based tests, molecular tests, and then one section is for immunoassays or biosensors. So if we talk of the immunoassay segment, this immunoassay, while developing an immunoassay, the key elements that we focus on are, there are 4 key elements. The first one is the capture agent, which becomes important because these capture agents, they bind to the analyte or the target of interest, which we want to detect in the analyte with great specificity. So how much specifically the analyte is being detected, that is governed by these capture agents. For example, antibodies that we all know. Then the second key element in developing any immunoassay or a diagnostic assay is reporter or signal producing agent. The entity with which we could produce the signal and with which we could confirm that some analyte and capture agent interaction has taken place. So how to record that this interaction is being taking place? For that, we need a reporter or a signal producing agent. And these signal producing agents could be enzymes, they could be nanoparticles, fluorescentides, and many more. And these are the elements which are actually responsible for introducing sensitivity to the assay. That means how, at what level we are able to detect an analyte in the bodily fluids with that assay. What is the lowest limit of detection? So that is being conferred from these reporter or signal producing agents. Apart from this, solid surface, which could be optional because there are assays wherein we can develop assays without using a solid surface, but in most of the immunoassays we see that microtiter plates are being used as a solid surface, on the surface of which we immobilize the capture reagent or the analyte also in some formats. But this is an optional parameter. We can use magnetic beads also. We can use these nitrocellulose membrane also as a solid surface. And then the fourth one is, of course, the sample, the test sample in which the analyte needs to be detected. So these are the four key elements which are an integral part of an immunoassay and of other diagnostic assays in which we are using these antibodies or nanoparticles. Now, the process of detection is, I would like to summarize it as the capture agent that interacts with the analyte, and that is, this interaction is being noted by the reporter, which is attached to some biological entity. And this, the binding of this reporter to the biological entity, that is then read out using some device. So some signal readout device is mandatory to quantify that analyte in this body fluids. So for quantification purposes, or if we want to quantify the assay, some signal readout device should be there and a display unit should be there, or else we can go for qualitative assays. So here the capture reagent I would like to focus on, because these capture reagents, they are also known as molecular recognition elements, and different types of capture reagents have been explored over years, like antibodies, receptor enzymes, etc., for developing these assays. But antibodies, of course, have scored advantages over others. However, despite the heavy use of antibodies in developing diagnostic assays, they suffer from certain disadvantages or they have certain limitations. Like to produce the antibodies, we are largely dependent upon animals, though antibody engineering has to some extent solved the purpose, but yet these are costly. So dependency upon the animal for producing antibodies, then the process of production of specific antibodies, I would say monoclonal antibodies, is time consuming and costly. So that increases their cost also. And then we could see batch to batch variations in the antibodies because they are coming from an animal system. So these batch to batch and lot to lot variations could be seen in polyclonal antibodies. Then being a protein, the antibodies are sensitive to extreme pH and temperatures. They could aggregate, they could denature also. So the use of antibodies in these extreme conditions is slightly limited by this fact. Again, the short shelf life, because of which we have to maintain them in a cold chain, continuous cold chain. And like that, they suffer from various disadvantages, though they have advantages also. But there is always a quest to overcome these advantages, or if we can have any capture reagent that could at least give equivalent results to antibodies. So in that line, aptamers have come into pictures. Now, I would say then what are aptamers? One could wonder that aptamers, they are also known as chemical antibodies. These are single-stranded nucleic acids, DNA, RNA, and nowadays proteins are also being explored as aptamers. But largely DNA, single-stranded DNA and RNAs have been used as aptamers. And these aptamers, they bind to a wide range of molecules as target with great specificity and affinity. And the binding of the aptamers to their target, that happens by adopting a specific three-dimensional conformation, which is because of the formation of some stem loop structures, some G quadruplex structures, etc. So as seen in this figure, single-stranded molecules, when they approaches in the vicinity of the target, they adopt a particular folding, and after that they bind to their target with strong affinity. So this is the concept of how aptamer recognizes their target. But how can we screen a aptamer that could bind to its target with great specificity? These are some of the possible targets of the aptamers. Principally speaking, the aptamers till date, they have been screened to bind against variety of targets. The aptamers could bind to large molecular weight proteins. On the contrary, they can also be screened to recognize very small molecules like drugs, like thrombin molecules or so. They can also be screened to bind to complete cells, whole cells, bacteria and viruses. So variety of targets could be used to screen the possible putative binding aptamers. Coming on to a brief history of aptamers, the concept of aptamers was first come into picture when in 1980s some research was done on HIV and adenoviruses, wherein it was seen that small structured RNAs could be encoded by these viruses, which binds to the cellular proteins. Means binding of RNA to the cellular proteins was one thing that was this was the thought that have further been explored as aptamer, that RNAs can also bind to a protein target with high specificity and affinity. Later on, the genesis of the technology with which the aptamers can be screened, that is the Celix technology, that was introduced in 1990. Now, in 1990, the scientists Professor Larry Gould and Craig Turek, who was the PhD student of this graduate student of Professor Larry Gould, was working on his thesis, and this group, they developed the technique Celix at University of Colorado. And what they did was they started from a pool of somewhere around 65,000 nucleotide sequences and tried to screen some possible sequences which could bind to their target protein. And they were successfully able to screen such target nucleotides which could bind the protein with high affinity. And for that, they had done the patent for the Celix technology in 1993. And because of that patent also, the pace in the aptamer research that was geared up after in late 2000 decade. Parallelly, Andy Ellington and Jack Scotia's working independently of this group, they also devised the same process simultaneously. And Ellington was the one who coined the term aptamer for such sequences which can bind to their target protein. And around the same time, Gerald Joyce at Scripps Institute, California, also developed Celix technology around the same time. So this was the seminal work of these scientists that introduced the technique to or technology to screen the aptamers which could specifically bind to the to some specific target protein. Now the question comes that how these nucleotides or single-stranded DNA or RNA molecules could be screened that could bind to a target with an affinity that could at least be equivalent to the antibody analyte affinity. So this was the technology which is named as systemic evolution of ligands by exponential enrichment, which is known as Celix technique. So in this Celix technique, this technology, it starts from a random pool of oligonucleotides. The sequence of the oligonucleotides in that library is very random. And this DNA library consists of 10 to the power 13 to 10 to the power 15 random oligonucleotide sequences. So we start with such a large pool of random single-stranded DNA library. And with that library, we start a process, a cyclic process or iterative process in which three major steps are there. The first one is the binding of the nucleotide, these random library with the target. The second one is the separation, that is the separation of the bound fragments, the fragments that have bound to the target molecule from the unbound segments. Third one is the amplification, that is amplifying those segments which binds to the target molecule with high affinity. And so in this way, this cycle is repeated for at least, I mean, it varies from protocol to protocol, but generally 7 to 8 cycles are minimally conducted, and we can go up to 12 to 13 cycles. Now, elaborating more on this journal select cycle, I would, and it's there, the step one that consists of the design of the library, which is a key step. We have to very carefully see that what should be the length of the random sequence that is there in this library. And this random sequence that is flanked on both the sides by primer binding sequences. So the selection and design of the library is very important. Once the library is there, then the target molecule which we would like to detect, or for which we want to develop the diagnostic assay, that target molecule is prepared and that is incubated with this library, single-stranded. If it is a DNA library, then we could screen single-stranded DNA aptamers, or else we can also go for single-strand these RNA aptamers. So once the target is bound to the library, thereafter, after incubation, the important step is separation of the bound from the unbound. After this separation, the bound fragments in the step 5, they are amplified using PCR conditions. And in the 6th step, since after amplification, it's going to be a double-stranded molecule. So we have to, separate the single-strand DNA, which is of interest to us. And that needs to be separated. And if it is an RNA aptamer, then it again has to be, reverse transcribed. And then these DNAs, single-stranded molecules, are again set for second round of selection by incubating it again with the target, So with every round of selects, we keep on increasing the stringency. That is like, we keep on reducing the concentration of the target that we are using. The ratio of the target is to amplified fragments. We can reduce the time of incubation of the target in the aptamer, so that we can get only those fragments which bind to the target within a short span of time with high affinity. So in this way, we can go for it. In between, we can also introduce counter selection rounds so as to increase the specificity of the aptamers. In the counter selection round, we can incubate the aptamer fragments with the possible cross-reacting molecules as target. So we can eliminate those sequences. So with this journal strategy, after all the rounds of selects are completed, then the sequencing of the final round products, they are that is done. And from that sequencing results, we make out that what are the sequences that are present in high frequency. And so we can get those sequences synthesized and then explore their binding ability with the target in the in vitro conditions. So this is the general strategy for selects. So over the period of time, the separation is very important here. How do we separate the bound fragments from the unbound fragments? So based upon that, different type of selects methods have been reported. Like in certain cases, the bound from the unbound fragments can be separated using capillary electrophoresis system, wherein on the basis of the charge, the molecules are separated. So the bound fragments can be separated from the unbound because of the difference in their electrophoretic electrosmotic mobilities. So that kind of select strategy is known as CE selects. Likewise, if the the target is immobilized on some magnetic bead, and any amplified fragment, if binds to the target, then those magnetic bead bound target aptamers, they can be, isolated using magnetic separation. So such kind of methods are called as magnetic bead select. So in this way, the kind of separation strategy that we are using, it defines the type of selects that we can do. Likewise, people also use affinity chromatography approaches to separate the bound and unbound fragments. So these aptamers nowadays, they have found application in almost every sphere of life. We have used them for diagnostics applications, in forensics, in food applications like aptamer-based analysis. They have been used in therapeutics. Their use has been demonstrated in drug delivery systems. In 2004, the first aptamer Macugen that was approved by FDA for therapeutics use. So they find application in various the segments of biology. So here's a comparison of aptamers with antibodies. How can we, on what parameters we can compare the aptamers with antibodies? So if we see the time of selection, if we go for, monoclonal antibody production, it takes several months for, screening a good antibody. But for aptamer selection, we can just do this on using several weeks only. Then the cost of selection is also very less, because once screened, the synthesis of a short oligonucleotide segment, that is not that costly as compared to antibodies. So the cost greatly is reduced using an aptamer. And since the synthesis is done chemically, so batch to batch variations are also bound to be less. Similarly, these being like DNA aptamers, if we use in diagnostics, they are very stable as compared to the antibodies. And they can be, they are amenable to modifications. We can modify their 5' 3', and we can introduce functional groups to these molecules, and we can introduce them on the surface of nanoparticles also. We can screen aptamers with high specificity and affinity with that could be equivalent to antibody also. So in this way, aptamers have proven to be a good capture reagent as compared to antibodies. And therefore, we had also started research on aptamers since we are working on diagnostic assays. So we initiated research towards screening of aptamers against certain molecules. So in our laboratory, what we have done is we have selected single-stranded DNA aptamers for binding and recognizing CA125 ovarian cancer biomarker. So we have taken this biomarker, and that was a part of a PhD thesis, and screened good quality DNA aptamers against CA125 protein. Then we have also successfully completed a DBT-sponsored project on screening of aptamers against RBP2 protein of canine parvovirus. And presently also in our lab, we are the work that is ongoing is on the selection of aptamers against Staphylococcus aureus enterotoxins A and B. So for the first two works for CA125 and RBP2, the strategy that we have used for aptamer selection was membrane selects. And for that, the library that we had used was N30 library. So what does N30 depict? It says that the central region of the library, that is of 30 nucleotides, is the random region. So the randomness that falls in this region, and the flanking regions are for primer binding. So the size of the library was N30. And this diagram, it shows the overall protocol for the membrane selects. So here we used 0.2 micron size membrane to filter out the target bound aptamers from the unbound aptamers. So that is a strategy that we have used, and rest of the protocol, general protocol, is the same. Then when we have screened these potential aptamers against CA125, we have published that work for aptamer analysis in Frontiers in Chemistry, and then we have used them for developing a lateral flow assay for CA125 detection, and which has been published in Biosensors and Bioelectronics. And last year only we had filed a patent for the same work also. So briefly speaking about that work, the aptamers that we designed for CA125, we have done first the in silico analysis of those sequences that we have obtained after cloning. And the secondary structures of the aptamers were then constructed, followed by the tertiary structure, and we do the docking of the aptamers with the simulated protein, and carried out the molecular dynamic simulations of that protein. And from those studies we have got an idea that, because if we, after sequencing, suppose if we get some 20 or 30 aptamer sequences which have shown which are present in good number, then it is really difficult to get them chemically synthesized and then test each one by one. So to reduce our work, what we have done is we have first characterized them using in silico tools and tried to figure out some top 2 or 3 sequences which showed good binding with the target in our bioinformatics work. And then we had also checked the cross reactivity of those aptamers sequences in silico only with the related proteins that could be present in the human serum. So after that, when we zero down that some 2 sequences we had figured out, one of which is aptamer 2.26, which is given here, we found out that this aptamer sequence that binds with the target protein with very good affinity. Then we had tested its stability in the serum, because finally the aptamer has to be exposed to the human serum only, and it could contain any nucleases also. So for that, or it could have high salt or ionic concentration. So we determine the stability of the aptamers in the human serum, and after finding out that this aptamers was working good, we used it for developing the assay. For assay development, what we did is we went for gold nanoparticle synthesis only, and we have synthesized 10 nanometer size gold nanoparticles and then conjugated them with our CA125 protein, not with the aptamer, and we characterized them, and we found that after conjugation there was slight increase in the size of the nanoparticles. And then another strategy that we have used here in this work was we use the intrinsic enzyme activity of these gold nanoparticles. See, various nanostructures are found to show catalytic activity. So, and that we have demonstrated in our laboratory, and we have published in that context also. So we have determined that these gold nanoparticles, they possess certain peroxidase activity, and we have optimized the parameters of substrate and the concentration that should be used. After optimizing that, we try to develop a lateral flow assay using aptamer as a capture reagent and the peroxidase mimic gold nanoparticles as the reporter molecules. Since we wanted to develop a lateral flow assay, a strip-based assay, so normally we see that the strip-based assays are, qualitative assays giving signals in yes or no. But here we wanted to have a quantitative type of assay so that we can really have a value that what is the value of CA125. So in that case, we have designed an in-house device also to read the intensity of the spot that is developed on the strip. So this device was developed, and what we worked is the parameters that we have selected to quantify the intensity of the this dot was help us to design an assay wherein the intensity of the signal was found to be inversely proportional to the concentration of CA125. So with that, we developed this alpha assay, which we named as aptamer nanozyme lateral flow assay for CA125 detection. Okay, so here this was a competitive assay. Since gold nanoparticles, they were conjugated with Ca125, and the aptamer that was specific for Ca125 that was immobilized on the nitrocellulose membrane. So when the sample has a high amount of Ca125 in the test sample, then that Ca125 would compete with the nanoparticle bound Ca125, and less amount of nanoparticle bound Ca125 would be captured by the aptamer, and so a weak signal would be produced at the test spot. On the contrary, if the amount of Ca125 in the sample is less, then large amount of these nanoparticle bound Ca125 would bound to the aptamer immobilized at the membrane, and so we got a strong signal. So in this way, this was a competitive assay, and we were able to develop this assay in a strip format, and the substrate that we used here was DAB, and we quantified the change in the signal intensity using the developed device. And with that, what we found was that the limit of detection of that assay was 5.21 units per ml, and it was specific also. Another work that I would very briefly explain that we have done in our laboratory is again.
Dr. Shreeman, may I interrupt you, please? Can you please wind up quickly, because others are also waiting.
Yeah, sure. I'm winding up.
Please.
So this was regarding screening of DNA aptamers against VP2 protein of canine parvovirus, which again, out of various aptamers that we have screened, 6A aptamer was found to bind very specifically with these aptamers. So with that, I would just like to summarize the research interest of our lab, that we are not only working on affordable diagnostics, but are also exploring the application of nanoparticles in theranostics and antibacterial for antibacterial work. This is the group at our end. Our PhD students who are working in this direction. Pranav Tripathi has passed out. I would like to acknowledge all the funding agencies who have given financial support to us, like SERB, UGC, Ministry of Education, and DBT. For that, thank you so much for patiently listening to me.
Thank you, Seemaji. Sorry for not giving you much time.
No. I understand. Yeah, I understand.
Please bear with us.
Right.
Thank you very much, ma'am, for the such informative talk on immune-based therapeutic strategies with a special reference to aptamers. Thank you very much.
Thank you.
Now, I would like to invite Dr. Shivendu Ranjan, Department of Biotechnology, CSJM University, Kanpur, Uttar Pradesh, for delivering his talk. Dr. Shivendu Ranjan is working as an assistant professor in Department of Biotechnology at CSJM University. Dr. Ranjan has done PhD in Nanobiotechnology from VIT University, India in 2017. Dr. Ranjan is working on improving the transfection efficiency of plant miRNA in breast cancer cells. Dr. Ranjan has aiming to improve the transfection efficiency of miRNA in MCS7 for different genes. He is nominated fellow of the oldest active biological society, started in 1778, the Linnean Society, London. He is also the fellow of Indian Chemical Society, fellow of Bose Scientific Society, and fellow of Indian Engineering Teacher Association. Recently, he has also been the community member of American Chemical Society. I request Dr. Shivendu Ranjan to kindly deliver his talk on electrospinning and electrospraying for active compound encapsulation. Please, sir.
Thank you so much, Dr. Pallavi Singh Chauhanji, and thank you so much to Professor Tomar and Professor Anurag Jyotiji for inviting me here. And I would be pleased to give a brief about the electrospinning and electrospraying, mainly with respect to the active compound encapsulation and its application in nutraceuticals as well as pharmaceuticals. So, considering that the major audience is masters, so I'll be describing a process in bit, and then I'll describe what our lab has done and is doing now. So if we consider the nanofibers or the nano or electrospinning or electrospraying, so we have to consider a high voltage. It means a syringe pump will be flowing a polymer solution, and that polymer solution will be injected, and at the spindle end, means at the end of the tip, Stretching will be done, and this stretching, if we can visualize, the formation of the Taylor cone takes place. And obviously, due to the formation of the Taylor cone, the molecular vibrations also take place, and due to the molecular vibrations, the solvents get evaporated, and further, due to the stretching, the fiber, due to the high, with respect to the high voltage, the stretching, the nanofiber formation takes place. As I already have told, that electrospinning is a major way to engineer the nanofiber, and which can also be used to manipulate the fiber diameter, the mesh size. Mesh size means how much, the area of the mesh we need, so that can be manipulated, the porosity, the pores which is being prepared in the mesh, and that can be also be altered with the level of electrospinning, the time spent of electrospinning, or the speed of the electrospinning. The texture also can be manipulated by selection of the materials and opting the proper process, opting the proper temperature also maintains the texture. The pattern formation can be manipulated. We can have a sonic, ultrasonications at the time of injection, and then the pattern can be manipulated. The addition with substrate can also be manipulated. So overall, if we can see, electrospinning is a process of formation of a nanofiber. If we will have a diluted solution of the polymer, or if we have the very high voltage, these threads which are being produced in the form of nanofibers do breaks. And at the time of breaking, to make them stabilize, they might take a spherical shape. So at means two conditions given, very diluted polymer solution in high voltage, it may get electrosprayed, which is nothing but the same polymer. Instead of taking the shape of the fiber, they are breaking themselves and taking the shape of the spherical nano powders, or even the micro powders also. So electrojetting variables, what are the variables we could have to optimize the process or to optimize the electrojetting? Electrojetting, why I'm using a terminology here? Because it can be electrospraying or electrospinning, but we have to jet the polymeric solution in very high voltage. So electrojetting, there are majorly three variables, that is the polymer variables, the solution variables, and electrospinning variables, that is the process variables. If we consider the polymer variables, the chemical composition, the molecular weight, even the molecular mass distribution of the polymer, so the distribution, if the distribution is not proper, then again it may have an impact on the process, electrojetting process. Polymer solvent solubility is also matters a lot, because while making the emulsion of two different polymers, so solvent solubility matters here a lot, because if it is not soluble, it will not get ejected. Once it will not get ejected, it will jam the syringe. Solution variable, we already have talked that viscosity, the tension, the quality of the solution, the concentration, even like I already have given the example that concentration, if it is too dilute, then instead of spinning, it will get electrosprayed. The charge density matters a lot, the solution conductivity also. The process variable, if we consider voltage, again, voltage is a major player here, is a major hero to create a Taylor cone and to have a stretching and then taking a shape of the fibers or a sphere. Then spinneret shape, what shape of a spinneret we are giving to the solution flow rate, the drum speed. If we are going for the drum collector and we are collecting the nanofiber on a drum, then the speed of the drum also decides the fate of the shape of the mat as well as the strength of the mat. Because obviously the nanofibers will get collected on a rotating drum. The distance between the spinneret and the collector. Suppose the spinneret, that is nothing but a syringe end where the Taylor cone is getting formed. And if the collector is very far away, then might possible that the fibers which are being created in the high voltage will fall in between itself. So to avoid this, the distance between the spinneret as well as the collector also matters a lot. pH, humidity, temperature also affects the temperature, also affects the process. Because let us say if we are handling with the polymer which are very reactive to some part of the like ions, it can be H plus or anything which is altering the pH. So it again affects the again electrospinning and electrospinning process. The collector shape also matters a lot here, because instead of the drum, we can have a plate collector, and on the plate collector, we can collect the nanofibers or powders, electrospin powders or electrospin fibers on the plate also. Even in the liquid also we can collect, and we can have electrojetting, and we can collect the powders or fibers directly into the liquid, that is the solvent electrospinning. So collector shape matters a lot to decide all this fate. What will the final fate of the fiber or the mat or the mesh? Major challenges what we were facing while handling the electrospinning machine during the research. Few of the examples I'll be also discussing, mainly in the field of biomedical as well as the nutraceutical delivery. So we have faced the challenge of the multiple jets formed in the single nozzle outlet. Let me describe it here. Like the process, we have a single outlet, means single needle here, but to enhance the process, we needed to have a multiple jet formation. So we have filed a patent in WIPO, which has been published also, and it is available, well available online. We have tuned something with applying the engineering concept, and we have created a multiple jet in this tuning. I'll show you the device which we have created and patented on this. So after this tuning, all these multiple jets, means from a single outlet, multiple jet can be created. So the process has been enhanced by multiple folds. So this was a challenge which we were facing. Like spinning process was very slow, because in the the flow rate is in something microliter per second or per minute even or per hour even. So this flow rate, the slower flow rate, decreases the enhanced the productivity of the mesh. So to increase that from the single outlet, multiple jets has to be created. Addition of nanofibers on substrate. Many of the times we do create a nanofibrous mesh, but it might not get adhered on a different substrate on which we need it to. Let us first say we have encapsulated some of the drugs in these nanofibers, and after even encapsulation, we have a mesh, but we are not able to adhere it to the biomedical surface or the biological surfaces which we need to. So again, we have to give a surface enhancement. It can be chemical or physical. So we have to give it, tune the surface enhancement. Few of the examples I'll show in the next slides also, so that it will get adhered to the desired surface. Maintaining the fiber uniformity, this is still a challenge, because fibers, once after the Taylor cone formation, the fibers before reaching to the collector, it is in the dancing mode. So fiber uniformity and consistently uniform getting from the electrospinning, it is still a challenge, but we can reach by optimizing the process and all the variables which I have shown in the previous slide. So we can reach by tuning them and optimizing them. Single-sided drug coating on a mesh. So we had again a separate project in which on a hernia mesh we had to coat the drug, but it should be in the single side. So we have used the electrospray technique and we have coated the drug in a single side, and I'll show you the whole all that mesh and the coating which we have created. The product I'll show you in the next slide. The drug encapsulation uniformity. While making the different composites of the polymer, two different polymers, and keeping the diluting the drugs or dissolving the drug in that polymer, my possibilities are there. Few of the drugs, means the drug encapsulation are not properly uniform. So again, we should look into the solution factor as well as the polymer factor, and then we have to optimize drug encapsulation. Should be very it should be uniform enough. Because suppose we are giving a 1 centimeter square of mat to pharma company, and it should hold, if we are claiming that it is holding 1 mg of the drug, then all the mats, whatever we are creating, it should hold 100 mg. And these variations should not be there. Otherwise, this formulation will get failed in the clinical trial. Then again, coaxial electrojetting on a single axis, multiple jets can be, means two different polymers. Let us say coaxial, so this is the one outlet and outer outlet. So two different polymers can be electrojetted and it can be collected over here. So this is also possible, and it has a very good means application, mainly two different drugs we can coat, or two different drugs we can encapsulate, and these drugs will be separated enough with different fibers. So with a single formulation, we can have a delivery system of two different drugs. Problem in different types of multi-nozzles, it can, like, more volumes of polymer solution might be required. So then we should have optimum, the syringe which are getting, like, which are holding the polymers. So it should be large enough to hold much of the polymer, so that whatever we need, suppose we need a longer mesh, so our solutions container should be bigger enough. Now, these are few of the examples of electro spinning or electro spraying process, right from the cosmetics to life science and life science delivery system, packaged materials, and obviously in sensors. In agriculture, again, it has a vast application. Filtration, again, because we can manipulate the pore size, and the pore size can be manipulated in nano range. So filtration also, it has a major application. The industrial application, micro and nano electronic devices, electrostatic dissipation, many of the industrial applications are there. In tissue engineering, again, 3D scaffold, artificial blood vessels, tissue engineering, substrate formation, skin graft. So these are a few of the examples which in biomedical sectors which we are using. Major challenges is obviously, once we consider the industrial research, industrial R&D ecosystem. So major challenges is the material selection. Because while opting for the material, while selecting the material, we should always keep in mind that it should, if we are working for some of the pharmaceutical company, then our material should clear the clinical trial. It should not be toxic enough. Because so that this is again a challenging task, because randomly, if we are opting for the newer material, even not as a drug, but as a polymer or formulation material also, if we will opt for the newer polymer, which is not being used in pharma industries, so getting the approval will be challenging. So we should again have a proper R&D on material selection, the chemical composition, the blending, the copolymerization, the polymer molecular weight, mass distribution. These aspects we should consider. Solution variables, process variables, whatever have been discussed in the previous slide. These are again a major challenge which should be optimized, and the biology of the delivery sites, where we want to deliver. As the previous speakers have nicely highlighted, aptamers. So even we can tag these electrospray nanofibers or the powders with the aptamers to have a targeted delivery. So delivery site matters a lot. If we are not reaching, we have to tag them, and then it can, like, it should reach to the target. The strategies of the material selection is obviously based on application, and it should have the biofunctionality. It means it should deliver the drug in the biological microenvironment. It should be inert enough or active drug, two active drug molecules, because if the polymer will react during the encapsulation process, if the polymer will react to the drug, then again it will change the composition. So it should be inert enough that it should not react with the active drug molecule. The biocompatibility and the bioavailability should be well enough, and user compliance, FDA and regulatory approval materials. Reproducibility and possible large-scale production is again a major factor, which we should consider before starting the industrial R&D on nanomedicine, and before considering that electrodating process. Characterization of the drug encapsulation is again a challenge, because many of the drugs, even if it will get encapsulated in polymeric solution, and once after drying through electrojetting, it will be very tough to characterize them. And even if they are getting characterized in outside the body or outside the biological microenvironment, once the delivery, the pharmacokinetics and pharmacodynamics of these encapsulated drugs. So this is again a major challenge, and there should be we need to optimize the protocol. So it differs from lab to lab, because equipment to equipment, many of the factors, analytical techniques, it varies. So characterization is again a challenge for different labs. Limitations from bulk is advantage of the nanomaterial. We all know that how nano will be useful. So I'll not take much of the time on this slide, because this is very general, that surface area to volume ratio get increased. So we all know about this fact. A few of the points which I would like to discuss over here is fast dissolving drug delivery system, that is FDDDS. So these are possible with electrospinning as well as electrospraying techniques. These are few of the examples from our lab, which we have studied the nanofiber-based wound healing patches. We have formulated two different, we have two different formulations. One is dissolving the curcumin with the silk fibroin and adding the PVA as well as the polycaprolactone. So this is the one formulation, and again, instead of curcumin, we have taken the phenolic acid, and we have analyzed it comparatively. What we have observed that formulations with the nanofibers, the delivery was faster, and in both the cases, the healing was better than the normal control or the toxic control, but it is much faster in case of the curcumin. Now, this is the patent I was talking about. From the single nozzle, we have designed. So from the single outlet, we have given a different pores, and each pores will now act as a new outlet, and we can have multiple jets of the polymers getting ejected during the electrospinning or electrospraying process, and we can collect it to the collector. So the process has been enhanced a lot. So this patent has been published in WIPO. This is a face mask in which the initial days of the R&D, I was the R&D head of this company, Nanotech, which is the startup of IIT Kanpur. And we have optimized the nanofibers face mask formulations. Obviously, later on with the company have evolved. So they have fine-tuned the formulation and scaled it up to the industrial level. So at the lab scale, we have used the nanofibers of the PAN and PS6 blend, and then we have optimized it to reach the filtration efficiency of 95% with the, that you can say that the particulate matter of 2.5 micrometer. Now, after the product is successfully launched in the market, it has been well certified, well verified with different agencies, which has been displayed over the screen. But the major challenge in the initial days, what we have shown, we have actually faced, was the optimizing the formulation, and then further deciding that the addition to the different surfaces. And for this addition, because once we have the nanofiber, it should be sandwiched with two other clothes so that it will be safe enough if we are launching to the market, and filtration shall not be altered. So this adhesion was a major challenge. For that, we have used a cold plasma, and then adhesion was better. Different challenges has also been faced because we were working in a lab where the filtration efficiency measurement system was not there. So this is what a small equipment which we have designed indigenously to make we have a cylinder, we have made we have kept our nanofibers membrane over here, given a HEPA filter, and then we have measured the process. And then after that, we have measured the filtration efficiency. So this machine was also a challenge because we had a limited fund. So we have to develop an ecosystem in which, with the limited fund availability, we can establish the prototype. And this prototype has further been translated, and the mask is well famous in India. So this is another formulation which we have worked on. This is a formulation termed as Biotechnology, in which in the nanofibers, the nanofibers formulation, the drug has been encapsulated, and then the nanofiber-based drug delivery system has been created. So in this, vitamin D3 and even many of the vitamins and active ingredients, this formulation has been used to encapsulate and deliver the drugs. Few of the taste enhancers or the mouth fresheners also has been launched using this technology. Now, this is again a different project in which we have worked on, funded by Dr. Reddy's, and we have electrosprayed the model drug. The model drug Dr. Reddy's have given was the curcumin as well as the metformin. So our target was to encapsulate the curcumin and the metformin to have a formulation But the challenge was to use a polymer which should be of food grade. Why, once after discussing with Dr. Reddy's, so the question, why only food grade? They told once we will use the food grade polymers, that launching the drug will be easier, investment on the clinical trial will be lesser. So this is the learning. Learning means once choosing the polymers for the encapsulation should be well-established polymers. Otherwise, the company or the industry has to invest on the clinical trial, which obviously they will avoid of doing that. So instead of electrospray, again spray drying versus electrospray, I'm showing it here. So with the electrospray, the uniformity of the polymers means the globules or the spheres can be maintained. But in spray dried, what we have observed that the uniformity was not well maintained. So this is again a learning that which technique we should use for the drying of the emulsion or any of the formulation, polymeric formulation, which we need to. So this is a electrosprayed coating on the hernia mesh because the company needed to avoid the post-operative contamination, and they needed an antibiotic coating, but on the single side. On the single side, because they wanted it to be coated over the outer layer, because the outer layer is the only exposed layer through which the contamination do takes place. So our target was to put the drug only on the single layer. Another factor why the single layer coating was required, because to avoid the use of the antibiotic to reduce the cost, because if it will be coated double side, so that another side is not required for the drugs. So to save the drug delivery, to avoid the overloading of the antibiotic in the human body, so single side coating was required. So we have used the PLCL polymer, which is nothing but polyl-lactide, co-caprolactone, and then we have dissolved it in PHF and DMF. So the optimized one was 1:1 of the solution. So what we have observed, we have successfully coated this, and the delivery was successful. So this is again the nano emulsion and the drying. This research is always close to my heart because we have seen the nano emulsion, the vitamin based nano emulsion, and we can clearly see the TEM images that the vitamins are well coated in a thin film of the oil. So further, we have again emulsified it. It means a double emulsification. Again emulsified it with the polymers and then made it into dry powders, because these nanospheres are in liquid phase. So we wanted it to be in solid phase to reduce the water activity and to market it. So we have dried it with electrospray, and so it is still going on. So we are still struggling to scale it up. And these are the products which I have been involved as in R&D as R&D head of the company. So previous company, obviously, that is Eastpin Nanotech Private Limited. So this is nothing but the Sosa face mask and the Easy Mesh, the hernia mesh, what I have shown, the thin oral technology, that is the oral delivery, and the WIPO patent of the multi-jetting. And obviously, my major research domain is nanomedicine and nanotoxicology. So a few of my authored books, one on 3D bioprinting, one on food-grade nanoemulsions, and these are the edited books which are well available in the universities, in the library of the universities like Harvard, MIT, Caltech, Stanford, like that. So this is what I'm still recently have joined the CSGM University Kanpur, and I'm still trying to establish the laboratory with a focus to work on nanotoxicology, nanomedicine, and extended interest as nano antimicrobials or environment and agriculture nanotechnology. So by this, I end my talk over here. I hope I'm on time and I have delivered it well. Thank you.
Thank you very much, sir, for your brief insight on the emerging strategies in nanotechnology like electrospinning and electrospraying, with a special reference to active compound encapsulation. Now, I would like to request the chairperson of the session, Dr. Rajiv Prakash, to kindly sum up the session.
Am I audible?
Yes, sir.
So thank you. It's my pleasure to giving a summary or a final remark on this. But before going to the final summary or the remark over the session, what do you think? Should we take some questions or discussion as we decided at the beginning of this session?
Better, sir. We can open the forum for discussion and questions.
Wonderful, wonderful. I think our young students and researchers, they may like to ask some questions. And as I observed that from various speakers, some of the very interesting work and opportunities of collaboration. Certainly, there will be a lot of opportunities for collaborations. So my request is especially to our young researchers and students, come forward. No?
So you can put up questions in chat box. Or panelists can ask the question directly.
I can see a number of participants.
Sir, if you allow, I have a small question.
Yeah, Anurag, please.
Sir, thank you, sir. My question is to Dr. Seema Nara. Ma'am, you were mentioning about the application of aptamers for diagnostics. My simple question is that, can we design some aptamers which can specifically bind any kind of cell, prokaryotic cell, maybe E. coli, Salmonella, any bacterial cell, so that they can directly attach with them and we can develop some sort of lateral flow immunoassay system kind of thing, which is...
Yes, that is Dr. Anurag, that is quite feasible, and there are publications in that regard where aptamers have been designed against bacterial cells, whole cells also, and
Okay,
other kind of eukaryotic cells.
And ma'am, I think do they work better as compared to the antibodies, the conventional antibodies?
That one has to design the protocol accordingly, and extensive screening should be done to find out one such sequence which could bind very specifically to them.
Okay, thank you so much.
No, thank you.
Yeah, anyone? Okay, then meanwhile, I can ask one question. Dr. Tomarji, is okay? So, Dr. Ranjan, very interesting talk on especially electrospinning. So when this your polymer or whatever materials is coming out from jet, can you tell me that what is the temperature at that time?
So whatever we have ejected through the jet is at room temperature, but one polymer which I would like to mention here, it is the molecular weight was very higher. We were working on a Reliance project, and at that time it was very tough for us to maintain that liquid in the liquid phase in the room temperature. So over the syringe, what we have used the glass syringe, and we have to keep a heater from the both sides, so that even at the room temperature, the liquid will be, the polymer solution will be in the liquid phase. But at the time of ejection, obviously the temperature is at the room temperature, and because we have recently published one of our article in Food Journal, and the reviewer has also the same question. Because here the solvent evaporation is actually not damaging the nutrition, because it is not heat-based evaporation, it is just the energy-based evaporation. So it is well-established fact that due to the Taylor cone formation and the vibration of the molecules, the temperature is not getting increased. And that's the reason including the electrospray or electrospinning is advisable method in terms of pharma or nutraceutical delivery system.
Yeah, actually, this is very important point. And here I would like to tell our biologists and others, those who are working in enzyme and antibody. So this is a wonderful technique when temperature is not raising, and you can hold the temperature at room temperature. So very sensitive molecules also you can use.
Exactly.
They will not
Yes.
damage. So, and once the molecules are within the polymer matrices, so extra stability will come. So we have observed these things. Biomolecules, if we are encapsulated in polymers, getting a lot of stability. So this is indeed is very interesting. So Dr. Ranjan, this is really nice that you joined a new position, and this is good that now we can collaborate. And as I said, it's a very good opportunity for collaboration. Yeah.
Thank you so much, sir. Thank you so much, sir.
So Professor Dr. Sharma from IITR, my friend, is also here. So again, this is as I was talking on phone. This is really very good that the things as he mentioned, we should take care toxicological point of view, control point of view, and this is another opportunity with the Professor Sharma that people can interact and get.
I am in touch with Professor Sharma from long time, and we are discussing over the nanoscience and nanofibers when I was in Lucknow. So yeah, that's a good suggestion, and thank you so much.
And fortunately, Anurag has worked with us at IITR, and
cool.
He is an ambassador at MIT. I can say you can always directly come to me and interact. And moreover, this is becoming critical as per the OECD and the GLP requirements. So take the benefit so that collectively India can rise at the different forefront in the nanotechnological concept.
Yeah.
Sure, sir. Sure, sir. Sure. Thank you.
Thank you, Dr.
Thank you, Dr. Rajiv, for putting this comment, and all my partners. I can say not 1, 2, but the total team of 300 participants from this wonderful group is with us. So now our strength is into 300. And let us see how 300 into N, including, I would say, Pallavi to Pushpika, other students, all to go ahead in this direction. And we are there to support them. All the best.
Good. Thank you.
Thank you, sir.
So any more comments or suggestions from audience? I have triggered. No, please come out. So don't hesitate. This is the last moment, last time that you can discuss and start collaboration and all these things. See, in such type of conferences, this is the only opportunity that we have, is to listen our peers, to experts, and then have collaboration. So if something more, otherwise, should we conclude now this session?
Yes, sir. I think we should wind up, sir.
Yeah. Thank you, Professor Tomar, and once again, thanks to Amity and organizers for holding such a wonderful seminar and where we discuss a lot on, especially the biological sensing and biosensors development, and some very crucial and important points as we discussed today on aptamers and some injection techniques, as I was just talking, and control and toxicity control and regulations, as Sharma said, and many others discussed over the subject area. I may not be able to summarize because of the limitation of my field, but wonderful discussions and talk from here. And we have something to take away, and I think everyone is having something to take away. And my to youngers, to students, to young researchers, my suggestion is to keep it this live means have the discussions, connections, and collaborations. I'm thankful to co-chair for helping, and also Pushpika and Dr. Pallavi for helping us, and Professor Tomar, thank you once again for holding all this in such a successful manner. So please, it is to you.
Very much, sir, for summarizing the session. And I think it's a kind enough that you have spent so much time, and you are online from last more than 2.5 months, I think. Sorry, 2.5 hours. So it's a long period that you are listening very quietly and very calmly to each and everyone and giving the final comments. So thank you very much. And soon we shall be meeting in physical mode. I think something is missing out, and you are talking on online mode.
Yes, I agree.
Sir.
Thank you very much. So on behalf of AIB AUMP, I would like to thank all the eminent persons who added glory to this session. Now, as per the schedule, we are moving for the next technical session with the same link. Session 3, Applied Biotechnology.
Thank you.
Thank you, Professor Basilis. Thank you very much. We shall be in touch, sir, for future collaborations with your laboratory.
I appreciate the opportunity, Professor Tomar. And as I said, hopefully you have my contact details. So anybody that might be interested in contacting about the collaboration or about the journal, I will be more than happy to actually come in touch.
We shall be collaborating, sir. Thank you so much, sir.
Pleasure. Thank you. Have a nice day.
Bye, sir.
Hello.
Good evening, everyone. So we are starting with the next session, session 3, that is Applied Biotechnology. The applications of biotechnology are found in nearly every industry, from crop production to the vaccine development and opportunities.
हाँ मूट
Sorry about the interruption. So, applications of biotechnology are found in nearly every industry, from crop production to vaccine development, and opportunities can be found in both urban and rural areas. Biotechnology opportunities in the area of agriculture, food production and processing, environmental and healthcare can be found in locations throughout the state. On behalf of MIT University, Madhya Pradesh, Gwalior, I, Dr. Pallavi Singh Chauhan, Assistant Professor AIB, heartily welcome Chairperson of this session, Professor Dr. Rajesh Tiwari, Professor, MIT Institute of Biotechnology, MIT University, Lucknow. Co-chairperson of this session, Dr. Alok Patel, Biochemical Process Engineering Division of Chemical Engineering, Department of Civil, Environmental and Natural Resources Engineering, Luleå University of Technology, Sweden, and to all the invitees, presenters, and attendees. In the beginning, I would like to invite Professor Dr. Rajesh Tiwari, Professor, MIT Institute of Biotechnology, MIT University, Lucknow, for delivering his talk. Professor Dr. Rajesh Tiwari is currently working as Professor and Deputy Dean Academics at MIT University, Lucknow campus. Earlier, he served MIT Institute of Biotechnology as Deputy Director and Head since 2009 to 2014. He has over 20 years of teaching and research experience in the field of hardcore biotechnology. He acquired PhD from CIMAP, Lucknow, and was involved in research program of horticulture crops at CISH-ICAR. He worked with Chinese Academy of Science for postdoctoral program on plant transgenic development, especially on recalcitrant Chinese medical plant. He was selected by Volcani Center, Israel for Mashav International Young Scientist Program in year 2006. I request Professor Dr. Rajesh Tiwari to kindly deliver his talk on harnessing science, technology, and innovations for social benefits. Please, sir.
Before you proceed, sir, Professor Tiwari, I welcome you to this platform, and we are really very much grateful to you that despite your health issues, we know that you are so much inclined towards the research and teaching. that you sometimes compromise your health also. I know you are not feeling well, but still
Thank
you are.
you.
Yes, sir. Thank you so much, sir.
Thank you, Professor Tomar. How can I dare to defy your orders?
No,
Despite
sir. It's a request, sir.
not in good shape, I'm trying my best to chair this session as
Yes,
well as I will deliver my talk.
Yes, sir.
So I'm audible, I hope.
Yes, you are audible, sir.
So Dr. Alok Patel, co-chair of this session, Professor Dr. Rajesh Tomar, the convener, Professor and Head and Dean Academics of MIT University Gwalior, and my friend, faculty colleagues, Dr. Pallavi Singh Chauhan, thank you very much for such a nice introduction and welcoming me. Guests, dignitaries, delegates, and dear students, I welcome you all in this session of Second International Conference on Recent Advances in Biotechnology and Nanotechnology. My topic is harnessing science, technology, and innovation for societal benefit. In this session, we will talk about the impact of technological revolution on societal development, advantages as well as the perceived risk. However, during the talk, we will abstain from the technical aspect of the scientific development. So I'm not taking any technical part of any of the aspect that I have been touching. Rather, I would try to explain how the technology is impacting the marginal farmers, society, and also how it is, improvising our teaching and learning processes. So now let's, at the outset, though it is very, very basic, but since the students are there, they have to develop a connect to the talk. At the outset, let's have a generalized view on what is science, technology, and innovation. That will help us in developing the whole idea. Here, you can see the science, the creation of the knowledge, the technology, the application of the knowledge, and the innovation, the application of the technology. So this is all about science, technology, and innovation. And here we must make a difference between invention and innovation. Invention is the creation of a product or a process, and innovation is value addition to the existing process. So with this very basic background, we start our discussion. Tremendous growth has been registered in the field of science and technology. Technological advancement has touched every sphere of life, be it healthcare, education, travel, transport, defense. Every field is impacted by technology heavily. But this is the present scenario. With the help of this depiction, you can understand the disconnect between the common man and those who are dealing with science and technology. There is a huge gap in between. In fact, the science and society are still two different worlds working on a different platform. There is a disconnect that needs to be addressed. So I believe that until essence of advancement percolates down to the weaker section of the society, it would not justify its existence. So this would be a focal point of our discussion through this talk, and this is exactly what we will talk in next 30 minutes. Specifically, no technical part will be taken care of. Now, as we have perceived that the technological advancement couldn't benefit the weaker section of the society, because sometimes regulation put hurdles. Here is the example of BT cotton, BT brinjal, which is still revolving in the vicious circle of the government regulations. Sometimes it becomes so-called altruism of the big companies, put hurdles to cater the need of the poor. For example, the Terminator seat, and sometimes false fear of the public perception because of the communication gap between these two spheres. And the gap is so huge that if I ask you, the students are also sitting here, to name the 10 scientists who's working in your field, who has brought disruption in the respective field, I would not be able to recall it. You will not be able to recall it, I'm sure. One such example is, even most of us didn't know anything about the Serum Institute of Technology, Serum Institute of India before COVID-19 pandemic. Serum Institute of India, as we know, is the largest vaccine producer manufacturer, and that we knew after this havoc. This unknowingness is not your or my fault. This is because there are two different birds working on two different platforms. Scientists are happy in confined environment of the laboratory, and society is busy in meeting out day and to day requirement. These barriers are blurred in unprecedented situations like that we have been facing nowadays in form of COVID, which is sometimes feel that we are in us facing some science fiction. Even the large disparities has been recorded in income and social development of the society. That too, despite such a tremendous growth in all spheres, you can see the income and societal development of the society and the gap between developing countries and developed countries. One of the reason is use of technology in agriculture and industrial revolution is a different platform on both these two words. Hence now, this is a strong urge at the top level to bridge this gap by ensuring greater collaboration with the society on one hand and scientific communities on the other, to ensure proper promotion of science endeavor and effective utilization of fruits of innovation for the welfare of the people. Technology has advanced into many areas. We don't need to be again and again, emphasize on it. But gone are the days when we used to go to library for access of the catalog and making print. Now things are on click away. How often when we see a beautiful sunset, do you take out your camera? No. A small click from your phone is more than enough. Getting train tickets and air tickets are now just a click away. You can do it by yourself. And some things simple to notice on the surface of technology effects are that people no longer memorize the phone numbers. So these are, the technological advancement that has touched the life of a very common man. So to address the gap in sharing knowledge, resource, and society, and Equality United Nations Industrial Development Organization has started a technical cooperation program with a focus to identify the need and initiate a need-based research rather than starting just for reaching your CV, and also share this knowledge at each and every level with the society. That is very, very important. Only then society would be perceive the knowledge and would be reactive. And finally, to execute it at level where it is required. The model suggests analysis of the gap in technological requirement and the work towards decreasing them in systematic and sustainable fashion. Only then desired result would be achieved. So capacity building based on need, researching, sharing, and after execution. This is the best model that has to be adopted to bridge the gap between a common man and the scientific development. I mean, it is not that scientific development is not percolating down, but I will give you some of the examples in genetically modified crop, as my field is plant biotechnology, at how the technology is being grabbed and utilized by handful of the people, not by the the poor people, those who are actually required for elevating his living status. Now, to take all the sections of the society, Government of India has also initiated many programs through central agencies. I have taken few examples through which science is popularized and the gap is, continuously efforts have been made to bridge the gap. These are the different programs that the Government of India has initiated. Even in the UGC quality mandate, efforts are being made to create a platform to train the graduates so that they can facilitate interaction with the local community, understand the need of the community, and devise strategies to find workable solutions. You have aware the green and white revolution, but there are series of revolutions that shown perfect execution of science and technology advancement for societal benefit and improve the social economic status of Indian citizen over a period of time. Here is a list. I have searched it, and you can see there are many revolutions that have, bearing the fruit now. This vaccine development platform has extended beyond the imagination. Earlier, it took 10 to 12 years of development of vaccine, but now the vaccine can be developed in, has been developed in 12 months, COVID vaccine. This could have not been possible if the scientist has not sequenced the genome of the virus proactively by using the database. Sequence of the genetic material enables scientists to disrupt the vaccine development. And these genetic sequences are used to grow the live virus and study its behavior, including how it enters the human cell and cause diseases. And you can see how this vaccine development is impacted our day-to-day life. Now we are, feeling like, we are come out from a science fiction, and we are now somehow manage ourselves because of this vaccination program to, enter into the normal life. Some of the disruptive technologies that I would like to present over here. The past few years have been completely disrupted the whole scenario by registering tremendous advancement of the technology and its penetration to the lowest strata of the society. Not all innovations are disruptive, even they are revolutionary. However, some really impacted the life of a common man. I will briefly explain and expose you to some of these. I've taken example of some of the high technological innovation that has disrupted the existing one and impacted the society. Disruptive technology driven by the industry revolutions are adopted more fire to already volatile, uncertain, complex, and ambiguous world, impacting our life, our work, our environment, and even our future. Now, first technology is that I would like to mention is biosensor. A biosensor is a technical device used for detection of the chemical substance that combines the biological components with the detector. Biosensors has amazing power of detecting symptoms and before they convert into the disease. And how this impacted our life, even anyone can use the glucometer for detecting the blood glucose level, even the lactose sensor, alcohol sensor, gas sensor, immunobiosensors are now household items nowadays. Anyone can use it, buy it, and use it, and make the life very, very, easy. I have taken one example of Israeli startup Sensible Medicals creating high-tech waste monitor called SensiWaste. The monitor uses radar technology to detect the accurate amount of fluid in the lungs and liver and send data to their doctors via secure cloud technology. You can realize during the pandemic, most of the deaths are because of the lungs problem. And this type of startup, they would definitely help the society to get early treatment so that life can be saved. Another important is the blue blood of the horseshoe crab using the detection of the bacteria. The chemical gelates in presence of the bacterial endotoxin. And when a crab is captured, nearly 30% of the blood is withdrawn before it is released back. And the price is around $16,000 per liter. So this is another biosensor example, very interesting for the young researchers and the students. Now, the artificial intelligence has large capacity to revolutionize the medicine, therapy, agriculture, to the great extent, mental health of the society. AI technology, as we know, Machine learning and deep learning impacted the process of drug delivery, detection, confirmation, treatment, diagnosis, and by using the large scale genomic data. As we know, the agriculture is both a major industry and foundation of the economy. Factors such as climate change, population growth, food security concerns have propelled the industry into seeking more innovative approaches to protect the improving crop field. AI is steadily emerging as a part of industry's technological revolution. Some examples: companies are developing and programming autonomous robots to handle the essential agriculture tasks as harvesting crop at faster pace and then the human laborers. Companies are leveraging computer vision and deep learning algorithm for monitoring crop and soil health by capturing by drones and software-based technology. Machine learning models are being developed to track and predict various environmental impact on crop yields, such as weather conditions. Here is an example of Blu-ray technology, which is being used for the wheat control. The Blu-ray technology has been developed by a robot called See and Spray, which reportedly leverage computer vision to monitor and precisely spray the herbicide on the wheat. Thereby, it can eliminate the use of eliminate 80% of the volume of the chemical and also 90% herbicide expenditure. The John Deere announced its acquisition for the Blu-ray technology. So this type of technology, these are not very popular in our country, and if it is used, it may have a very big impact on the socio-economic status of the farmers. But these cannot be percolated down without the help of those trained people. The Harvest Crow Robotics has developed a robot to help the strawberry farmers pick and pack their crop very in a speedy manner. Harvest Crow Robotics claim that its robot can harvest 8 acres in a single day and replace 30 human laborers. So thereby the volume can be, a large volume can be handled in a very short span of time. The Berlin-based agriculture tech startup PET has developed a deep learning application called Plantex that identify potential detect and nutritional deficiency in the soil. Like same way, the analysis is conducted by software algorithm, which correlated particular foliage pattern and soil defects, plant pests, and the diseases. So the users are then provided with soil restoration techniques, tips, and other possible solution, and this would be a proactive, intervention before to address the issue before it become problem. Likewise, the Trace Genomics developed a system which used machine learning analysis, soil strength and weaknesses. The Sky Squirrel technology brings drone technology to vineyards. Aware satellite for weather prediction and crop sustainability. And FarmShot is another startup claim that its software can inform users exactly where the fertilizer is needed and can reduce the amount of fertilizer used by nearly 30%. So the precision farming that we are in, we are nowadays talking too much. By using these technologies, the precision farming outcome can be achieved in a desired manner. Now, every student is nowadays very fascinated by this 3D bioprinting technology, and which is of great importance nowadays. Even the brain, cartilage, skin, heart balls can easily be printed. So the students can explore the possibility of working in this field as well, because it has a tremendous scope. A progressive field of regenerative medicine is the tissue culture engineering. Regeneration of the improving skin, bone, muscles, up to entire organ is made possible by tissue culture engineering, and the development of either synthetic or natural human tissue in a laboratory is used for the patients with severe burn or falling organ, amongst other. The big data analysis, big data, one of the most important traits of the biological system. Data such as genome, RNA sequence, protein interaction, gene expression is called to generate a better understanding of the basic biological mechanism. So for computational biology students or bioinformatics students, there is a huge opportunity in this field. CRISPR is again a disruptive technology, clustered regularly interface short palindrome sequences known as CRISPR, a powerful gene editing tool, that biggest gene changer to hit the biology since PCR. So many of the proposed application involve editing of the genome of the somatic non-reproductive cell. As you know, the CRISPR are specialized strength of DNA. The protein Cas9 is an enzyme that acts like a pair of molecular scissors capable of cutting the strength of the DNA. Now, virtual reality replaces the physical world with computer-generated virtual elements. The VR platforms are already implemented to support rehabilitation of the patient to recover from various injuries. A BioLucid has developed a fully immersed 3D representation of the human body using virtual reality. The startup BioLucid U is capable of depicting the body in a healthy state as well as when infected with a disease. This solution holds significant promises for diagnostic medicine and training. Then the augmented reality is the virtual reality's cousin and make no pretense of creating a virtual world. The AR combines with the physical world and computer-generated virtual elements. Berlin-based Scopus Medical developed AR-based clinical navigation system. The startup technologies serve in the field of surgical education, planning, navigation system, and ENT and spine surgery. Another AccuVein is a handheld device that aims to improve accuracy by scanning a patient vein network and eliminating it. So these are the few examples where these high-end technologies are being used. And now we are each and every person in the society is well aware now what is antibodies, what is vaccine, but we may not be aware with this word edible vaccines. Now, very soon we need not to have a shot in arm. Rather, we have to take a banana or apple for being immunized very soon. So this technology still has some issues that has been addressed very soon, and for the children's, this type of edible vaccine would definitely hit the market in next 5 to 10 years. The molecular farming is now absolutely well standardized, and it is in use. Even the pharmaceutical proteins, industrial proteins, monoclonal antibodies, antigens, or edible vaccine, they all can be produced by using the cell in vitro condition. Now, this technology, hairy root technology, the hairy root cultures are largely used for production of secondary plant metabolites. And by using this hairy root technology, you need not to grow the medicinal plant in the field, in a big fields. Rather, just by culturing the roots in culture conditions.
Sorry, sir, I'm interrupting you, sir. You take care of a time. Your session may go up to 6 o'clock.
Yes, I'll finish it within 5 minutes.
We'll have to take care of others also.
I will finish it within 5 minutes. So that way, this hairy root technology is being adopted by various companies for production of the secondary plant metabolites, which are biologically active components. So these are the few pictures that I have taken from my work. This is a hairy root of one of the medicinal plant. This is another Chinese medicinal plant where the transformation has been done by my group. and we have developed and published a paper. It's another medicinal plant. We have raised the hairy roots, and we developed the culture and also got very high content of biological active components. There is some of the important biocontrol of the disease by rhizospheric bacteria, endophytes, fungi came into short, and nowadays frequent use by farmers. It is very cost effective. No threat of the environment, and farmers can generate it by own. Nowadays, the market is full of these products, and these are being used in place of insecticide and pesticides. Stubble burning, we are well aware, those who belongs to this region. Now there is a challenge to the students, those who want to work to solve this problem. They have to work for the bacterial consortium, which can be workable, which can provide a workable solution, and some of the mechanical seeders through which this stubble burning problem, stubble disposal problem can be addressed. Now, since the time is very less, but I will try to complete my task within 5 minutes. So impact of the transgenic technology on society and environment is huge, but there are perceived risks and ethical issues. As you know, as far as hunger is concerned, while the cost of the food is impacted by various factors, GMO plays an important role in keeping those prices as low as possible. It is estimated that the corn-based products would be priced 6% higher and so 10% higher if genetically modified crops were not grown. Okay, so this genetically modified crop has very high level of impact on the socioeconomic status of the marginal farmer as well. But however, due to the powerful expression of some of the activists and NGOs like casting of the hunger, demon seed, terminator seed, Frankenstein food, and their arguments are about values and ethics, which are neither absolute or nor universal. But this controversy surrounding the transgenic crops and polarize the society into a proponent and opponent. There are several myths associated to it. Say, for example, the adequate regulation is not there, negative impact on the environment, the technology is not known, and we are playing with the God. There are several constraints as well. Say, for example, if the artificial sweetener is promoted, then it will negatively impact the sugarcane industry. Another argument is if the terminator seed is promoted, then no seed for the following cycle for the farmers, and marginal farmers will not be able to use it. There are so many examples are there. Say, for example, the soya bean in the Brazil is grown by the wealthy farmers, but the government has, in 1998, the government has, developed a Roundup glyphosate herbicide-based herbicide plant, and with the aim to provide it to the marginal farmers, but the consequences were different. In the rainforest is cleared for more cattle pastures, and subsistence farmers who not grow soybean and whose crops has been severely affected by the drought received no benefit from the transgenic technology. So it was basically used by the farmers who has manipulate the system, but not by the subsistence farmers. There are several other, say, for example, the positive side is also there. The apomictic maize, which was developed by the Mexican government with the French scientists. This has, bring a ray in this, problematic area. So with this thing, I would sum up my lecture because I wanted to talk some more thing, but because of the time constraint, I stop it here. Thank you, Professor Tomar, for giving me this opportunity. I have tried to, justify my presence in the session. Thank you very much.
Thank you, sir. Session is in your hands.
Thank you.
Thank you very much, sir, for enlightening us with your talk on impact of technology and innovations on society and societal benefits. Thank you very much.
Thank you very much.
Now, I would like to invite Dr. Alok Patel, Biochemical Process Engineering Division of Chemical Engineering, Department of Civil Environmental and Natural Resources Engineering, Luleå University of Technology, Sweden, for delivering his talk. Dr. Alok Patel, Associate Senior Lecturer at Luleå University of Technology, is working as a researcher in Biochemical Engineering, Luleå University of Technology. He finished master degree in biotechnology in 2011 and joined as research assistant in foodborne infection surveillance. Next, he has been in several collaborations with the National Center for Disease Control, Ministry of Health and Family Welfare, Government of India. He joined his PhD in biotechnology from IIT Roorkee, India in 2017. His research interest is mainly focused on development of biotechnological process for conversion of organic matter to bioenergy, biofuels, and biochemicals. I request Dr. Alok Patel to kindly deliver his talk on bioprocessing of volatile fatty acids as building block for lipogenesis in oleogenic microbes for the production of nutraceutics. Please, sir.
Hi, this is Alok, and I would like to thank Professor Tomar for inviting me.
I welcome you to this platform, and we are meeting for the first time. I think we were not introduced earlier, but now we shall be meeting again, not only online, in the physical mode as well.
Yeah,
welcome, sir.
Of course. Yeah. So right now I'm going to talk about the nutraceutical production, and almost like Tiwari sir introduced, like how it benefits to the society. So I will finish my talk very soon, and like hope you will enjoy this talk. So currently I am working in the world northernmost technical university, and here we are just nearby the Arctic Circle, and it is almost like 136 kilometers from here. So there is no life exists from the beyond, and we are currently have like one chair professor, and there are two assistant professors in our department, and there is one important thing here that is northern light. If you come here and you can see every time like in the sky there is green things is dancing. So it's very interesting to visit our university. And recently I granted one European project from European Horizon 2020. It's Flaxi Green Fuels, and they granted me as a principal investigator to produce biofuels that is used for some jet fuels and like for shipping fuels. And this project is associated with Lufthansa Airways, Novenjive, and Ariel. And last year I also granted one major project from the Sweden Scientific Society, and they granted me to work on hydrophobic waste substrate, and it can be turned into some nutraceutical things. And this year I granted with another project that is directly linked with the CRISPR-Cas technology to boost the spelling content in the microorganism. So let's talk about my talk. So we are trying to reduce the animal sources for the nutraceutical production, because if you talk about the marine fishes, these are the main source of some nutraceutical fatty acids like DHA and EPA. And the shark liver is the most important component of spelling. And of course, like for the biolubricant, we use some petroleum refinery products in our diesel engines or petroleum engines. So this work is directly related to another project that is granted from the Swedish Scientific Society, where we produce BFAs from some waste material. And of course, like this is biorefinery process where hydrogen and methane is also produced. But here we are mainly focused to use the VFA for the production of some nutraceutical things. So let's talk about the DHA. What is the DHA? What is the fatty acids? So this is the fatty acid, like, and it has like methylene and carboxylate. But how we can differentiate between someone is like omega-3 fatty acid, someone is like omega-6 fatty acids. So if you count the first double bond located between the third position, if you count from the omega end, it is called omega-3 fatty acid. If it is first appear on the 6th position, so it's called omega-6 fatty acids. And like if we talk about the polyunsaturated fatty acid, there are more than 2 double bonds, and every double bonds like make some kinks in the structure of fatty acid. Of course, this is the secondary structure, but like it has it influences the physical property of the any cellular material. So these are the table with the omega-6 fatty acids and omega-3 fatty acids. So if we talk about the linoleic acid, this is the parent omega-6 fatty acids, and if we talk about alpha linoleic acid, this is parent omega-3 fatty acids. But our body can't like convert to the higher chain fatty acids like DHA and EPA, and we need to take it from the outside diet sources. But it can be like converted in a very small amount to the higher chain fatty acids. If you talk about like Omen has like high capacity to convert alpha linoleic acid to DHA and EPA. Almost like 21% of ALA is converted to the EPA by Omen, but this efficiency is reduced in man. So like Omen is always most powerful creature in the world, in the earth, or you can say in the universe. And if we classify the essential fatty acids, there are so many essential fatty acids like linoleic acid, alpha linoleic acid, DHA, EPA, but this is very difficult to get the higher fatty acids from the vegetarian sources. So it's very difficult to get these fatty acids, higher chain fatty acids for the vegetarian to take it from its diet. So it might be supplemented with other dietary sources. Like most important sources like oily fish and krill oil, algae oil. Algae oil is right now in the trend. So this is the pathway of omega-3 and omega-6 fatty acid synthesis. And there are so many elongations and desaturations work to convert the parent omega-3 fatty acid to higher chain fatty acids. But eventually the human don't have this kind of enzymes to convert from the parent fatty acids. So it must be taken from outside diet sources. And this is very important for our health perspective, because like it has several biological activity. Mainly the DHA and EPA is mainly stored in our retina and our gray matter of our brain. So it helps to enhance our vision. It helps also for our nervous system, and this fatty acid also help to synthesis of several lipid mediators that finally converted into various hormones. So if we talk about the food sources of this omega-3 fatty acids, so for the vegetarians, the flaxseed oil is like the most important source. If we'll take like 1 tablespoon, so it will provide like 7.3 gram of alpha linoleic acid. But if we talk about the EPA and DHA, they are mainly present in the marine fishes in high amount. But like marine sources are not like sustainable, or like it's not good option for omega-3 fatty acids, because like if the fish is resides in like marine environment where some pollutant will come, then it might be get like the fish will also have some amount of toxic substance. So it's not a sustainable way. American Health Association suggested that the 70 gram per like per gram linoleic acid is like the intake requirement. In the same way, if you talk about the other sources like alpha linoleic acid, the man required almost like 1.6 gram per day. So I don't think so like this fatty acid can be taken from like our regular food sources. So it must be supplemented anyhow from other diets. So if you talk about the market segmentation of like global EPA and DHA market, so almost like 43% is using as a dietary supplement, and they are also using in the infant formula. And if you talk about the global scenario of EPA and DHA, North America is leading for that one, And we'll say like, of course, if we targeted the marine fishes, it risks for our marine environment. So in our laboratory, like we are extracting this kind of fatty acid from the marine microorganisms. Marine microorganisms, mainly thyrestochitosis, that we are using in our laboratory. If you'll check about, like, this one is like the most like popular fish oil here in the Sweden supermarket, and I just run it by GCMS, and you can see the profile. There are so many kind of other fatty acid as well besides this DHA. And if we talk about the microorganisms that we extracted, the APA and DHA, so you can see the amount of DHA is higher even than the fish oil, and this is like lacking of other like unwanted fatty acid. So we can say like we can replace this fish oil from this microorganism. Okay, so let's talk about the squalene, another product of microorganism. Squalene is naturally synthesized by our cells that provides moisturizing to our cells. And of course, like the major source of this squalene is the shark, and almost 30,000 sharks they killed for a year, and that's why the IUCN listed it as like in the red list. So we need to jump to the another like sources for this squalene. And of course, squalene is also used as a vaccine adjuvant. So recently, like Swedish Research Council granted me as to produce this squalene from the microorganism. So even like there are so many other vaccine that is also used for like squalene as a adjuvant, like coronavirus, SARS-CoV. Even like there are so many like other COVID vaccine that is right now using SARS-based squalene. So this is not sustainable anyway. And they extracted the liver, only liver, and they just throw out all the SARS to the sea. So it creates a major environmental pollution. So why they choose shark? Because like shark has liver. Like if you talk about the anatomy of journal fish and shark, so there are major difference between their like biobuoyancy, how they like float on the sea surface or like the sea. So shark has unique like anatomy of liver. Almost liver has like 10 to 15% of its total weight, and liver is filled with the oil. Like other fishes, they have like some air sacs to float on the water, but sharks has liver, and liver is almost like made up of like 90% of squalene as a oil. So like they prefer to extract this squalene from the shark. So what we usually do in our lab, like we used like forest biomass to produce glucose to cultivate these microorganisms, and in a biorefinery way, like we just extracted the squalene, DHA, and rest of like unwanted lipid can be used as a biolubricant. So we are working on like the sustainable development strategy of the UNICEF. And this one is like our organosolve reactor. This is the world second largest reactor that we are working with to produce cellulose-rich biomass. So we have lignocellulosic biomass. When we feed this lignocellulosic biomass, all like hemicellulose and lignin will like goes to the liquid part, and we have cellulose-rich solid part that can be further converted into like glucose after enzymatic saccharification. So we have very good results for this one, and that this results we published in the ACS Sustainable Chemistry and Engineering, and like editor was very happy for this, and they selected as a special issue for this one. So I'm not going to talk about all results because it will take time for me to introduce all the things. So I can say like with this biorefinery process, we have high amount of DHA, squalene, and biolubricant that can be used as a sustainable material to replace the animal sources. And of course, we are right now improving our microorganisms by genetic modification. We are just overexpressing some desaturated and elongated in our strands, so it can be like produce more DHA and more EPA. And of course, like we have granted another project where we are working mainly with the CRISPR-Cas technology, and one postdoc is working with this topic. So he's mainly work on like squalene synthesis to overexpress in the thyrostropatites. And of course, hopefully we will get like good results with these things. So this is like my key publication that is already published in the Trends in Food Science and Technology, Chemical Engineering, and SCS Sustainable Chemical Engineering. So I can just say thanks for this topic. If you have any question, anyone can ask.
Any question from audience? You can post it in chat box.
Yeah, I can see like there's someone wrote like beautiful northern lights.
Yeah.
Thank you very much, sir, for your informative talk on nutraceutics. Now, I would like to invite Dr. Jyoti Priyadarshini Srivastav, Associate Professor at Government Gajra Raja Medical College, Gwalior, India. Dr. Jyoti Priyadarshini Srivastav currently working as a professor at Department of Pathology, Gajra Raja Medical College, Gwalior. She is interested in the research related to oncopathology and hematology. Her current project is on oncology. She is also interested in innovations in teaching methodology in pathology. Awareness about the cancers, voluntary blood donations are her mission. She is associated with the Mission Pink. She has a flair of writing both academics and literature. I request Dr. Jyoti Priyadarshini Srivastav to kindly deliver his talk on applications of nanotechnology in healthcare. Please, ma'am.
Before you start, I would like to welcome Dr. Jyoti Priyadarshini on this particular platform. And I should tell all the participants, panelists, that she is associated with us since the beginning of MIT Institute of Biotechnology. And whenever we organize any such program, she is part of it. She is a medical doctor, she is a researcher, she is an academician. So welcome, Dr. Jyoti.
Hello.
Please.
Good evening, everybody.
Yes, you're audible. Please go ahead.
Yeah. Big thank you to the Amity Institute of Biotechnology and especially Professor Dr. R.S. Tomar sir for giving me this opportunity and this platform. And I am really amazed to hear all the lectures and how the biotechnologists and scientists are working to provide all the health professionals with the newer diagnostic modalities, screening methods, and therapies that we are then offering to our patients. And this pandemic that we have witnessed, we all know how the diagnostic test, RT-PCR, be the vaccine, was made available and helped the patients. So,
can you make it full screen, Dr. Jyoti?
Yeah.
Yeah, it is now it is full screen.
Yeah, so I'll be speaking on advanced applications of nanotechnology in healthcare, which will be beneficial for the students of biotechnology. Nanotechnology, as we all know, is a revolution of the 20th century, and where we study extremely small structures. Nano, as the word originated from the Greek meaning dwarf, we study small or miniature sizes varying from 0.1 to 100 nanometers, at least in one dimensions. Nature, as we all know, operates at the nanoscale, and cells in the living organisms are 10 micrometer across in dimensions. Cell organelles are further submicron size, and we are acquiring an increasingly profound understanding of natural processes at this scale, which is enabled by new generation of scientific instruments. Nanotechnology has helped us to design devices that can either directly interact with or influence the behavior of living cells. Further, the understanding of biological processes on the nanoscale is a strong driving force behind development of nanotechnology. And what we saw in movies years back is coming to reality. As you can see in this slide, the scale of nanoparticles, which I've shown, that the three figures on the right, diameter of human hair, length of the housefly or a boy, they are macroscopic. You can see them through your open eyes, naked eyes, whereas those on the left are microscopic and nanoscopic structures, which we are now utilizing in the healthcare field to serve the diagnostic and screening and therapeutic modalities in a better way. Another figure, you can see how we deliver all the nanotechnology in medicine by means of liposomes, fullerene, dendrimers, etc. Now, how the nanoparticles are being used? Why? It is because the inherent physical and chemical properties that they possess. That is the electrical conductance, chemical reactivity, protein absorption, magnetism, optical effects, and physical strength. All these properties have made them ideal particles to be used in the healthcare system or the medical field. As you can see in this picture, a small probe or nanorobos, which is seen in the blood. You can see the RBCs, and these probes, they carry a camera, a capacitor, and a swimming tail, and help us reach those remote corners of the human body where, with the current modalities that are available, we are unable to do so. Another dendrimers, you can see another nanoparticle which is very beneficial in the healthcare. So coming to nanotechnology in medical field proper, it is the treatment of individual atoms, molecules, or compounds into structures to produce material and devices with special properties. It involves work from top to down, that is from macro to micro or minuscule levels by reducing the size. Various types of nanomaterials and nano devices are available. And through the nanomedicine, we are peeping into the future. In fact, we have stepped into the future, and the entire diagnostic, screening, and therapeutic modality in the healthcare system is experiencing a paradigm shift. So you can see the wider and broader applications of nanotechnology in medicine, starting from disease diagnosis, detection by imaging, monitoring, targeted therapy, mainly in cancers, theranostics, drug screening, gene delivery, nano implants, bone tissue engineering, and to treat the musculoskeletal diseases, drug delivery, therapeutics, very important, and nanosurgery by means of nanorobos. Now, why do we require this change? Change is the law of nature, as we all know. Our technology needs to be updated because there is increase in the lifespan and active life of the humans now. Way back when we acquired independence, the expectancy was somewhere around 32 years, and now the life expectancy is 75 years and above. Diseases of civilizations have cropped up. Initially, we were fighting infections, but now with time, be it zoonosis, autoimmune diseases, genetic diseases, they have come up and require special modalities, special investigations. Cancer is another threat to the human race. The way cancer incidence is rising, we have to develop technologies and take steps to combat it. As you can see, the pyramid of nanotechnology embraces diagnostics and imaging, treatment and prevention, and further regeneration therapy. Nanomedicine has given us a promising path for certain diagnosis, which was not possible earlier. And these diagnoses, they have made economical and friendly, easy and deep accessibility to the remote tissues in the body, enabling smooth and effective drug delivery with minimal toxicity, effective treatment, and also paves way for prevention of chronic diseases. You can see in this figure, versatile nanomedicine production. We have nucleic acid lipid nanoparticles, polymeric nanoparticles, liposomes, and emulsions, which pave a way to help the healthcare system. Richard Feynman, we would like to owe our thanks to this great physicist who, for the very first time in his very famous lecture, "There's Plenty of Room at the Bottom," pointed towards the existence of nanoparticles and their use. And he said that we can arrange the atoms at our will and use them accordingly. Liposomes were very first described in 1965 and nanoparticles in 1994. Since then, there was no look back, and first nanosensors have also come up in the year 2001. We began this journey of nanotechnology in medicine way back in 1990s when the first generation of nanomedical capabilities were developed, and since then they have undergone dramatically a rapid expansion. Another picture to just explain the broad stage that the biotechnology is providing in the healthcare by means of biomarker mapping, targeted therapy, molecular imaging, detection and diagnosis, gene and drug delivery. The utility of nanomedicine is that the nanoparticle drug delivery is by very small sized containers of vessels, and the molecular nanotechnology and nanovaccinology, they have potential future applications. In fact, we have started using them, and smaller non-invasive devices can be implanted in vivo. They have shorter biochemical reaction times. All these devices are faster and sensitive than the conventional devices and procedures that are available till date. Nanotechnology and antimicrobial therapy. We have nanocrystalline silver, which kills the microbes. Nanoparticles with nitric oxide gas are bactericidal, and these nanoparticles can cross blood-brain barrier and other natural barriers, which are the presently available several drugs cannot. And the treatment of resistant tuberculosis. India, being the capital of world tuberculosis, is now presenting with multi-drug resistant tuberculosis and HIV. We need nanotechnology to fight these diseases. Nanotechnology in imaging modality, we have quantum dots and nanocrystals, which has enhanced the potential of biological imaging. Also, the nanocrystals offer optical detection up to 1,000 times better than the available conventional devices used, such as MRIs, which we consider ultimate devices even today, MRIs and PET scans. Nanotechnology in drug delivery. So nanotechnology-based drug delivery is based upon three facts. First, efficient encapsulation of the drug so that we do not lose drug and our dosages are limited, successful delivery of the drug to the targeted region, and successful release of the drug. Effective drug delivery helps to make the drug cheaper, less toxic with minimal side effects, reaching to the deep and remote areas, crossing the biological membranes. Nanotechnology drug delivery system, we use nanoparticles such as liposomes, micelles, liquid crystals, and all, as you all must be aware, being the students of biotechnology. Nanorods, nanotubes, nanopipettes, nanobubbles, they are a big help to the healthcare professionals. Advantages of nanotechnology is that nanoscale powders of anti-asthma and analgesic drugs are easily and completely absorbed. This has been explained by Dr. Shivendu Ranjan in his lecture, and I'm really at awe to know that how these nano powders are available and are being used by the patients. The benefits of these nanoscale powders are that The conventional drug delivery systems are manually controlled, so there are chances of errors which can be totally avoided, and they are site-specific with minimal toxicity or no toxicity at all. Now, coming to the nanotechnology in surgery, that's a big change in the modality of surgery. The conventional surgery is now becoming outdated with the use of nanotechnology. Minute surgical instruments and robots are used to perform microsurgeries on any part of the body. And with minimal damage to the human tissues, the surgery is precise, accurate, and at the targeted area. Even we can visualize the surgical site much better with the use of the nanorobots, which are carrying the nano-sized surgical instruments and nano cameras. While using the diagnostic nano devices, we can perform the therapeutic surgeries or treatment side by side. So there is minimal delay in the patient receiving the treatment. Coming on to nanotechnology and its role in cancer. Cancer, as I told you, is the disease of the 20th, 21st century and is now become still the second number 2 cause of death after cardiac causes. So nanotechnology has brought about, we can call it as a small technology with a big impact. It blends diagnosis and treatment, and cancer nanotechnology is a novel interdisciplinary area of comprehensive research. Here I would like to say that this nanotechnology in medicine is a disruptive technology, and it is very desirous technology. It is a very diverse technology, and as we can see how it has unfolded several changes in the therapeutic means, and it has enwrapped the entire healthcare modality. Nanoparticles and cancers, they are also effective because nanoparticles have high surface area to volume ratio, and it also allows functional groups to be attached to nanoparticles, which can seek and bind to certain tumor cells, and they also target a particular tumor cell, they accumulate there, and the limitation of the conventional chemotherapy, which included drug resistance, lack of selectivity, and lack of solubility, are overcome by these nanoparticles when they used in cancer. As Dr. Seema mentioned, how they have developed for the ovarian cancers. Nanoparticles have the potential to overcome all the problems of the conventional cancer therapies. You can see in this picture how these nanoparticles, they target the selected cells only and cause minimal damage to the healthy cells. This technology will definitely bring a blow to the cancers, and it can be used both in vivo and vitro. Nanoparticles, they target tumor cells at initial stage by particles like dendrimers, quantum dots, etc. Here, for the cancers, I would like to mention that early detection of cancer is a key for cure in the future. Nanotechnology can be used to develop signature proteins to treat cancers. By means of this figure, the same thing I am depicting that how these nanoparticles, they only cover and attack the tumor cells, leaving the healthy cells, sparing them, and hence the morbidity and mortality in patients is reduced to minimum. You can see in this picture that in a blood vessel amongst the RBCs, you can see a bacterium-sized cylinder on the left top. This is the scale of minuscule nanoparticles that can reach to every nook and corner of the human body. Applications in various cancers, just to mention a few, how Abraxane is being used in lung cancer and pancreatic cancer. This is a drug which is albumin bound. Likewise, drug encased in liposomes have been used to treat Kaposi's sarcoma, ovarian cancers, and multiple myelomas. So these nanocrystals, owing to their physical and chemical properties of drug solubility, dissolution rate, improved absorption, and high bioavailability, serve our purpose. They have enhanced permeability and retention effect, which is very essential to kill the cancer cells, which are very sturdy, very resistant to the presently available drugs. Nanotechnology and tissue repair and tissue engineering. Many a times, the cells in the nervous systems, cardiac cells, that is the heart cells, as you know, skeletal muscle cells, once lost, cannot be repaired, cannot be regenerated. But by means of nanotechnology, we are able to perform tissue repair and tissue engineering and to repair several defects, which will go a long way in the future.
Sorry to interrupt, Dr. Jyoti, please keep the close track on time.
Yes, sir.
Yeah.
I'm just about to wind up. Nanotechnology and stem cells, all those scientists and biologists who are interested in the future prospects of human healthcare should divert their attention towards stem cells, because stem cell therapy is the future of the medical field. We have the nanotechnology has a role in genomics and proteomics. We are aiming at gene therapy, non-viral gene therapy. We do not want the viruses to enter, so we are carrying the gene repair by means of these nanoparticles. But along with it, we have certain emerging threats, because every technology has two sides of the same coin. While providing cure with complete genomes and proteomes for humans and all known pathogens, plus it provides cheap, highly parallel DNA and protein analysis and sufficient computer resources. It will be possible to spot any new pathogen almost immediately, but certain human and ethical issues are also coming up. And what this technology provides us, or what side effects will it have, it will only the time will tell. It has a great economic impact in the economic scenario. The market for product enabled by nanotechnology has reached up to $263 billion way back in 2012. So providing the highest growth rate ever. As I mentioned, there's nanotechnology in the field of medicine is a big paradigm shift. Entire classes of existing pharmaceuticals, such as chemotherapy agents and the treatment modalities, will undergo total change. They'll be displaced. So nanomedicine is a friend or foe? Of course, the time will tell, because we really need to study it when it is applied to humans. It requires a lot of research and team efforts. Of course, the conventional will be replaced by something which is novel and updated. Education will be key for nanotechnology implementation. And nanotechnology has a lot of revolutionary change in the human healthcare. It has replaced the need for transplants and artificial organs, which is nowadays going on a very large scale. And it will make the healthcare facility personalized, affordable, with improved quality of life. Again, the broad modalities that it is serving. And to conclude, I would just like to quote Richard Ismaili, who said, "Human health has always been determined on the nanometer scale. This is where the structure and properties of the machines of life work in every one of the cells in every living thing." So the practical impact of nanoscience on human health will be huge. Thank you so much for the patient hearing. Thank you.
Thank you. Thank you very much, ma'am, for providing your insight on various applications of nanotechnology in healthcare. Now, I would like to invite Dr. Sonia Johri for her talk. Dr. Sonia Johri is Dean Academics and Associate Professor in the Department of Life Science at ITM University, Gwalior, Madhya Pradesh. She is a postgraduate in biochemistry and was awarded her doctoral degree from Jivaji University, Gwalior, India. Her postdoctoral research was sponsored by Council of Scientific and Industrial Research, New Delhi. She has over 40 papers to her credit in journals of national and international repute. She is an excellent researcher and an external member of the Board of Studies in Biochemistry at Jivaji University, Gwalior. Dr. Johri has 23 years of research experience and has worked extensively in the diversified field of metal toxicity, anemia, hepatoprotection, oxidative stress, and biofuels. Though there is a vast diversification in her field, she holds expertise in toxicological evaluation of the metal toxicant and their therapy in the rat models, phytomedicine, oxidative stress, physicochemical characterization of natural product as well as in vitro and in vivo biochemical characterization. I request Dr. Sonia Johri to kindly deliver her talk on therapeutic approaches of watermelon, biosynthesis and characterization of selenium nanoparticles. Please, ma'am.
Thank you, Pallavi. Am I audible to all of you?
Yes, ma'am, absolutely.
Okay, thank you so much. And good evening, everyone. I know it's too late and the participants are listening very patiently since long. So I would not be taking too much of the time and just be a bit, I'll be summarizing the things. So we have basically worked on the therapeutic approach of watermelon rind against acrylamide toxicity. So in the previous discussion, we have already studied a lot about the applications of nanotechnology. So I would be proceeding with that, and I would just skip that part. And yes, now our study basically focuses on the biosynthetic methods of using watermelon rind with selenium nanoparticles. And this biosynthesis method reduces the selenite to the elemental selenium by secondary metabolites, which are present in the watermelon rind, and thus producing the selenium nanoparticles. And the rind, which is usually discarded, contains many nutrients and is also edible. So it has shown different constituents, that is the secondary metabolites, which I have already mentioned, such as alkaloids, saponins, cardiac glycosides, flavonoids, phenols, lipids, proteins, fiber, carbohydrates, citrulline, cellulose, and carotenoids. So it's so rich and it's usually discarded. So we have used the watermelon rind in our study, and this is the nutrient value, mineral content of watermelon rind, and we have found that it's a review rather, that it is rich in iron, which is 1.29, manganese 1.42, highly rich in phosphorus 135.24 mg per 100 gram, calcium, sodium, copper, zinc, magnesium, potassium, everything is present in the rind of watermelon. It is also rich in vitamin A, B, as well as C. These are the composition of the vitamins. So it is also rich in anthocyanins, which helps the immune system to function more effectively against viral infection. Coumarin is also present as an antioxidant that scavenges the free radicals. Terpenoids, tannins, which have analgesic and anti-inflammatory properties, is also present here. Now, we have tried to assess the selenium nanoparticles against acrylamide toxicity. Acrylamide toxicity is gaining a lot of importance these days because we are consuming acrylamide knowingly or unknowingly, because this acrylamide is present in the potato chips, which we are consuming in McDonald's. Many of our children are fond of McDonald's, and they are just taking burgers and the patties which is inside, and we see that the Maillard reaction, which is basically a reaction between amino acid and the reducing sugar. So this acrylamide which is being formed is very toxic and is rather carcinogenic to our body. So what we are doing is we have planned our study in a form that selenium intake can reduce the acrylamide toxicity. So selenium intake has enhanced the proliferation of the T cells, and higher selenium concentration in serum HIV patients has decreased viral load. So it's also antiviral. Selenium is meant to be very essential for brain, as its deficiency can cause irreversible brain injury. So what we have just aimed in our study is the synthesis of the selenium nanoparticles from watermelon rind, its characterization by UV visible FTIR, XRD, PSA, DSC, and TEM analysis of selenium nanoparticles. This is the medicinal value of the whole watermelon, because it's being consumed for constipation. Diabetic patients consume a lot of watermelon. Edema, skin fungal infections, kidney stones, liver dysfunctions, kidney infections, vomiting, gout, nausea, skin spots, heart disorders. So what we did in our study was the watermelon was collected from the local vendors, washed, and the rind was separated from the flesh, and it was chopped, kept in the oven, dried for 48 hours, and then a fine powder was obtained. And this was oven dried, finely powdered, and this was then kept in a desiccator. This is the watermelon rind which is being obtained after drying. Now what the aqueous extract preparation was done by decoction study. The fine powder of the rind was added in distilled water in the ratio of 1:20, and then it was kept in a boiling water bath for 4 to 6 hours, and it was further analyzed. So the aqueous, the solution was kept in a magnetic stirrer, and when the color changed, the sodium selenite was added to it. The watermelon rind and sodium selenite was kept on a magnetic stirrer for reduction in dark at 27 degrees centigrade. And then in the orbital shaker for 24 hours, and the change in the color indicated the formation of the selenium nanoparticles. And then it was characterized by UV visible spectroscopy. The selenium nanoparticles were analyzed on UV 1280 instrument to know the behavior, and the scanning ranges of the sample started from 200 to 1000 nm at a speed of 480 nm. Data taken by UV visible was recorded on the WinLab software. I'll just show you the figure and the FTIR. Yes, I'm just showing you the graph showing the selenium. This is the graph. Now, the FTIR analysis. FTIR analysis of selenium nanoparticles extract shows major absorption bands at 3278, 2921, 2849, 1638, 1535, 1395, and 539 per centimeter. And the bands appeared at different wavelengths. The absorption peak at 2921 due to the presence of the CH bond of the methylene. The band at 2849 is due to the presence of OCH3 bond of methoxy, and the strong bond at 1638 shows the presence of C double bond C, which is alkene. The another band at 1535 shows the presence of amine. And the short band at 1395 shows the presence of the OH bond, which is tertiary alcohol. And the band at 1238 indicates the presence of ether. And the strong band at 1030 indicates the presence of phenolic groups. So the FTIR spectrum indicates that the variation in the graph indicates the presence of secondary metabolites in the watermelon rind and selenium nanoparticles. Now the X-ray diffraction pattern. The X-ray diffraction pattern showed this figure, and we also analyzed the nature and the structure of the synthesized selenium nanoparticles. And the X-ray diffraction shows the sharp peaks, as well as it specifies the synthesized red selenium nanoparticle, which was crystalline in nature. And the nanoparticles are more effective and show more properties in different perspective, and is more effective in showing their properties in the crystalline form. We had given this study for SEM, but we have not received the results as yet. But we have gone into transmission electron microscopy as well. But the transmission electron microscopy results, images have been shown in this figure. You can see the figures, and then we have also conducted the particle size analysis. This is the particle size analysis. The particle size analysis was done by particle size analyzer. The laser diffraction studies reveal that the particle size obtained from highly dispersed mixture was in two areas on the scale of normalized particle amount. And the 0 to 10 particle diameter was 0.05 to 0.5, and the particle diameter found to be at 48 to 100 micrometer in range. So this was the study, and then we have also performed some in vitro studies to see the therapeutic effectiveness of the aqueous extract of the watermelon rind. Though we have not performed it with the nanoparticles, but we have done it only with the watermelon rind after acrylamide exposure, and the control depicted 98% cell viability. So we can expect that we get a better result with nanoparticles. And the lymphocytes were treated with aqueous extracts of watermelon rind at 6 different concentration ranging from 25 to 500 microgram per ml after acrylamide exposure on lymphocytes. And the treatment with aqueous extract of watermelon rind at a dose of 500 microgram per ml concentration showed maximum cell viability. This is the graph showing the viability and the different concentration. So finally, I conclude my study that the synthesis of selenium nanoparticles by using Citrullus lanatus rind extract, which is economically environmental friendly and non-toxic process, and the characterization of the nanoparticles was done by UV visible FTIR, DSA, and PSC. So this study can be processed further to come to further conclusions, and this will be done by the in vivo studies. Thank you so much for the patient listening. Thank you.
Thank you. Thank you very much, ma'am, for your informative talk.
Yeah, thank you so much for joining us, because it's an online platform and you are not able to get to direct link. I think there was some communication gap.
It was solved. It was
so finally we got it solved.
Yes,
That's
yes,
great.
yes.
Okay,
now, I would like to request chairperson of the session, Professor Dr. Rajesh Tiwari sir, to kindly sum up the session.
Thank you very much. It was a very exciting session, though we are running behind the time. First of all, Dr. Alok Patel, I congratulate you for such a exciting work and the presentation. Dr. Alok talked about the bioprocessing of volatile fatty acids and the nutraceutical production. He put light on how to reduce the animal source for nutraceutical production and how to use fungi, yeast, and other plant and other microorganism instead of the animal source. Dr. Jyoti Priyadarshini has beautifully explained the application of nanotechnology in healthcare system, how and why nanoparticles should be used, and how is it effective in the drug delivery and the specific drug delivery and therapy. She beautifully explained through beautiful pictures about the nanorobots, nanobots, and other nano devices being operational in healthcare system. She put light on threat of zoonotics. COVID is one of such viruses find ways in to the human from the animal, and we must, be very careful while dealing with the, breaking the species barrier. Dr. Sonia Johri, I have gone through your presentation, and this is very, very informative. Basically, if you see how a waste product can also be used for making some therapeutic use can be, think of the therapeutic approaches of the watermelon rind. She has speaked on this topic. She has spoken. We got to know about the watermelon rind properties and how it is effective in reducing the acrylamide toxicity and the synthesis of silane nanoparticle. So the students, those who are present here, they must get an insight and they also think of working in this field. With this, I believe that this interaction with such an expert, having proven track record, would enable young graduates to define the future path and also establish linkages for future growth. Thank you very much, and wish you all the best, and we will get in touch.
Thank you, Professor Tiwari. In fact, it is you who could manage the session, and it's I think now it's evening, and you are about to finish by 5 o'clock. But it's okay. In scientific field, it goes like that. 1 hour is equal to 1 minute. So
yeah, the discussion is important. That
right.
is,
time. Yes, yes. That margin we have, and we are still sitting in our offices. So thank you very much for joining us and summing up the session. Thank you, sir.
Thank you.
Thank you very much, sir. So on behalf of AIB AUMP, I would like to thank all the eminent persons who added glory to this session. Now this is the time to close today's session. And as per the schedule, tomorrow we will continue with the session 4, that is omics. Thank you very much.
Time.